Good morning, everyone. Welcome to our first 8 a.m. session. Thank you so much for being here. Give yourself a pat on the back. 8 a.m., I made it. It is my pleasure to introduce Professor Masami Yamaguchi, who is joining us from Chuo University in Tokyo. She's a developmental psychologist who studies face perception in infants. And honestly, I'm very excited about her talk. I really love her infant face perception work. So let's welcome Professor Yamaguchi.
Thank you very much. Yeah. Little bit more. I think more. Okay. Can you hear me? Okay, perfect. Yeah. Good morning, everyone. My name is Masami Yamaguchi. I'm a developmental psychologist. Thank you very much for my introduce myself, and thank you very much for my opportunity, such a wonderful special NIRS conference. So, because I have lost the opportunity to have a talk in person, especially for Japanese people in English. So, I'm very excited now. So, thank you very much. So today I will talk about infant face perception. Okay, I don't know. Okay, that's okay. I think it's something happened. No video, no PowerPoint. Yeah, we need the slides. Thank you very much. Okay. Today I will talk about infant face perception by functional NIRS studies. All of the experiment had been, some of the most important experiment had to be done. Dr. Megumi Kobayashi over there. Yeah. Anyway, at first, I'd like to introduce my lab and myself. Here is our recent neuroimaging article, longitudinal infant data to show processing different views of the faces. Our participants' age was under 8 months of the infants. In this study, we tried to take longitudinal data from 3 to 8 months, very hard work. And the results show that the processing frontal view faces is easy for the infants. Even 3-month infant can, I think, sorry. Okay. Only 3-month-old infant can do that. But processing profile view faces, I think the pointer. This is very hard for the infants. We have to wait the development. I think that this point is very critical period. So the over 6-month-old infants can do processing profile view faces, and there is a significant individual differences in development. Okay, our participants are aged at under 8 months old infants, and here is how to set up the infant experiment in NIRS. Okay. Sorry. We have an experiment for awake infant. We presented visual stimulus like this. So there is in front of the infant, there is a monitor. We presented stimuli, and under the monitor, there is a camera to check the infant behavior like this. And before the experiment, we check the infant head size and to prepare the probe setting. And the key important point is to attract infants to the monitor. So we presented animation, very attractive animation film for infants. And to avoid the infant's attention to the probe, it's also important. But sometimes clapping the hand, or sometimes very attractive toy to show the infant to not to attention to the probe, but for the something like this. And to keep attention not to the probe and to setting the probe is very important point. And we explain mother how to keep the infant, especially the hands. So softly hold the hand not to touch the probe. That is very important.
何しちゃったダメしちゃった。大丈夫ですよ。
So we explain mother keep the hands not to touch the floor and to keep the posture of the infants. During the experiment, to the not to touch the head. To keep the posture is very important for the experiment. Experiment will start. And our interest is to clarify neural basis of face processing in infants. In adults, the previous neuroimaging studies revealed that face-specific regions, such as fusiform gyrus and superior temporal sulcus, are preferentially activated by faces. We use very simple question. The right hemisphere advantage of the face processing should be found in infants. We have been testing this question by using NIRS, using special infant probe system to measure the temporal region or face areas. The advantage of NIRS over MRI is non-invasive and silent small equipment and requiring less stabilization of the head and the body. So it's suitable for measuring brain activity in awake infants. So we have 24 channels on bilateral temporal area in infants' brain. Most of our studies showed that infants' brain activity around STS areas. And our previous NIRS studies in infants revealed the pattern of brain activation to various types of face stimuli. I'm the developmental psychologist like to make so many kinds of stimulus like this, the inverted face and scrambled face and the side view face and mother face and different expression and also face-like objects. And our first NIRS study in infant is in face inversion effect in infants. Generally, in face processing, the inversion face is hard to discriminate individual faces compared to the upright faces. In this experiment, for 5 to 8 months infant, we presented 5 different familiar female faces for infant in upright and inverted. And results show that the significant activation is only for the upright face, but not inverted faces. So we found the inversion effect even in infants, and the activity of the bright temporal region is face-specific. After publication, our first study in NIRS in infants, 2007, and face inversion effect, we published many different kinds of stimulus, such as the side view faces for infant, and scrambled faces, and the point light display, that is the biological motion of the faces for infant, and mother's face, and also the lateralization in different expressions, and the gaze direction of the three-dimensional face recognition in infants, and the contrast reversal of the eyes. White eyes impair the infant's face processing. And not only for the infant, but for the ADHD children, and also anorexia children, that is eating disorder children in face perception. And not only the face, we also the color category perception, even in 7 months old the infants. And also the cross-modal association of auditory visual properties in infants, and mother face and baby face for the infant, and yawning, perception of yawning, and lateralization in sound symbolism, and so on. So these studies, especially face studies, we suggest that infant's temporal region is involved in face processing. However, it is an unanswered question. So which aspect of the information are processed in the infant's brain in temporal areas? In order to solve the question, we applied face adaptation paradigm in NIRS infants. Today, I will show our series of face adaptation studies by NIRS in infants. In adults, the technique of MRI adaptation is well established. This paradigm is a tool to examine the functional properties and the nature of representation in particular neural population. The MRI signal is referred to the attenuation of MRI signal is attenuated to the occurred by the repeated, like this, same identical faces presented repeatedly compared to different stimuli. So in this study, we use this neural adaptation paradigm in NIRS in infants. We conducted 4 experiments to the following question. The first is we check the infant's face adaptation paradigm in infant's temporal regions. The second is the face adaptation in size invariant. The third is pure invariant face adaptation, and the last one is face adaptation in invariant to non-rigid facial transformation. That is expression, different expression. So according to these 4 experiments, we can show infants variant and invariant identity processing of the faces in bilateral temporal areas. Here I will show the general procedure in all of the experiments. In all of the experiments, infant age is 5 to 8 months old age. In all experiments, we presented 2 test periods. The one is different identity test condition. The other is same identity test condition. In different identity face condition, we presented 5 different unfamiliar faces for 10 seconds. 5 different unfamiliar for the infant female faces. And in same identity condition, 1 identical unfamiliar female face presented repeatedly 10 seconds. The presentation time is the same, but the different identity condition, 5 different female faces presented in same identity, only 1 female face presented repeatedly 10 seconds. And in our experiment, before the test period, we set the baseline. In baseline, we presented non-face object. Non-face object is 5 different vegetables. 5 different vegetables were presented over 10 seconds. And we compared brain activation in bilateral temporal area. The experiment was continued to 10 trials. I will show the demo of the infant NIRS experiment face adaptation. The left is monitor. We presented stimuli at first as a baseline. 5 different vegetables were presented as a baseline at least 10 seconds. Then the different identity condition, 5 unfamiliar female faces were presented 10 seconds. And back to the baseline, the over 10 seconds, the vegetables are presented. And you have to wait. And in same identity condition, same face, same identical female face was presented repeatedly for 10 seconds. The order of the same identity condition and the different identity condition was counterbalanced between the participants. In experiment 1, we tested the infant's face adaptation to identical face in bilateral temporal region. In experiment 1, the stimulus like this, the participant's age was 5 to 8 months old infant. And in this experiment, in different identity condition, 5 different female faces, the frontal view faces were presented for 10 seconds. And in same identity condition, one identical female faces. All of the faces are unfamiliar for the infant. Unfamiliar female face presented repeatedly 10 seconds. And here is the result. The figure shows the time course of oxyhemoglobin. The vertical axis represents the oxyhemoglobin changes. The horizontal bar represents time. The 0 is the beginning of the test period, and 10 is the end of the test period. And the red line represents the different identity condition, the death score in different identity condition. And the blue lines that represent the death score in the same identity condition. We can find graduate activation in bilateral temporal areas, especially for different identity condition. In the strong activation for different identity condition, and attenuation, smaller activation for same identity condition. And there is a significant difference between two conditions. So we can say 5 to 8 months infant shows attenuate to the same identity adaptation in the bilateral temporal areas. And this is a significant activation channel, significant difference channel between same identity and different identity in this experiment 1. So in experiment 1, we can find infant face adaptation in bilateral temporal areas by using NIRS. But we can find face adaptation, but there is one possibility. The adaptation is just the same image because we presented same female images all of the experiment. So just for the image, not the face. So in experiment 2, we set the different experiment that is face adaptation view in size invariant manner. So in this experiment, we change the size of the faces. So we examined whether infant show size invariant face adaptation in experiment 2. In experiment 2, we in different identity, 5 different female faces were presented in different size. And in same identity condition, one identical same female face presented in different size, 10 seconds. And here is the result. We also find the significant difference to condition. We found strong activation for different identity condition, even in different size. And smaller activation for same identity face, even in different size. So we can say 5 to 8 months infant showed face adaptation in the bilateral temporal area, despite of change of the face size. And this is a significant channel from experiment 2. And in experiment 3, we step forward. We tested the infant face adaptation in view invariant, that is face representation in view invariant manner or not in this experiment. In this experiment, we presented in different identity condition, 5 different female faces in different views. And in same identity condition, one female face presented for different views. So we compare the activation between two test. And in this experiment, we set 2 infants group, younger one and older one. Younger one is 5 to 6 and older one is 7 to 8. The reason why we set 2 groups in infants is I already shown the recent study in infants. The infant processing different views of the faces. The processing frontal view faces is easier for infants. So even 3 months infants can do that. But processing profile view faces is harder for infants who have to wait their development. So over 6 months old can do that. So in this experiment, we set 2 different age group. Younger one, 5 to 6, and older one is 7 to 8. And here is the result. For the younger infant, 5 to 6 months of the age, there's no significant difference to condition. That is, there is no attenuation effect in the younger infant. But so for 7 to 8 months old infant, we found stronger activation for different identity phases, even in different views, and attenuate small activation for the same identity phase in different views. And there are significant difference between two conditions. So we can say the only 7 to 8 months infant show the phase adaptation in bilateral temporal area under the various viewpoint. And here is a result of significant channel of experiment 3 to a wider match channel in the right temporal areas. And the last experiment investigate why the infant show face adaptation invariant to facial expression. The facial expression also different face type, and the identity also different face type. Maybe infants confused. So we tried to infant show the face adaptation, identity adaptation invariant to non-rigid facial transformation. In this experiment, we set two age groups, the younger one and the older one. And we present in different identity conditions, we presented 5 different female faces in different expression. So this expression is only for the positive expression, not so negative one. Smiling, neutral, puffing cheek, opening the mouth, looking the mouth like this. And in same identity condition, same female face presented in different expressions. And we compare these different identity and same identity in the activation is here. The result of the younger infant, 5 to 6 months old of the age, there's no difference between different identity condition and same identity condition in different expression. But older infant, 7 to 8 months old infant showed strong activation only for the different identity condition, even in different views. And small activation, and that is attenuate same identity, even in different expression. So we can say only 7 to 8 months infant show the face adaptation in bilateral temporal areas despite facial expressions. Okay. And this is the significant activation channel in experiment 4. And the summary of this experiment. So using the neural adaptation paradigm, we found face adaptation effect even in infant, even in NIRS experiment. And the development of facial identity processing in infants. From experiment 1, we found face adaptation effect even in infants in bilateral temporal regions. And in this region, the face adaptation is size invariant manner. So infants' face adaptation is with no effect of size. So this is the 5 to 7, 8 months old infant. And we can see the development face adaptation view invariant and invariant to non-rigid facial transformation is only for 7 to 8 months infants. So from experiment 1, the only 7 to 8 months infants showed face adaptation effect in view invariant manner. So we can say view invariant representation of face would develop between 5 to 6 months of the age. And from the experiment 4, we found face adaptation effect invariant to facial expression, also only for 7 to 8 months infant. So invariant to non-rigid facial transformation identity processing, we have to wait between 5 to 6 months of the age. This is the summary of the significant channel for all experiments. The larger circle shows match channel, 3 channels here, and the smaller one is only 1 channel activation. So we can see the match channel and larger channel in right temporal regions. And from our NIRS studies in the development of face perception during early infancy, we found at 5 months, we found infant face inversion face effect in right temporal region. And from 6 months, lateralization for facial expression was found even in infant. And holistic processing and biological motion and special activity for mother's face is 7 months. And the processing profile view face is, we have to wait the older infant, 8 months old age. So, and in this conference, we will have 2 posters from my lab. The first one is Dr. Megumi Kobayashi's new studies. She showed not only the face, but also the body area in infants. That is, we found body area in infant and development of body area and face area during 5 to 8 months of age. And the second one is my graduate student. She showed the cartoon face image brain activation. She showed the cartoon image of a female face. It's face specific, but very attractive cartoon character image. It's not infant brain activation. So thank you very much for the kind attention. That's all. Thank you.
Thank you very much for that wonderful talk. We have time for a couple of questions. If you have a question, you can come up to one of the 2 microphones. Well,
additional one is our future study.
Yeah.
Our future study is COVID-19 effect in development. In Japan, is everybody wearing the mask. All 3 years, everybody keeps mask wearing. So very amazing effect in Japan. So I interviewed some of the newspaper from about the Japan. It's specific that everybody keeps the masks. So I think this is some of the effect for the development infant or some adulthood, everybody. So we will try to do this effect by me. Thank you.
Well, I actually have a quick question. I'm going to be completely selfish here. I have 2 questions. One, I've always been very inspired by your choice of the active baseline of the vegetables. I'm going to ask, it actually inspired some stimulus choices that I've made myself, and I'm wondering where that decision came from.
So sometimes I have the non-face of the just circle or something, but in this study, we try to do the bilateral temporal area, especially the face specific activation. So we compare the set very similar to the face, the very many colors, the many different kind of the object, but not the face. Such object we choose as a vegetable, and some of the experiment, the recent the Megumi experiment is not the face, maybe the butterfly. Yeah. Anyway, the colorful one, we use colorful faces, the colorful one, and no face object is important for us. Thank you very much.
Wonderful. Thank you very much. Oh, sorry, we've got one question here.
Really interesting talk. Thank you. I was just wondering if you've looked at adaptation to expression across different identities as well as adaptation to identity across different expressions.
Actually, the facial adaptation of the facial expression.
Yeah.
So sorry, what?
So I'm just wondering if you've looked at like babies adapting to the same expression on different identities instead of the same identity with different expressions. Yeah.
This is a very important one. It's you mean the same expression, different identity.
Yeah. Exactly.
I think it's also something happen. The next step is the next experiment, I think.
Yeah.
Yeah. Thank you very much.
Okay, very quick question.
Really quick question. That's a wonderful talk and an area that I don't know much about. But as I was watching the clip of the child reacts. The child's facial expression reacts to the facial expression they're looking at. And I guess my question would be, if you did video analytics on the child, would you do better or worse than fNIRS as determining what the response to the images is? Thank you.
That's right. Yeah. We have many experiments for babies for the color perception and the face perception, but by infants' behavior, we can, what experiment now running, we can check from the infant's behavior. The infant is like this, is express their expression for the faces, not for the non-face object. But we cannot stop this expression for infant. We cannot ask the infant to keep normal expression. So that is the one problem. But the infant is naturally cannot stop their expression to seeing the face. Thank you very much.
All right, we're going to have to unfortunately switch to the next talk. Sorry about that. Let's thank Professor Yamaguchi one more time.
Thank you very much.
All right, and our next speaker is Ana Blasi Ribera.
Thank you very much. So good morning, everyone. And thank you to the organizing committee for the opportunity to present the work that I'm presenting today in the conference. This work has been so I'm going to talk about the fNIRS, the analysis of data that we collected in the context of the brain imaging for global health study, the BRIGHT study. And this data is from our fNIRS task, habituation and novelty detection, which we call HAND as well. The data analysis has been possible thanks to Borja Blanco, who is probably here in the audience. He's presenting 2 posters at the conference. If in case you want to speak with him, he is going to be at those 2 posters. So in recent years, several groups, including ours, have been presenting data, neurodevelopmental data from low and middle income countries, because this is very important data, because at the very early stages of life, very early years of life, there's a particularly vulnerable window for development. There's a large proportion of infants living in low and middle income countries that fail to reach developmental milestones, and a large proportion of those infants live in sub-Saharan Africa. So in the for over 70 years, the Medical Research Council has set field stations to study the impact of undernutrition and environmental and socio-economical factors on infant growth. They have implemented strategies to try to curb the growth faltering and stunting of those infants. And they have collected a lot of data on growth by age. Those charts tell us not only characteristics of the population in these areas, but they can also allow us to quantify discrepancies from WHO standards, and also quantify the efficacy of those strategies to try to curb growth stunting. So the question is, can we do the equivalent with functional brain imaging? So this is one of the motivations of the brain imaging for global health study, to come up with such charts and information. This is a collaborative effort of several centers in the UK. It's collected data from over 200 infants in the Gambia and 60 in the UK. And it's not only neuroimaging data, it's also behavioral data and physiological data. There's a follow-up study to the BRIGHT study, which is called BRIGHT Kids. And the Gambian cohort kids were asked to come back for one more session between 3 and 5 years. And this data has been collected. And there's also another study, the Indigo study, it is a micronutrient supplementation intervention study that Sam McCann is presenting as a poster. So there are several posters actually linked to the BRIGHT project. So I have a poster in the next session with another analysis on this data. Chiara Burgelelli is presenting functional connectivity data from BRIGHT. And then Liam Collins-Jones is presenting the cortical mappings of our NIRS arrays. And Sam McCann, as I said, is presenting the Indigo data. So why do we study, why do we incorporate a habituation and novelty detection task into our study? So first of all, habituation is the repetition suppression, the suppression of the response after several presentations of the same stimulation. It is associated with discrimination, selective attention, learning, and memory mechanisms. Novelty detection is the response recovery with the presentation of a novel stimuli, and it is associated with identification of novel information, integration of this novel information, and it enables exploration. So those are all elements that are integral to learning, and they have the potential to have an impact on neurodevelopment. So this is the stimulus paradigm. While the babies are being entertained by an experimenter, there's a sentence that is played on the background. This sentence is presented 25 times. Here we've grouped them in epochs. So the first 15 trials are familiarization trials. Then it's a female voice speaking that sentence. And then there's a switch to a male voice saying the same sentence for 5 trials. And then 5 more trials with the initial female voice. And this is what we're expecting to see. So this is data that we published in 2019. This is from our UK cohort. And what we see is that with the initial repetition, so the familiarization trials, during the familiarization trials, there is the decrease of the amplitude of the response. When there's the male voice, the novelty, there's a recovery of the response. And when the same female voice that initially was played returns, there's also a return of the decrease in the amplitude of the response. So this is what we see in our UK participants. So the data that I'm going to present today is only from the Gambian cohort. And these are the numbers of the valid data sets that were included for each time point. So I think I failed to mention that, but this is the longitudinal study, and the data that I'm going to present is from 5 months, 8 months, 12 months, 18 months, and 24 months. So as I said, I analyzed this data with Borja's code for using the threshold-free cluster enhancement method. This method starts with a statistical map. So you get you calculate one t value for each sample. Then you transform this statistical map into a TFCE map with this formula. The values E and H control and give more weight or less weight to the width or the height of these TFCE values. So the idea here is to enhance the clusters and make the decision on where the threshold is, the threshold that defines what's a cluster and what is not, make it less sensitive to small variations in the data. The next stage is to create an empirical distribution of max FTE values and then infer statistical significance from it. So here I used 10,000 permutations and a p-value of 0.01 for statistical significance. So the first thing that we looked at was where are the infants responding, or how are the infants responding to our first set of familiarization, so to the epoch familiarization 1, that is the first 5 trials that are presented. And what we can see is that at all age points, there's a consistency of the regions that are activating. In these regions, this is more in the posterior section of our arrays. At the older time points, we see that the channels that are activated are more into the anterior part of this region that is activating, and the younger ones are activating a little bit more to the posterior part. So these channels are registered into middle and superior temporal regions, as Liam's poster shows. And it is consistent with prior analysis that we did, where we found ROIs. Here highlighted, the triangles represent the ROIs that we found in a prior analysis. And we based that prior analysis into these ROIs. So I'm going to show you now examples of the time courses and the activation that we see to FAM1 versus baseline in this left hemisphere. So this is at 5 months, and we see a very localized activation. And then we see here 24 months. By the way, I looked at oxy and deoxyhemoglobin to look at the condition versus baseline activation. And then next we looked at FAM3. So that is the last 5 trials of the familiarization period, FAM3 versus baseline, and then the presentation of the novel stimuli versus baseline. And so what we see as the number of trials presented increases is that there's a consistency in the channels that are activating at all age point and for all epochs. So there's this region is the one that responds to these type of stimuli. And then we also see that as the number of trials presented increases, the number of channels being active decreases. So next, we're going to look at the condition contrasts. So for habituation, we looked at FAM1 greater than FAM3. And for novelty, we looked at novel versus FAM3. So, and we looked at this contrast within these regions where we found FAM1 active. For the condition contrasts, I only looked at oxyhemoglobin. And what we saw habituation at all time points. So this is an example at 8 months with oxyhemoglobin at FAM1 in red, deoxyhemoglobin at FAM1 in blue, magenta for oxyhemoglobin at FAM3, and cyan for deoxyhemoglobin at FAM3. So this is at 12 months, and this is at 24 months. So what we also found was that at 5, 8, and 12 months, the responses continued to decrease, even with the presentation of the novel condition. And this is an example of what we see. And then we only picked up real or something similar to what we would expect in novelty at 18 and 24 months. It's not massive, there's not a lot of channels, but it is there in just a few channels. So this is a summary of what we have found. So I've calculated the percentage of channels that show contrast significant from the number of channels that were active at FAM1. And what we see is that there is significant habituation at all time points in most channels. There is no novelty detection at the younger ages, and only the novel presentation is greater than FAM3. It only starts appearing at 12 months. 12 months is not even fewer channels at 24, but then at 18 and 24, there's more channels. And finally, there is evidence of continued habituation at FAM3 versus novel, and this is something that we found in these prior analyses that I mentioned that we did based on ROIs. So, as conclusions, we can say that there's a persistence. So in this group of Gambian infants, there's a persistent of the habituation response. And a delayed onset of novelty compared to what we had seen in the UK. But this discrepancy may be due to several factors. One of them is that these infants grow up in a very social environment. They are carried on the back of their mums, while they do their business, their usual daily life. So maybe that's one of the factors that influences these responses. So there is also a response that is consistent from our ERP study that Laura Cates published in her image in 2020. It is from ERP study, the same cohort. And she also found this sort of diminished, the continuous habituation and diminished response to novelty. So the TFC value method has allowed us to find these channels with novelty that we couldn't find in the ROI-based analysis. And that's so it has provided a lot more information that we could look at with other methods. So a better understanding of this timing, of the timing and the space location of this type of activation might help us find these markers of neurodevelopment that we are looking for to produce those charts. So this project is very labor intensive. So we have a wonderful, huge group of people working on it. This is the list of present and past collaborators and researchers and people working on the project. And last but not least, of course, this wouldn't have been possible without the wonderful families that have contributed to the study. So that's the end of my presentation, and I'll be happy to answer any questions that you have.
Great. Okay, quick question from Judith while I set up the next talk, if the next speaker could please.
Thank you very much, Anna. So I have maybe a comment and then a question. The comment is whether your stimuli are linguistic. So I was wondering whether you correlated individual responses, whether they habituate or detect novelty with the linguistic development of the babies.
We are not yet working. Well, so we were starting to analyze the data at infant level, and no, we haven't done that yet. But that is something that we need to do that in the future.
Yeah. Thanks. We need to switch speakers. Sorry. All right, next speaker. Thank you so much, Anna. Wonderful. And so our next speaker is Alejandra Russer, who's joining us from Université de Montréal.
Good morning, everyone. So my name is Alejandra Russer. I'm a PhD student at the Université de Montréal under the supervision of Anne Gallagher. And it's my pleasure to present you today the findings of my doctoral studies on the cerebral reorganization in children with frontal and temporal lobe epilepsy. So to begin with, why is it important to study pediatric epilepsy? It's because it affects 1% of all children under the age of 16. So it's the most common neuropediatric disorder. Epilepsy is a cerebral dysfunction that is characterized by an hyperactivity or an abnormal synchrony in neurons, which leads to epileptic seizures. So more precisely, we are interested in frontal and temporal lobe epilepsy because they are the most common subforms of epilepsy in children. And there's a number of studies that report the cognitive difficulties in these children. There are some difficulties that are very typical for frontal or temporal lobe epilepsy, but what they both have in common is that these children often have impaired language functions. And this is why we are interested specifically in language processing. There are a lot of studies that also investigated how the language processing in these children is altered. So one group of studies was looking into the atypical localization of language processing. So when we look at atypical, we also have to know what is typical. So typical language representation in children and in adults as well is in the left hemisphere. So the left hemisphere is dominant for language processing. In children with frontal and temporal lobe epilepsy, the atypical representation means that, for example, the right hemisphere is dominant for language processing, or that there's even a bilateral representation. There is another group of studies that is looking into network characteristics, so how networks differ in epilepsy patients compared to healthy controls. And what they found is that there is a reduced task-related functional connectivity for language processing in children with epilepsy. When we look at the resting state networks, the functional connectivity can actually go in both ways. So there's increased and decreased functional connectivity in children with frontal and temporal lobe epilepsy, depending on the different networks. And finally, looking at network characteristic can also mean that we are interested in networks efficiency, segregation, and integration. So these are the graph theoretical metrics. And there's also several studies that report differences in children with frontal and temporal lobe epilepsy compared to healthy controls. So despite all these individual results of studies that report there's differences between children with epilepsy compared to healthy controls, a recent meta-analysis actually could not identify any consistent, any robust effect, and there's no clear pattern that there's no clear alterations between that we found in language processing in children with frontal and temporal lobe epilepsy. And this is actually what motivates our study, and why our objective is to better characterize the language, the alterations in language networks in children with frontal and temporal lobe epilepsy, and also to study how the network alterations are associated to cognitive performance. To do so, to investigate, to address these objectives, we recruited children between the age of 6 and 18 years old at the Saint-Justine Children's Hospital in Montreal. This was all children that had a confirmed diagnosis of frontal and temporal lobe epilepsy, and recruited a control group of healthy peers that did not show any health concerns. So our study groups include 26 healthy controls and 13 epilepsy patients with frontal or temporal lobe epilepsy. What is important is also that our groups do not differ regarding age or the socioeconomic status, so we did not have to control for this to compare our groups. All children underwent a standardized neuropsychological assessment where we aim to evaluate the general cognitive capacities and also specifically assess their level of receptive and expressive language functions. Then also all participants had a combined functional near-infrared spectroscopy and EEG acquisition. The installation, as you can see on the picture, covered regions of the frontal and temporal lobe in both hemispheres. And we assessed the cerebral activity both during a 12 minutes resting state period with the eyes open and during a receptive language task where children were listening to 18 different segments of the story of Snow White. It is important to mention that for the current analysis, the EEG data was only used to control for the electrophysiological abnormalities. So there was a clinical epileptologist who screened all the EEG data to make sure there is no subclinical seizures that occurred during our acquisition, and also to make sure that there is no pathological findings in the control group. Then the subsequent steps aim to ensure that we have a good signal quality of the fNIRS signal, and also to remove confounding noise. Once we had we obtained the signal of, the oxyhemoglobin concentrations, we computed the functional connectivity matrices with cross correlations. And we also applied a regression for the age because our sample has a rather large age range and we were not interested in doing any developmental investigations. And as a last step, we aim to really isolate the cerebral activity related to language processing, and therefore we subtracted the functional connectivity matrices of the resting state signal from the task condition. And this not only allowed to isolate the language processing, but also to control for potential epileptical discharges that occurred during the acquisition. So what you see on this slide is actually the group comparison of the functional connectivity matrices between the healthy controls and the epilepsy patients on the left and in the middle. Then on the right is the differences that we obtained, so the significant differences between the groups. The matrices are organized based on the left hemisphere and the right hemisphere, and we regroup channels according to 4 different cerebral regions: the frontal, the temporal-parietal, and the temporo-occipital regions. This is really just to show you the steps that we applied in our analysis. The next slide will be much easier to interpret and to see the patterns of differences that we observe in the 2 groups. So you see the connectograms that show the connections within the interhemispheric connections within the left hemisphere in blue, the interhemispheric connections in the right hemisphere in red, and the interhemispheric connections in orange. On the left, you see the connections where the control group actually has an increased connectivity compared to the epilepsy patients. And on the right, you see the connections where the epilepsy patients have an increased connectivity compared to healthy controls. So what we can see here is that there's actually an increased functional connectivity within the control group in the left hemisphere compared to the children with epilepsy that show an increased connectivity within the right hemisphere. So this is a rather global interpretation, and we were interested in seeing whether there's also some more precise network alterations regarding the network's segregation and integration. And what we found, there is a significant difference between the network's integration between the healthy controls and the patients with frontal and temporal lobe epilepsy, which actually is in favor of the epilepsy patients because they have a shorter characteristic path length. We can discuss about this later on, how we can interpret this finding. Then we also looked at the cognitive outcomes, so all the different domains that we assessed in our neuropsychological evaluation. And you can see that actually both groups perform within the normative range. So they have, we consider them as being in the norm, but we see that there's a significant difference in performance between the two groups in favor of the control group. So they have a higher cognitive performance compared to the epilepsy patients. And then the last step of our analysis was to look whether there is a relation between the cognitive differences we find and the differences in the network metrics. But so far we see that there is actually, well, the cognitive performance does not significantly explain the differences that we observed in the network's integration. So as a conclusion, we see so far that the differences between children with frontal and temporal lobe epilepsy compared to healthy peers is that we see a different hemispheric functional connectivity of cerebral networks related to language processing. We see that there's a different network efficiency, and we also see that their cognitive performance is reduced compared to their healthy peers, even though still within a normative range. So our data does not yet allow to conclude what the specific link between the cognitive performance and the network alterations are. And so the next steps would obviously be to increase the sample size and increase statistical power, but also to apply more advanced analysis to better understand how the cognitive differences are related to the network alterations. And I think one point that that could be addressed is to look more into the interactions also between the different cognitive domains. So maybe there is not a direct link between language performance and language processing in the brain, but it's more of a complex interaction. So thank you very much for your attention, and also like to thank all the participants and the families that made this study possible. I'd like to thank my supervisor, Anne, and Fetzelmann and Schüli, who supported the data acquisition and analysis, and all my colleagues from the Université de Montréal and the clinical epileptologists who supported the recruitment of the patients of the Saint-Justine Hospital. Thank you.
Fantastic. We have time for a couple of questions.
Thank you very much for the great talk. I have two question. First of all, have you considered looking at other network features? Maybe you have too few channels for that, like small worldliness. And the other question is, like, as far as I understood, you did static functional connectivity analysis on this data. And have you considered looking into like dynamic functional connectivity, and what was the time window for your static functional connectivity analysis?
So yes, actually the reason why we chose these metrics of integration and segregation is because it's just in the literature on epilepsy, these were the most common reported metrics. So we wanted to make sure it's comparable with the current knowledge. But obviously, there's more to look into. For example, the modularity would also be a very interesting metric to look at. And regarding the connectivity, no, we did not look into the dynamical functional connectivity yet.
Maybe it's a good idea.
Yeah.
Thank you.
Well, thank you very much.
Thank you very much for the talk. My question is about the laterality. What do you make of that? The difference between the right and left connectivity between epilepsy and healthy patients?
Well, it's something that has been reported extensively in the literature. So it may really be a reorganization that actually allows to maintain the function, so that there's really an adaptation of the brain to the pathology of epilepsy. It would also be interesting in the future to look more into the differences also between the children that have a left lateralized epilepsy compared to those with a right lateralized epilepsy. Yeah.
Thank you.
Okay, I think we'll take one last quick question before we switch to our next speaker, and you can come up and.
Yeah, thank you, Alejandro. It's a very interesting study. So could you talk a little bit more about the results of the cluster, like what those cluster like indicates in terms of the effect of epilepsy on brain function?
Well, there was no difference, so we don't really see a difference between the groups of the clustering. So, I mean, what we can conclude is that it's actually comparable to the healthy peers. Yeah. It would be interesting. Again, it would be interesting to see whether there's still whether there's a variation even within the healthy control group. If there's a variation of the clustering that links with the cognitive performance, so to see what is more advantageous or yeah.
Yeah, thank you.
Thank you.
Okay, thank you very much.
Thank you.
So our next speaker is Luisa Gose.
Hi, everyone. Good morning. I'm a postdoc at the Birkbeck Baby Lab and Toddler Lab, and I'll be talking about some ongoing work that we're doing to try and understand the association between sleep and development in babies. So first of all, why should we care about baby sleep in the first place? If you're a parent, you know why you should care. But actually, when you look at baby sleep, it shows a lot of variability. So some babies sleep really poorly, and some babies sleep really well. And in the sleep literature, we know that sleep is really, really important for memory consolidation and learning. And we think it's because sleep affects synaptic plasticity, and that then drives memory and learning. And synaptic plasticity is also related to brain connectivity. Now, we know that brain connectivity patterns in infancy and in development in general are also reflective of developmental status. And we see altered brain connectivity patterns in children with neurodevelopmental conditions. And similarly, if you think about it, memory and learning are crucial things for development. So this is kind of where we're starting from. And we want to kind of understand how sleep and how babies usually sleep. So are they good or poor sleepers? How does that cascade downwards onto development? And one way in which we think this could be is by looking at connectivity during sleep. So this is what kind of this talk will be about. Now, in sleep research, EEG is the gold standard because it is currently the only way in which we can accurately and reliably classify sleep stages. So rapid eye movement sleep and non-rapid eye movement sleep. But of course, if we want to look at connectivity, EEG is not ideal because you need many, many electrodes to reliably do it. And I'm interested in how babies usually sleep. So with that many electrodes, it's not going to be very comfortable for them to sleep with. So if we think about fMRI, that's great for connectivity, but it's not a particularly normal environment for babies to sleep in. It's very loud, it's not very comfortable. And if you add the EEG onto that, you can imagine we're not really measuring how they usually sleep. So of course, we thought about using fNIRS. because we can combine it with EEG and it gives us better spatial resolution. And we're not the first people to think about using fNIRS in sleep research. So people have done that in adults, and we actually see that hemodynamic changes as detected by fNIRS drive these sleep stage transitions. That's important because we know some sleep stages, like non-rapid eye movement sleep, are where we think the memory consolidation processes happen. And actually, functional connectivity differs depending on sleep stages in adults as well. So we're thinking that we're going to look at what happens in a brain during an infant sleep. So by looking at functional connectivity, what kind of functional connectivity patterns can we identify when babies are sleeping? And does it vary with babies who usually sleep poorly and babies who usually sleep well? So the first thing that we needed to do is basically create a headgear that's very comfortable for them to sleep in. You see all the cables and all the wires. So we have our EEG system, which is a 20-channel wearable Inobio system. And we started with a fiber-based NIRS system because that's what we had available when during my PhD. And we spent many months tuning this headgear, making it very comfortable, tuning the research protocol, because it's not that easy to get them to nap when you want them to nap. And we ended up testing 26 infants. And then the study got interrupted by the COVID-19 pandemic. So luckily, maybe not luckily, when I was able to restart testing, we actually had a wearable NIRS system available as well. So I changed the headgear to integrate and make it wearable. So that enabled parents to interact with the children more. And you can see I changed the array slightly, but it mainly covers the same areas as it did before. And we are focusing on the age range of 5 to 9 months. So in terms of study design, one thing, I'm going to be talking about the analysis from the fiber-based system, and then we're replicating those in the wearable system. But we're still collecting data with the wearable system. So we call parents and get information on their sleep habits of the child, and then they come into the lab and nap. So we put the headgear, the fNIRS EEG headgear on. We give them the opportunity to either nap in the pram, in the cot, or in parents' arms. And we basically just wait until they fall asleep and wait until they wake up again. So just as a reference, this study takes about 5 hours from start to finish. When they wake up, if they have slept, then we will also do some eye tracking with them to collect some information on attention and developmental status. And then we send them home with a sleep questionnaire to assess kind of how they usually sleep and good and poor sleep. And we also collect actigraphy data, so to get an objective measure of their sleep at home. And we have some developmental questionnaires to assess developmental status as by-parent report. In terms of analyzing the data, so we have this fNIRS pre-processing pipeline. I'm not going to go into too much detail on that right now. But then we assessed the data quality using qt-NIRS, and we looked at the number of poor quality channels and the good window percentage. And we compared this with between the wearable and the fiber-based system. And then we also looked at the EEG data quality, because it is important that when we collect this data, we have the information about sleep stages. And lastly, quite importantly, how many of those infants that we invited to the lab actually sleep with the headgear on? Because we can invite all of them, but if they don't sleep, then that doesn't help us very much, right? Now, once we've collected the NIRS data, we divide it into 120-second epochs. We do a correlational-based connectivity analysis, and then we exclude all the bad channels. And actually, then we run a k-means cluster analysis on all of the connectivity matrices. And that we did that because we're interested in connectivity patterns that reoccur across participants, so to say, as a proxy for our developmental stages and maybe sleep stages. And then we relate those clusters to how they sleep at home. So we're interested in what kind of states can we see during sleep, and do more of those connectivity states. So if kids have lots of different connectivity states, does that relate to better habitual sleep? And we identified the number of K clusters using elbow plots and gap statistics based on each system separately. So if you compare the data quality between the fiber-based system and the wearable system, so you can see that we get a good data quality for both systems. You see that the data quality for the fiber-based system is actually slightly better. But then when you look at how many babies actually slept with the fiber-based system on, you can see it's much less than with the wearable system. Not a big surprise, because with the fiber-based system, you can't really move the child around, you can't hold them, you can't breastfeed them. And that is quite crucial as like a bedtime routine for many babies when they're this young. And then when you see the EEG data, actually, we only get 45% of valid EEG data with the fiber-based NIRS system. Again, this is because it has this heavy fiber bundle that pulls on the cap, and so the cap moves. But we get 100% valid EEG data from the data sets that we record from the wearable system. So we basically conclude from this that actually the wearable NIRS system, or the headgear that we designed, actually represents a really good compromise between data quality, but also experiment success. So we can move babies around, they're more comfortable, they actually sleep longer, and we have this opportunity for caregiver and child interaction. So these are the preliminary results of the connectivity and cluster analysis. So this is for the HBO data, and we can identify 3 different clusters based on the input that we get, and they look kind of different. So that's what we wanted. We wanted to see differences in connectivity patterns. When we do this with the wearable NIRS system, we actually see that 4 clusters is the optimal number of clusters that we should put into the analysis. And some of it looks similar to the fiber-based system, but some of it doesn't. We'll be doing some more validation on this. But if you remember, we want to relate this to habitual sleep. But the first step is to see actually if our kids differ in these connectivity patterns. Because if they all have the same connectivity patterns across the same nap, then that doesn't tell us anything about development or individual differences. And actually what we see, we can see that some kids show all clusters, some kids only show one of them, and some show 2. And we see this across both the fiber-based system and the wearable system. So that's great news. We see this individual variability. And then we checked, how does it relate to the habitual sleep? So first off, we can actually see that this cluster number 2, how much of it occurs during a nap in lab in the infants, actually correlates negatively with the nap duration. Now, when we relate this in the same infants to how long they usually sleep at night, so a measure of how good or how poor they sleep, we actually see the same negative correlation. And lastly, when we compare it to a developmental measure of a parent report developmental measure, so the ages and stages, we also see the same negative correlation, also for the same cluster. And this was quite exciting because it kind of shows what we expected, right? That our connectivity pattern of this one cluster correlates in the same way across our habitual sleep, our nap in the lab, but also our developmental measure. And we think that actually maybe this is indeed a way in which sleep can cascade onto development. But when we try to replicate this with the wearable system, we can't really replicate this pattern. Now, there's a number of reasons for this. The data was a lot less clean with the wearable system. And what I would like to do eventually is combine both of those systems together. So we pool the data, we have similar regions, and then we can maybe see if we can replicate something like this. Now, the key message is we can use fNIRS to study functional connectivity during infant sleep, which is a great news. And we have our wearable headgear enables data collection from lots of different infants and circumstances. And actually, I think the system has great potential to be used in home, in different settings, and also with other populations. So for example, toddlers. And lastly, we think that these connectivity patterns are actually really maybe a way in which we should be looking at sleep. and how it can influence development, though we need to do some further research on this. So our next step is kind of increase the sample size of the wearable system. And our big thing that we are going to try and do is look at the EG data, because I've shown you how it's related to habitual sleep, to in-lab nap, and to the developmental measures, but we haven't looked at sleep stages. So this is what we're currently doing. We're looking at the EG. And my thinking is that this pattern that we see is probably a type of sleep stage that keeps recurring. And if we can find this association, then that kind of supports that theory a bit more. We'll also look at objective sleep and our eye tracking measures as an objective way to look at development. And I just want you to end with thinking about how we can use this kind of approach to study sleep and development also in settings that are not in the lab itself, but in different age groups and different populations, and children with neurodevelopmental conditions, for example, where sleep is a huge topic. And then, of course, how could we make this system scalable so that we have more data than, you know, the 30 kids that I have in my sample. So yeah, I want to thank my co-authors, Paola Pinti, Frank Wiesemann, Claire Elway, and Emily Jones, especially, and all the parents and babies that participated. Thank you.
Thank you very much.
So time for a couple questions.
Hi, good morning. Really beautiful, cool stuff. So I'm just curious, do any of your data sets have the two different systems on the same kid?
No, they don't. Yeah, that would be ideal. But basically it covers the entire head already. And they
Not at the same time, but just if they came in on day one and then came in again on another day.
Yeah, I think we should do that. It's very hard to get participants in for this. But yeah, we should aim for that. Yeah.
Very cool.
Thank you so much for your talk. So I have two quick questions regarding the data analysis part. So first is about the time series. I was curious about for those babies who cannot sleep for the entire 5 hours, how do you control, the time series for you to calculate function?
Yeah, that's so we basically, most babies did not sleep 5 hours, right? So most of them actually sleep between 30 minutes and an hour. So they all sleep relatively similar. We have some babies who sleep 2 hours, but very few. So far in this analysis, I haven't accounted for that I have more data from some of the babies than I have from others. I think it should theoretically be okay because I look at the epochs and then the cluster analysis is run on all the epochs together. But I think if I look at the habitual sleep association in more detail, I think you're right. I should account for that I have more data from some kids. Yeah.
Thank you. The next one is, could you please clarify why you include that I think 3 different types of motion correction method in your data pre-processing?
Yeah, so I actually based it on Renata's paper, where the combination between spline and wavelet worked really well. And I noticed in the sleep data, when you move the baby, I have this data where it's really clean data, and then suddenly you move them, which is quite a big difference between the two. So that's why I just wanted to make sure that I get rid of these abrupt things.
Thank you.
Thank you, Lisa. It's a very great talk. So my question is you just mentioned the babies have different sleeping time, right? Some have 30 minutes, some have 5 hours. So I just wondering, when you calculate functional connectivity, are you using the whole length of the sleeping time? Yeah. And another question is, do you find a huge difference when you calculate functional connectivity with different sleeping time?
Yeah, so in the kids who sleep longer, you see different patterns, which has to do with the fact that they probably have different sleep stages. So when they nap only for a very short time, you usually don't go into REM sleep, for example, because you don't have that opportunity. So you see some difference in the kids that nap for a very long time, but because most of them do nap between 30 minutes and an hour. So yeah, it's the kids that nap for a long time that show more things like REM sleep, and then the connectivity patterns.
Oh, so basically you still like segmented your recordings like into different like period of sleeping, like different stage of sleeping.
Yeah, so exactly. So that's basically what we aim to do, right? So when I look at the connectivity patterns per time, they change more in the longer nerves, as you said. But I think it's because we're seeing the REM sleep occurring. Is that your question? Did I answer it?
Yeah.
Yeah.
Yeah, thanks so much. Yeah.
Okay, last question. Let's maybe keep it brief so we can stay a little bit on time.
Yeah, thanks a lot for the great talk. Actually, my question was a bit related. What was the size of your segment when you compute the connectivity? And also, what I cannot read, what was the filter that were used, the bandpass filter?
It was 0.1. Wait, yeah. So it was 122nd epochs, and I have to look up the filter exactly.
Okay, just probably you removed it, but the respiration rhythm is changing a lot during sleep.
Yeah,
So
exactly.
that's
Yeah, we
important
counted
part.
for that.
Okay.
Yeah.
Thanks for great.
Great. Thank you very much. And our next speaker is Caroline Kelsey.
Great. Thank you so much for having me. I'm so excited and grateful for this opportunity to talk a little bit about the work that I'm doing in the Nelson Lab. I'll wait a second for the slides. Perfect. Okay. Beginning in the first few weeks of life, infants are continuously exposed to emotional stimuli in the context of faces and voices. And with this comes a decision, should I approach or avoid? And this decision can be informed by a variety of factors. So are these people, contexts, objects, familiar, happy, and inviting to us, like this conference, the Society for fNIRS, and we want to approach and go to them? Or is this an object or context we're not so sure about, and they look scary or fearful, and we may want to avoid them? However, not all infants respond the same way to the same stimuli, and these individual differences may be informative for the development of psychopathology later in childhood. So the overall objective of this study was to utilize fNIRS to investigate if and how infants respond to emotional stimuli and if these are related to their emotional and behavioral problems later in childhood. So frontal brain asymmetry is a common marker used in EEG, and it's considered to be a neural endophenotype of motivation behaviors in children. Specifically, greater relative right frontal asymmetry is thought to index a withdrawal motivation, and greater relative left frontal asymmetry is thought to index an approach motivation. It's also considered to be a marker for psychopathology, with greater relative right frontal asymmetry being related to depression and anxiety in adults, although the results are really mixed. And so I would like to argue for potential ways to clarify these mixed effects. One of the ways is to study frontal asymmetry within a given context. And so it's thought that studying frontal asymmetry within a particular emotional challenge may confer some advantages, such as minimizing uncontrolled variance, which might be within a resting state condition. And it's also thought theoretically that maybe this is a marker of adaptability to a particular context. Another possible way to clarify the mixed findings is to use a complementary, wonderful neuroimaging technique, functional near-infrared spectroscopy, which has the potential to clarify and extend previous EEG work by possibly reducing by allowing for the spatial resolution and allowing for this more precise localization of the signal. And so for this study, we had 3 main research questions. So are there group level differences in frontal asymmetry responses across the different emotional contexts? Are individual differences in frontal asymmetry in infancy related to emotional and behavioral problems at 5 years of age? And are these responses specific to frontal asymmetry, or do we see asymmetries in other areas of the brain, also allowing us and seeing this association with the psychological outcomes? So children first came in infancy, and we had 90 babies, and the infants viewed a series of happy, angry, and fearful emotional expressions, and we computed a frontal asymmetry score by subtracting the right dorsolateral prefrontal cortex region of interest from the left dorsolateral prefrontal cortex. And so throughout, a higher score is going to index greater relative right frontal asymmetry, thought to index that withdrawal response. And so for our first question about group level differences in frontal asymmetry to the emotional conditions, we did not find any significant differences in frontal asymmetry scores between the happy, angry, and fearful faces. We also asked if overall infants showed a greater relative right to left frontal asymmetry, and we did not find any evidence for group level lateralization patterns. Next, we assessed if individual differences in frontal asymmetry in infancy are associated with emotional and behavioral problems, and we assessed this using the child and behavioral checklist. Here we found that greater relative right frontal asymmetry in oxygen and hemoglobin, or this withdrawal response specifically to happy faces, was associated with increased internalizing and externalizing problems at 5 years of age. We also found that greater withdrawal to the happy faces and fearful faces was associated with an increased likelihood of developing a clinical anxiety diagnosis, which we assessed using the diagnostic infant preschool assessment. Finally, we found that only asymmetries in the dorsolateral prefrontal cortex and not in the other areas of interest were related to 5-year outcomes, suggesting this may be a localized effect. So to summarize our findings, we found that greater relative right frontal asymmetry indexing this withdrawal response to emotional faces was associated with an increased likelihood of developing psychopathology at 5 years of age. If you're interested in learning a little bit more about early neural markers of later behaviors, please visit. We have 2 fabulous honors thesis students who I have the pleasure of mentoring, and they're giving great poster presentations, so please go visit their posters. So with that, I'd like to thank my co-authors, the Emotion Project team, our funders, and our families for helping with this wonderful work. Thank you, guys.
Okay, we have some time for questions. No? Oh, Lauren has a question.
I think this is really fascinating. I'm interested in sort of the value added of the NIRS here. And I'm quite intrigued by the fact that you're finding it in dorsolateral prefrontal cortex. Would that have been predicted by the EEG work? Is that a surprising finding? I'd love for you to tell me more about that.
Yeah, so we based our areas based off the 1020 system, where you typically compute the frontal asymmetry score, so F3 and F4. And so I think that EEG research with their source localization tend to focus on dorsolateral prefrontal cortex. So it was a region that we did predict, and previous NIRS work also using this, like Katie Kroll used a very similar region of interest. So it was the region that we predicted, but I think it helps add to the literature of the EEG because you can't be sure that it's just coming from that particular region. So I think this helps to give us more confidence that it's coming from this region. Yeah, great question. Thank you.
There's one question there. Yeah. It's intriguing that you followed these kids for 5 years, right? That's a big investment of time. Do you plan to continue following them later on in life?
Yeah, that's a great question. We just got a grant to continue following these kiddos. So I think that's a big part of being able to do this beautiful longitudinal work is having the funding to be able to continue doing that. And so now the kiddos, we're going to follow them through adolescence, so about 10 and 11 years of age. So I'm used to participants that, can't talk very much. And so it's a big and fun and exciting endeavor that, you're able to talk to your participants and they have their little lives. And, developmentally, we're really excited to see how these early measures can be related to these later outcomes. Yeah, great question.
Okay, wonderful. Looks like those are all the questions.
Yeah,
Thanks so much.
Thanks so much.
Okay, so we can welcome our next speaker, Meredith Petrokonis. Okay, wonderful.
Good morning, everyone. My name is Meredith Petrokonis, and I'm really excited today to have the opportunity to present some preliminary findings from one of our new studies here at Boston University. So during the first few years of life, children's language development is shaped by their household language environment. And the household language environment can be defined as the quantity and quality of language that children are exposed to at home during everyday activities, such as shared book reading and screen time. Previous research has shown that frequent, high-quality shared book reading has a positive impact on children's language development, while too much screen time is associated with poor language outcomes. However, we currently do not know how children's brains process language that is presented during these everyday activities, nor do we know if children's brain response to language is related to their household language environments. There are, however, a reasonable number of neuroimaging studies that have explored the neural bases of language processing in children more generally. So a recent meta-analysis of 27 fMRI studies conducted with children reported that regions of the frontal and temporal lobes are involved in language processing. However, this meta-analysis also reported that only a handful of these neuroimaging studies included 3, 4, and 5-year-olds. And this means that we know very little about how the brains of preschool-age children process language, which for the developmental folks in the room, know that this is a really big problem because the first 5 years of life are considered to be a sensitive period in language development. Another limitation of these neuroimaging studies is that the results may not be generalizable to the social world that exists outside of the well-controlled lab environment. So in all of these fMRI studies, language was presented to children during non-interactive contexts. So in other words, children listened to stories and audio recordings of other speech sounds while they were socially isolated inside of an MRI scanner. And of course, as Joy wonderfully mentioned last night, this is not naturalistic, right? So this is not how children are exposed to language in everyday life. And I would actually argue that what these studies were measuring was how children's brains responded to audio recordings of language. Some studies have found really creative ways to simulate social interactions within the MRI scanning environment so that we can better understand how the brain processes live language that's presented during social interactions. So for example, studies have used video chat platforms to measure how the brains of both adults and school-age children respond to language that is spoken by a live social partner. And these studies found that certain regions of the brain respond more strongly to live language that's presented during a live social interaction compared to recorded language that's presented during non-interactive contexts. Despite the really innovative nature of these neuroimaging studies, these social interactions are still artificially constructed because of the restrictive nature of the MRI scanning environment, right? So we have to present our stimuli through screens and audio recordings. So these studies are lacking ecological validity, and we still do not truly know how children's brains process language that's presented during live social interactions. So with the help of fNIRS, we've begun to address this limitation, and we've really leveraged the strengths of this technology to measure the brain function of infants and children as they interact with live social partners, as is demonstrated in some of these photos here. Nevertheless, no studies to date have measured how the brains of preschool-age children process language that is presented during a live social interaction. So the goal of the present study was to investigate how the brains of preschool-age children respond to live language that was presented during a shared book reading interaction compared to recorded language that was presented during screen time. And so we first aimed to identify which brain regions were activated when listening to live language and which were activated when listening to recorded language. And we next investigated whether the brains of preschool age children responded differently to live language compared to recorded language. And last, we explored the associations between children's household language environments and their brain response to live language during a shared book reading interaction. So to address these aims, we created a novel fNIRS task with 2 conditions that simulated shared book reading and screen time. So in the live language condition, we had a live experimenter initiate interaction with the child by reading them a scripted story from a book. And while listening to the story, the child was seated next to the experimenter so that they could see words and pictures on each page of the book. And in the recorded language condition, the child listened to an audio recording of the same experimenter reading a different scripted story. And so for this condition, words and pictures were presented on a computer screen instead of a book, and the experimenter remained seated next to the child to control for the presence of the social partner. We created 2 stories to use in our fNIRS task, and these stories were matched on a number of different language and literacy variables. So one of the stories was presented in the live language condition, and the other story was presented during the recorded language condition so that children did not hear the same story twice. Each story was divided into 18 trials that lasted 10 seconds each, with 10 to 15 seconds of rest in between each trial. And 18 trials of the same condition were presented during the first run, followed by a short break, and then 18 trials of the other condition were presented during a second run. And the story used in each condition, as well as the order of conditions, was randomized and counterbalanced across children. So just to give you a sense of what this task looked like, here's an example of a child completing a trial in the live language condition.
Next, Maisie added some whiskers. What a beautiful snowman.
This 3-year-old sat very nicely for this study. All of the kids did not sit like this. And then here is the same child in the recorded language condition.
Next, Maisie added some seashells. What a beautiful sand castle.
For our equipment, we collected our fNIRS data using the Tekken CW6 system. And this is a picture of one of our fNIRS caps with and without our optical fibers. Here's a 3D reconstruction of our probe design that we projected onto a 4-year-old's brain template. So our probe included 16 sources in red and 26 detectors in blue. And this resulted in 48 long separation channels and 6 short separation channels. And the cap was placed over the CZ anatomical landmark on top of the head to ensure that channels covered similar brain regions across children. Our data was processed using Homer 3, and a summary of our processing stream is depicted here. So just a couple of things to point out. We use Spline SG, motion artifact correction, and Ted Hubbard's autoaggressive, iterative, robust least squares model for GLM. Average HBO values were calculated from 6 to 10 seconds, as this was approximately 2 seconds before and after the peak of our average hemodynamic response. So today, our sample included 13 children with an average age about 4 years of age, and then kids ranged in age between 3 to 5.5. And here's just more additional information about our sample. So while data collection is ongoing, I'm excited to present some preliminary findings, and due to the time limitations of today, I'll just be presenting results from HBO concentration values. So to address our first research question, we conducted within subject t-tests to identify channels that showed a significant increase in HBO compared to baseline. And we defined baseline as 2 seconds before the onset of our trial period. So which brain regions showed significant brain activation? So starting with our live language condition, We had 2 channels in the right hemisphere demonstrate a significant increase in HBO relative to baseline. And these were channels 42 and 38, which covered the right STG and right PCG respectively. And in terms of the hemodynamic response for these channels, both channels showed a significant increase in HBO, shown in red here, compared to baseline. So next for our recorded language condition, one channel, which was channel 1, that covered the left IFG, showed a significant increase in HBO relative to baseline in the recorded language condition. And here's the hemodynamic response for this channel. So on to our second research question, do the brains of preschoolers respond differently to live language compared to recorded language? And if so, which region showed this difference? So channel 5, which covered the left IFG, showed a significant difference in HBO between conditions. And so looking at this figure, we can see that HBO during the live language condition, which is our solid red line, was higher than HBO during our recorded language condition, which is our dashed red line. And unexpectedly, HBO in this channel actually decreased over the course of the trial for both conditions. So this suggests that brain response to language more generally may decrease in this channel, but less so for live language compared to recorded language. Channel 44, which covered the right STG, also appeared to show greater HBO during the live language condition compared to the recorded language condition. This difference, however, did not quite meet significance with a p-value of 0.057. So with further research and a larger sample size, we'll see if this meets statistical significance. For our last research question, we're interested in exploring associations between children's brain function and their household language environment. So we measured children's household language environment using the Reed scale from the STEM-Q, which is a parent report questionnaire that assesses both the quantity and quality of shared book reading that children experience at home. And we conducted exploratory correlation analyses and found that HBO values from channel 14, which covered the left PCG, were significantly correlated with children's STEM-Q read scores. And this correlation was positive. So children who experienced more frequent, higher quality shared book reading at home, as indexed by higher STEM-Q read scores, had greater brain response to live language during our shared book reading interaction. So in summary, live language during our shared book reading interaction activated the right STG and PCG, and recorded language during screen time activated the left IFG. And when we compared conditions, brain response in the live language condition was greater than brain response in the recorded language condition, and this was true for the left IFG and potentially the right STG. And finally, children's household language environment was positively associated with brain response to live language in the left PCG. So in conclusion, our preliminary findings suggest that children's brains may process language differently depending on the activity in which language is presented, which provides a neurobiological explanation for why shared book reading and screen time influence children's language development in different ways. And we also found that children who experienced frequent, high-quality shared book reading at home showed greater brain response to live language that was presented during a shared book reading, which suggests that household language environment shapes brain development during the preschool years. We'd like to quickly acknowledge the following limitations. So first, these results should be viewed as preliminary due to the small sample size. We didn't correct for multiple comparisons, so just interpret these results with caution. And we have not yet accounted for probe placement variation. So the brain regions mentioned here may vary slightly across children. Our data collection is currently ongoing. And so with a larger sample size, we'll be able to conduct more analyses with larger sample, with more increased power. And we're also interested in looking at analyses with HBR concentration values as well. And lastly, we'll be collecting data from a group of children with autism to determine whether the neural bases of language processing during these different activities differs in a population that experiences challenges with language and communication. And with that, I would just like to thank my mentors, the team at the Neurophotonics Center and CARE, and my funding sources, and of course, the families that have participated so far. Thank you.
Thanks very much. We have a question?
Yes, thanks a lot. Really exciting. I'm wondering, so kind of a suggestion or comment. So in typical live interaction language studies, the point is that the parents or experimenters responses are contingent on the child. Whereas what you've shown, at least from the videos, it looked like even your live interaction is extremely scripted. And so parents really, essentially, the only difference is whether the sound comes from the computer or from the person, but the person actually does not really behave contingently. So I'm wondering whether you find much larger differences if you actually allow the interaction to be more contingent on the child's behavior.
Right. Yeah. Excellent point. I was also thinking about the contingency piece because especially for autism, that is like crucial, right? So we decided not to, the way we have it structured is like if the kid tries to initiate interaction with me right now, I just kind of briefly acknowledge it and then we move on just to control for contingency across conditions. But we actually have another task that we're doing where parents are reading a book with their kids. It's a book with no words, right? So that is a completely naturalistic interaction. So we're really excited to see what brain activity looks like during that condition.
Oh, I accidentally turned it off and back on. So I actually have a quick question, if I may. So we know that shared book reading is such an important activity. We encourage parents to do this with their kids because it's associated with really positive reading outcomes later on. And so I know you've just started this study, but I'm wondering if you've given some thought to potentially following these kids and seeing whether this type of activity now during this really critical, pre-reading activity correlates or predicts future reading outcomes.
Yeah, that's a great point. We're collecting measures of children's language skills. So we're using the preschool language scale to kind of get like a concurrent measure of their language skills. We don't have any reading measures, unfortunately, but yeah, if I continue on with this sample, that would be a great idea.
Okay, great. Let's thank all our speakers for a wonderful first session. And I believe we now have a break until 10:05 for coffee. So thank you all.
Good morning, everyone, and welcome back to this session. We have the guest speaker session now. I'm very happy to co-chair this session with Professor Arjun Yot, who will introduce our guest speaker, Professor Tromwer.
Okay, can you hear me? Oh, okay. So I guess good morning. I'm excited, and I hope all you are excited to hear from our friend and colleague Bruce Tromberg today. I'll say a few things about him. So many years ago, many years ago, Bruce was a teenager shooting baskets, playing politics in Washington, D.C., and now he's returned. And so Bruce he's the director of the National Institute of Biomedical Imaging and Engineering at the NIH. And as director, Bruce plays a critical role, arguably the critical role in the US investment strategy for developing and translating basic science to the clinical and commercial sectors. And he's gonna, I'm sure, talk about that. By way of background, Bruce did his undergraduate at Vanderbilt in 1979. He did his PhD at the University of Tennessee, working with Tuan Bodine. And then he moved to Irvine, and he's worked mainly at the Beckman Laser Institute, where he rose from assistant professor in 1990 to eventually become director of the whole institute in 2003. And then I guess became emeritus professor in 2018 and went to NIH in 2019. So, Bruce has had tremendous impact in biomedical optics, and his research is actually his own research it's very closely related to the stuff we're doing in the fNIRS community. I met Bruce in the early 90s when Bruce came through the Johnson Foundation to Britton Chance's lab to give a talk. We were both actually beginning our assistant professorships at those times. And, we were both intensely interested in how to use modulated diffusing light to probe tissue physiology. And I should say, actually, David Boas was a grad student there. Joe Culver was a graduate student there. Turgut was, I think, an undergraduate. Turgut Durduran was an undergraduate there at around that same time. So it was exciting for the field at that time. Collectively, as a community, we were figuring out how and demonstrating things that you can do. And, Bruce and I became friends and collaborators right away. And I think it's safe to say in the research area, Bruce sustained his love for modulated diffusing light, but in multiple incarnations. Okay, so he found different ways in which to use it. But importantly, like Britton Chance, Bruce really always had a vision for its physiological uses. And so, for example, especially for cancer monitoring and diagnosis and surgery, Bruce took a lot of time to learn cancer biology, to think about how our signals that we look at in hemodynamic signals are related to cancer biology. And so that was a really very important thing. And he did similar kinds of thinking for other problems in the brain and the muscle. And so I think it's safe to say scientifically, this combination of his technical and physiological vision basically has inspired a lot of research activity in our community. And it's boosted a lot of careers, actually, of people in this room. And so, that's by way of research. I think that it goes without saying in parallel, Bruce, he's been a major, played a major unifying role in our community. He founded the Journal of Biomedical Optics. He started and helped sustain many important biophotonics initiatives at the SPIE and at the OSA. He was chair of Gordon conferences, Engineering Foundation conferences, as well as the conferences of SPI and OSA. He connected our community, helped being glue to connect our community internationally. And he organized a major Akron, multi-institutional Akron study about breast cancer therapy. He helped start some companies, train many, many students and postdocs and more. And so I just want to say that, when you talk to him, one thing that is really nice is you find that he's not just interested in science. He has interesting observations about lots of things, really, from music to sports to literature to family life to people, politics. So, and he's generous in the best sense of the word. So, it was my good benefit to meet Bruce so long ago, and please join me then in welcoming the lecture today.
Thank you, Arjun. That was an amazing introduction. Wow, very thoughtful. And it's really been my privilege to be part of this community and grow up in this community. And I originally was going to give a virtual talk, and I just was having physical pains thinking about that because it's like being homesick or having withdrawal. I really wanted to come back and meet my colleagues. It's been very, very long. It's been much too long. And we're all in this stage of trying to rebuild our communities and reconnect. So Mari, thank you for the invitation. Thanks to the organizing committee, all the volunteers. This is such a special society. I mean, you can see all the love and the passion that goes into it, from the beautiful website with all the colors and the design to the shared tools and technologies that people use. It's just the spirit of what we do in science that's so powerful. Arjun went through a lot of what I did technically, and I just thought when I applied for this position at the NIH, there are 27 institutes, and I'll tell you a little bit more about the NIH. And the directors are pretty, they're more traditional. I really never thought that I would be selected as a director because I like to talk about photons and electrons, and mostly they're physicians and are very deeply into public health and human behavior and, the big issues where lots of money goes. And I can say it's been a privilege for me to learn from them. I almost feel like I've just gone to grad school again. And I'm just about to graduate, maybe, or I may be stuck in this loop for quite a long time. But I can also say, and you'll see from my talk, a little bit of our community. I bring this whole experience of our community, our obsession over how things work. And I've been able to bring that to the NIH, and people are paying attention. So I think that's a real testament to all of you and what you're doing, the envelope that you're pushing. And I hope you see some opportunities in my talk to continue to advance that, because all the work that all of you have been doing has contributed to the kinds of things. It's our community that's done what I'm going to talk about right now, which will be new opportunities. And I hadn't thought about in vitro diagnostics for quite some time until the pandemic hit. And I suddenly became in charge of in vitro diagnostics for the country. And I'll tell you that story. But certainly our impact in optics and photonics and the kinds of things that we have naturally done as a community in terms of sharing approaches, model-based imaging, those kinds of technologies, are lessons that the traditional imaging community, radiology, can also learn, and we can learn back from them. So I'll go into some of those details. So first, not that many people I've learned know what NIBIB is. That's the National Institute of Biomedical Imaging and Bioengineering. And we're one of the smaller institutes, although I think our community nationally, worldwide, has grown quite dramatically. But before 2000, there was no NIBIB. In fact, the NIH director at the time felt that there really didn't need to be a special institute. But in 2000, December 29th, President Clinton signed this bill. And there's a little bit of a yellow highlight here. And in the bill, the language, I always like to sort of pull this out. This is kind of the creation act of this institute. It really underscores what I think all of you know intuitively, which is that physics, engineering, computer science, chemistry, physical sciences, engineering sciences, mathematical and computational approaches, they're very different from what normally goes on in medicine and biology, and certainly at that time, very far afield. And there were many, many people in the United States that felt that because they're so different, there needed to be a separate institute, representation at the NIH of that particular community. The other school of thought was, okay, you know, every institute should have a little bit of that, the Heart Institute, the Cancer Institute. Fortunately, I think for me, and I think ultimately the test of time will show that for humanity, the idea of having a separate institute that's dedicated to this amalgam of disciplines, where people are really outwardly looking and bringing in ideas from other areas, but also combining that with engineering, physical science, computation, that really won the day. So that institute was created. Around the same time, if you look in the middle, you see that there was enormous growth in biomedical engineering departments all around the country. The Whitaker Foundation in the United States invested around $760 million over a period of 20 years to help grow biomedical engineering departments. And I believe Boston University was one of those that's benefited from that. But many, many around the country, there are currently about 180. When NIBIB was created, there were about 40 of those departments. And then there's been a paradigm shift really in engineering and schools of engineering in the United States and all around the world, where human health has become a very top priority, a high priority. It wasn't like that in the 90s. Human health really wasn't so much on the radar of schools of engineering. And now the flip is occurring. In the US, there are now 2 medical schools, University of Illinois, Urbana-Champaign, and Texas A&M University, that have engineering embedded in their curricula. So you would go to a normal med school and find nothing related to engineering. You could graduate entirely without that. But in these 2 schools, it is part of the core curriculum, and students graduate with masters in engineering. And this is going to continue to grow. And there's even a catchy term for it. People call it fNIRS. So this will continue to expand as we go forward. A lot of joint faculty appointments. And of course, one of the major economic consequences is that many of our community are developing practical devices. And to make a device that actually ultimately has impact, you want it to be commercialized so it can be disseminated and used widely and standardized. So a lot of these activities have driven innovation and entrepreneurship and diversity, attracting students from a variety of different backgrounds who really like that blend of physical science with biology and medicine and altruistic intent. So it's kind of a unique space that our community gets to occupy. So this is kind of my pictogram of what the NIBIB invests in. These are really the areas that we're responsible for. It spans from engineered biology. I think many of you know broadly what that is: cellular and tissue engineering, engineering extracellular matrix. It also includes things like synthetic biology, which is having a physical representation of single cells, much like you would do for integrated circuits, but instead of transistors being your fundamental switching element, you can have transcription factors that are switches. So you can model cells much in the same way that you model an integrated circuit. And you can reprogram them just in the same way. Sensors and point of care technologies, imaging technologies from the biggest physics imaging, the highest field strength magnets and PET, CT, and whole body PET, to small imaging devices like this head-only MRI. We also support therapeutic devices, which tend to be external energy beam delivery, ultrasound, RF, optical, also using waveguides to insert energy in the body, and bioelectronic activation, for example, of the brain, and also activating molecules with those external energy sources. So we support, this is kind of our broad mission, all of it tied together, as all of you do, or many of you do, with modeling, computation, machine intelligence. When I arrived at the NIH, everyone was going crazy over AI and ML. And I thought, wow, did they just discover math at the NIH? I mean, this is kind of crazy. Everyone had gone nuts. And I was immediately put on the Scientific Data Council, and I got to contribute in a lot of ways. What I did find, and this is one of my first advertising plugs, there just not enough of people like us in these sort of classic biomedical entities. So I encourage, if you're interested in careers and moving in that direction, you're desperately needed. And that's been really amplified by my experiences throughout the pandemic. And I'll go into a little bit of detail on some of those. So here's my view of the NIH. Of course, I'm pretty biased. The NIBIB, the Engineering Institute, is in the middle. And our technologies, like many of you who work collaboratively across disciplines and look outwardly to neuroscience, to cancer, to cardiovascular disease, we look outwardly as well. Most of the institutes are for very, very specific diseases: heart, lung, and blood, kidney and digestive diseases, allergy and infectious diseases, musculoskeletal. So it's a little bit like going to a med school when you go to the NIH. Many of the institutes are connected to those medical disciplines, and they're run in structures that support large medical enterprises. And when I say large, it's big. $45 billion a year is the budget for the NIH. And most of that is sent out to institutions. So virtually everyone in the United States and many people around the world are supported by NIH funds, NIH grants. And we're relatively small. We also have a clinical center that is on the campus, and it's the biggest research-based clinical center in the world. It's quite inspiring. So what's been the impact of all of this? I put us in the middle, but there are actually some numbers, and I've been able to go in and extract these numbers. Francis Collins was the director of the NIH starting in 2008, and he left in 2021 at the end of the year. Over the course of his tenure, the overall fraction of the NIH budget, that's all the institutes, that has gone to bioengineering is 13.5% just in the most recent year. So it's increased at a rate that's 2.5-fold the rate of the total budget increase. So there's been substantial investment. Now, at the same time, NIBIB, which supports bioengineering, we're 1% of the NIH budget, but 13.5%, all the institutes are investing. The National Cancer Institute spends more on bioengineering, quite a bit more. than our total annual budget. And that's a good thing. So we've kind of accomplished both of those goals. We've been able to get bioengineering into the operations, the normal operations of all the institutes. And we've also created a separate entity that's driving and a home for people who are really developing enabling cutting edge technologies who don't have to have a biologic hypothesis to come to the NIBIB. So in 2020, I actually joined in 2019, and I got enough familiarity to know where my parking place was and my office and those kinds of things. I met some people, and then the pandemic hit in 2020. And all of the institutes really went through this very intensive exercise to think through how could we contribute? How could each contribute to the pandemic response? And at NIBIB, we identified 3 specific areas: imaging and artificial intelligence, in vitro diagnostics, and digital health platforms. And I'll go into a little bit of detail on each of these. But to make anything happen, you need to have new budget. And we, over the last 3 years, have become kind of like 2 institutes. NIBIB1, which is our normal congressional appropriation, which you can see in blue, tracking from fiscal year 18 through current, which is right around $400 million per year. And then you can see this Public Law 116-139. I had to read that. If any of you have had the experience of getting a really good grant review, and you look at it and you read it over and over again, and it said, you got a great score and you're going to get funded, and you're kind of a little bit in disbelief. So if you look at this carefully, this public law said $500 million would go to NIBIB. I had to read that many, many times over to fully accept it as reality. Now, it was not without strings. So we got 500 million. The NIH overall got 1.5 billion, and they said, and you will develop in vitro diagnostics, you'll develop testing technologies at point of care and other settings immediately, like right now. So that was a little scary to see that. It's exciting, but scary, and it changed the entire way that we've been operating. Essentially, we're like 2 institutes, and one of them is a startup. And that's what I've been operating over the past few years. So you see that supplemental funding in red, that's increased our total budget each year to about $1 billion. So that's a sudden surge of money and a huge surge of activity. And we've been able to invest that in these 3 areas. So that's kind of the second NIBIB. Going back to our core programs, what we do, most of the institutes operate off what is called a pay line. And that's for us, last year, it was 19th percentile, 24 percentile if you're an early stage investigator. So we give extra 5 points. And we have a number of signature programs. There's a Trailblazer Award for young investigators with little or no data. It's a really great program. We have very unique training opportunities for underrepresented minority students. And even we've been able to create diversity and low resource settings, what we call R01s and R21s. So those are new. We have signature programs that are partnerships with industry. So if you're working collaboratively with industry, you should look at these. It's what we call a U01, bioengineering partnerships with industry. We have kind of the jewel in the crown of technology development, what we call P41 centers. So those are national centers for biomedical imaging and bioengineering that are aggregations of really beautiful technology, which are shared and disseminated, a lot of training. So those are really unique programs. We work very collaboratively with NSF's engineering directorate. So this is kind of a sampling, and you can see if you go to our website and you look at all the different programs, it's a little mind-boggling, but our program directors are terrific. And if you just contact, and I have a number of names throughout the talk, contact them and they'll help navigate where there may be opportunities for funding for you. But I really wanted, and this is Fruz Anderson, some of you may know her. She is our program director for optics and photonics. And I really was wondering, how much do we actually invest in optics and photonics at the NIBIB? And when you break down our whole portfolio a little, and we break it down in 3-year groups. So in 2020 to 2022, a little over $1 billion in total. Overall, looking over a 10-year period, almost 2/3 of the investments from the NIBIB go into either developing optics and photonics technologies, cutting edge development, or programs that are developing technology that are dependent. They actually use optics and photonics technology. So it's kind of a surprising bragging factor that, you know, we want to point towards, that this community has such a critical role, not just in NIBIB, but I think if you extrapolate this all across that $45 billion, so we're in the process of trying to do that. And you'll see that optics and photonics is really fundamentally enabling, and it's absolutely essential that we keep pushing the envelope on the development of new technologies so they become the ones that our colleagues in other application areas are adopting and using to drive discovery. What about micro versus macro scale optics? Macro scale being more of the functional regime, and how does that break down? So if you look since 2018, it's been steadily increasing, and I would just say this may fluctuate, but anywhere between 20 to 60% of the total optics portfolio that we support is in macro scale optics, what you might call diffuse optics or fNIRS. I put photoacoustics in that because even though that tends to be more of a structural technique, it is driven by a diffuse optical pulse. So that is pretty impressive, actually, and you all should be very proud of that. If we look at the total NIH funding for macro scale optics, that's really visibly increased over this 10-year period, almost twofold. And the fraction that NIBIB has of that has also increased. So in this most recent 3-year window, 2020 to 2022, it's close to $1 billion, $900 million in total all across the NIH. About 10% of that is NIBIB investment. And what is everybody doing with this stuff? Well, you all probably knew the answer to this before I put it up, but from macro scale, the 2 biggest areas of application are in cancer and neuroscience. And if you compare that to all of optics, it's actually, I think, a validation that there's really a true impact in macro scale that's going in brain. Because if you look at all of optics, it's about 30% cancer and about 15% neuroscience. So if you just carve out the macro scale, there is that really huge impact that you're having in looking at various nervous system diseases, neuroscience more broadly. So let me kind of switch gears now and talk about NIBIB2 and how we dealt with the pandemic response and all of that additional money that came to us. One of the first things that we thought would be relatively easy to get going was to have an impact in imaging and artificial intelligence. We knew that there was really a huge problem in emergency departments with people coming in and having chest x-rays and CT scans and really being able to do rapid analysis and triaging of patients. So here we started a consortium to help with this major challenge that radiologists were facing, not enough radiologists fundamentally, who needed to have better tools, urgent need for AI and machine learning, both to diagnose and assess the extent of disease, but also to predict outcomes for patients. And now increasingly, we look at long COVID or post-acute sequelae of COVID, PASC, and having imaging as a way to have quantitative imaging biomarkers and AI and ML-based algorithms to really assess that for individuals is essential. So we formed a national network, and I hope this inspires this community. In a sense, this meeting is an international network of people working together, sharing algorithms, doing validation. This is a formal structure that we were able to form in partnership with the 3 major imaging societies: Radiological Society of North America, RSNA, ACR, American College of Radiology, and AAPM, the American Association of Physicists in Medicine. So they're actually engaged in this network. It's about a $30 million 3-year contract. And it has many participants from all around the country that are engaged in taking in data and developing as their primary role new algorithms and releasing this data to the community. So there are 40 different sites all across the country in 23 states that are involved. The principal investigator, the leader of this, is a pioneer in computer-aided diagnostics and AI, Mary Ellen Geiger. Some of you may know her from she's a past president of SPIE and an absolute pioneer in this area, as are these investigators from all around the country. They've been able to recruit other societies to be part of this. And I really encourage you, this would be okay to go on your phones and look at midric.org. There are a lot of terrific tools there that I think can help inspire what you're doing with fNIRS data, and maybe even get access to some of those databases with some of your patients. There may be some patients that are in common. So some selected work that has come from this are algorithms to assess severity of COVID. These are AI-based approaches. One of the really important reasons why the network was formed, essentially AI has been over-advertised and underperforming in imaging, and there really have not been that many examples where there is truly an impact and outcome for patients, in part because oftentimes there's enormous amount of bias, whether geographic, gender, or instrumentation or investigator bias that goes into the assembly of the data that goes in the first place. So this network is really focused, and there's quite a lot on the MIDRIC website that goes into detail on this, of how to develop databases with validation data sets. So 10 terabytes of data have been now released to the public. Probably another 30 terabytes will be released before the end of the year. And that's essential, but it's also essential to withhold data, to have validation data sets so that ultimately algorithms can be independently validated, and then regulatory agencies can use that as these things become authorized. There's also a very practical tool called RADgraph, which de-identifies data from radiology reports, which is not a trivial problem. And they've worked to identify PHI, personal health information, and actually replace it with dummy information. So it's really quite powerful, open source, and available on this platform. And probably the last major challenge, when you go to the NIH and you see these 27 institutes, they all have databases, and they'll all have their databases oriented around patients, clinical data, electronic health records, genomic data, metabolomics. Some even have mobile health data, and they're all kind of in silos, and hardly anybody has imaging data. So we wanted to really solve this problem of how do you get interoperability between the data that comes from the imaging community and the data that comes from the normal mainstream clinical community. There are all these silos around. So we've been working on that with the National Heart, Lung, and Blood Institute, NHLBI, the National Center for Advancing Translational Science, NCATS. They have databases, very, very well structured, established databases for patients for COVID patients, with all that information that doesn't have anything to do with imaging. And so now we found that we have thousands of patients in common with each of those databases, and are developing new approaches. And you can see some of this on MIDRIC for interoperability between those subjects. Because you can imagine an image alone is fantastic, but you want to have the context. Radiologists would like to have at their fingertips all of the information about that patient so they can optimize a decision and project a therapy and outcome for that individual. So I think we're getting closer, but I was actually quite shocked at all of the data silos. People are putting terabytes of of clinical data out there without any ways to connect across platforms. So the technical community is tackling that. So now on to what we've done in vitro diagnostics and digital health platforms. What we've managed to be able to, we've really responded to an urgent need that became pretty clear in April of 2020, when in the United States, the only tests that were available for COVID were laboratory tests. And we were doing, as a nation, around 7 to 8 million tests in a month. And there was we had a rocky start to in vitro diagnostics in the United States. The test that was released by the CDC didn't quite work as well as they had hoped initially for a variety of different reasons. It took a while for that to get out to laboratories. And public health people, epidemiologists, the physician community basically said, look, everybody should be tested in a lab. Everyone needs to come to labs. That's how medicine works. Then you get your data. We send it to a doctor. The doctor makes the decision for you. And we thought, well, that, is that really necessary? Can't we have tests in people's homes or in the point of care? Technologically, that was certainly possible. So we had a concept, a plan to be able to do that. And then this editorial came out in the Washington Post. These two gentlemen they're senators, Senator Alexander and Senator Blunt. And they said, we need more COVID-19 tests, which was kind of exciting to see senators, come down, and we need a Shark Tank to get us there. Now, those guys didn't dream up the Shark Tank. I happened to get called into a phone call with the NIH director and one of these senators, and I was trying to explain our idea with Senator Alexander. And of course, I didn't have enough media training, at the time, and I wasn't making any sense. And Dr. Collins said, oh, it's like a Shark Tank. That's what they plan to do. And so these guys really latched onto that. And that turned out to be a pivotal moment, because after that call, just 5 days later or 4 days later, roughly, and this editorial came out, there was that $1.5 billion that went to the NIH for testing. So kind of shows you sometimes how randomness and serendipity can play an important role. But we were prepared for the program, and that part wasn't random, but just having a phone call with them was kind of crazy. So we moved quickly to launch this program. 5 days after the 1.5 billion was appropriated, we had things in place to be able to do what we call the Rapid Acceleration of Diagnostics Technology Program, or RADX. And the goal here was to dramatically expand the technologies, get out of the laboratory, and bring things to point of care and over the counter, and make the performance better. So we know technologically that the performance can be improved. That's what we do. That's what our community specializes in. And that was our goal. So we built it around a network that already exists, that we had supported at the NIBIB. It's called the Point of Care Technology Research Network, POCTERN. And the central organizing element of that is CIMMIT, the Innovation Institute at Mass General Hospital. But it also has Johns Hopkins, UMass, Northwestern, and Georgia Tech Emory. So we essentially asked this whole network if they would stop what they were doing with all the their other research and dedicate entirely to the pandemic response. And there was an external advisory board meeting. Dr. Collins and I got on a call with all of them, and everybody said yes. And so what was really amazing about this is we were able to effectively send out a call to the innovation community. If you think back to those times, everybody was really interested in contributing in whatever way that they could. So we gave an outlet and eventually built up around 900, almost 1,000 people around the world who were engaging and participating in this network in various capacities. There were specific things that we built in to the network that were different than what you might normally find. One was a core to do validation of every technology that we knew would be proposed. And that core is at Emory. And they have now evaluated hundreds, if maybe even thousands of every technology that we've taken a look at. And in fact, everything that the FDA has authorized, every time a new variant comes out, this validation core ensures it's working. So if you've used any COVID tests in the US, it's been evaluated by this particular core for its performance. And every time a new variant comes out, that happens. We also have a clinical studies core at UMass that can do digital trial design, and a deployment core to deal with deployment and manufacturing and scale up of these technologies. And the major engine behind all of this on the left side of the screen is what we call the innovation funnel, or in the language of the senators, a shark tank. So this is a process where any proposal is easy to come in to the network, but there are multiple stages of review that progressively become more and more intense with respect to your performance and milestones, very much like what venture investors do. And we had overall 824 applications come in. About 50 ended up coming through getting phase 2 investment. We supported many applications at various stages, but ultimately those 50 were the winners that received the most amount of funding, and they had to deliver in terms of the testing technologies. Over the past 2 years, in addition to that funnel, we've been able to launch additional programs. RADx Mars is a mobile application reporting standard. Shockingly, so we made tests to go to people's homes. People have said, gosh, there's no reporting. And there is actually no way to report your test if you take it at home. Even if you wanted to, there's no application standard. There's no standard protocol for transmitting the information. Public health authorities say, in general, we don't want that, or they certainly did at the beginning of the pandemic. So we created that highway, and I think that that will be engaged more and more in the future. We also created new ways to test how the tests work in different communities, and that was the Say Yes COVID test program, which for the first time made tests available online in different communities all around the country to see if people would use over-the-counter tests and whether they felt that they were of value. That experiment, which we did for about 10 million people around the country, is what ultimately Congress and, well, the president decided to use as the test distribution site for COVID.gov. That same model that we pioneered moved out into the national distribution program. So we've been involved in many, many other programs besides supporting the early innovation. And this is the impact. If you look at the total cumulative capacity of tests, our investment, it's kind of a mind-boggling number. It's close to 5 billion tests from this investment that came from Congress with the original charge of make point-of-care tests, make over-the-counter tests. About 11 of these are over-the-counter tests, 46 or so emergency use authorizations from the FDA. And just in April, we were contributing to about 500 million tests, August, about 500 million tests in the month of August. And this has driven this massive paradigm shift. So now if you go back to laboratory testing, where we all started, that's a distinct minority of the number of tests that are done each month. It's overwhelmingly, and why not? If you can do tests, and certainly if you can get them for free on a website, and you can do them at home, then that's really, it empowers so many people, and it really makes a difference that way. But ultimately for sustainability, governments have a hard time buying these and distributing them for free. So one of the major challenges as we go forward is to have price points that are much lower than they currently are. So I think they're still around $7 to $10 per test. So that's what is a new challenge. So all of this has impacted national policy, and the president declared that 1 billion tests would be distributed for free, and that a lot of those tests are ones that we worked with the FDA to accelerate their authorization to get independent data, so the FDA would be confident that they could be authorized. And also we pioneered that distribution network so that that could be done for people directly. As we go forward, there's still performance challenges. If you pay attention to the guidance on how to use these tests, there's some concern about if you just get one negative test. And we recommend that you do 2 tests in a 48-hour period. We call that sequential or serial testing, because you may have a very low viral load on the very first day you take the test, below the limit of detection of a rapid antigen test. And this really kind of illustrates that. Rapid antigen tests have detection limits that are on the order of about 1 million copies per mil. So it's pretty high. If you get a positive on a rapid antigen test, you have a lot of virus inside you. But the ROC curve is sigmoidal. So that means if you go down to 500,000 or 50,000 copies per mil, there is a very low probability that you're going to get an accurate test. You may still have virus inside you. And that's why people recommend that you use PCR. PCR in a laboratory can have detection limits on the order of 100 copies per mil. So the question is, are there home-based and point-of-care technologies to bridge that gap? And there are. And we've seen hundreds of ideas and applications come into our system using nanomaterials, everything from nanoparticles and nanopores to silicon nanowires and carbon nanotubes. So a lot of really cool cutting edge technology that I think is going to be the next wave of transformation that is impactful. Just a couple of examples of things that have come out. This, if you've seen this test by Illum, it's an integrated optoelectronic device. It has a source, optics, a detector, a Bluetooth chip in a small piece of plastic, and the readout, the reporter system, are quantum dots. So you can now buy a beautiful commercial product with beautifully integrated and disposable optoelectronics and quantum dots on the shelf of a drugstore. So pretty amazing that that's actually out there now. And I think it portends for the kinds of things that are coming in the future. Here's another one. This is not optics based, but it is pretty interesting. It's an integrated circuit based approach that uses field effect transistors. So it's built on 3D flash memory, or VNAND. And if you look carefully at those structures, they're actually FETs in 3D with hundreds of thousands of potential sensing elements. And if you can immobilize DNA probes in those, you can highly parallelize nucleic acid detection. So this kind of view is the view of the future for how home-based diagnostics and point-of-care based diagnostics will look. Instead of using nitrocellulose and chemistries that are like 40 years old, this is the the direction that things are going. Build tests and diagnostics on substrates, materials that are semiconductor-based, solid-state materials that will have better performance, and ultimately, we hope, price points and be more accessible. I had to throw this one in because it's really quite amazing. And Arjun may appreciate this. This is actually cavity ring down spectroscopy, which in the old days used to be incredibly esoteric and very difficult experiment to do. This company, Ring IR, has reduced using, the quantum cascade laser in the mid-infrared has really facilitated this entire technology. So making that portable, rugged, and miniature, they've put all of this in a small box, and they use mid-infrared spectroscopic detection tuned over a sufficiently broad enough region to where exhaled breath can potentially detect the presence of COVID or other pulmonary viruses or infections. It's still in the research phase, so it's not out there and fully released, but it's promising. There is one exhaled breath technology built on gas chromatography mass spectrometry. So that's already approved by the FDA, but these kinds of exhaled breath technologies are also coming. To help spur this on, we currently have our innovation funnel open again, and there are 2 major areas that we're supporting. One, if you've used those home tests, you can kind of understand that they can be difficult to open the box. The packaging is tough. Seeing the line can be hard. I've had lots of experiences where I was looking at it thinking, is that a positive or not a positive, you take a picture of it, you try to blow it up, you look at contrast ratios, contrast to background. So it's, you know, they're not perfect. And if you have impairments, so manual dexterity or visual impairments, certainly patients who are blind cannot use those at all. They can use the ILLUME test. And actually that's our major test that we're providing for that population. But we're trying to stimulate the redesign of these kinds of testing technologies so they're accessible for populations with physical disabilities and impairments, and then canonize some of those design features so they can go into other medical devices so they're more widely accessible. And the second major area that we're supporting are those next-gen technologies. Can we make high performance, low cost, and accessible technologies that work as well as lab tests, but you can have in your home and you don't have to do serial testing in order to get a definitive diagnosis? Now, the last thing I want to show is that RADx has really influenced virtually all of our programs. So now you can say, okay, what's a hard problem that needs to be worked on? And can we use those same principles of bring communities together and provide unique resources to validate and accelerate the technologies, and then move them through to commercialization and dissemination, and work with the FDA to get them authorized, because that's the ultimate step. Don't underestimate how hard and important that is. So we're doing this with maternal health. I think many of you may know this is a huge problem. The US maternal mortality rate is the highest of any developed country. And more than half of pregnancy-related deaths occur in the year following birth. So millions of people in the United States live in maternity care deserts. So we wanted to create, we've worked in collaboration with NICHD, the National Institute for Child Health and Development, which focuses quite a lot on this issue to try to accelerate the development of new technologies that would have an impact in maternal health. And this is structured just like our RADx program. Unfortunately, we don't have billions of dollars to invest in this, but we have a start. We have at least $8 million. We'll use exactly the same principles, and then we provide substantial support to work on the commercialization, validation, and moving the technologies through the FDA to ensure that they will ultimately be disseminated. So this deadline is November 1st for these applications. Some of you are maybe familiar with the Blueprint MedTech program. It's a consortium of NIH institutes. We've been able to take the lead with an idea to RADxify, a new initiative. We formed 2 incubator hubs. One is at CIMMIT, the other one is at Johns Hopkins, and they will function very much in the same capacity that we did for in vitro diagnostics, but for neurotechnologies. But a real focus of this is many people, you may be familiar with the 510K or the predicate process and authorization for FDA. Those are built on essentially prior authorizations or prior clearances. What we're looking for here is to support the hardest ones, what we call PMA or de novo clearances. Those are, by definition, the ones that are building on stuff that never existed before, the ones that are the most innovative and risky. So we want to have a new mechanism to help support those so they come starting from scratch and we can help build those up. You can apply directly to these incubator hubs, up to $500,000 per year support for 4 years. And we anticipate up to 30 awards per year. We're taking pre-proposals right now, and these are due October 25th. I think you're all familiar with the BRAIN Initiative. Spectacular investment in bioengineering technologies. About 2/3 of the BRAIN Initiative has gone to bioengineering technologies. This is a particular one. There are only 3 days left for this, but hopefully you've got applications in the works. These are transformative imaging technology development that are partnerships. It's a 2-phased corporate agreement mechanism. You can look at the RFA for details on this, and definitely contact Shumin Wang, our program director who leads it if you're interested. And then the last thing I'll show, this community, and I'm looking at Claire, has pioneered, and Mari, So many amazing technologies in pediatric health and pediatric devices. And we want to keep that going and expanding. So we had what's called an RFI, request for information, open. If you didn't have a chance to comment, that's fine. Contact Ufruz Anderson, the program director for optics. Keep your eyes open for opportunities here. We're trying to form new coalition and consortia, working with the Foundation for NIH. And the goal here is to really push the envelope on these technologies and get them authorized. As you know, it's extremely difficult to get them approved and authorized and out into widespread use. So my final slide is just kind of what's happening is a kind of a radixification of the NIH and certainly the NIBIB, where there are a variety of different input technologies. Our entire community and everyone we know is working on these things, from nano and biomaterials to waveguides and computational approaches. Those are kind of the ingredients in the soup that are going into that funnel. And it's an alternative to the kinds of funding mechanisms that we have out there. Those are not going to go away, but this is a way to be maybe better matched to our community that designs new technologies, builds them, wants to deploy them quickly, and then keep moving and leveraging that technology for other things. So we'll see, hopefully, the emergence more of digital twins in this space, a very rich area for development that combines hardware and computational modeling, virtual trials, which are a powerful way to get to truths and also save a lot of expense, because we don't need to do actual physical human trials with all of our technologies. Build databases, expand wearable, implantable, and point of care devices, and so forth. So thank you very much. This is the overall pictorial vision of the future. I hope you can see yourselves fitting into that in one of those pathways. Thanks again.
Thank you for this very inspiring and interesting talk. We have time for a few questions. Yeah, please go to the microphone.
Thank you. Thank you for a wonderful talk. My name is Valencia Kumsen. I'm an associate professor at Tufts University. And I wanted to ask about the recent reports that have been published about pulse oximetry technology and its level of inaccuracy based on skin pigmentation, and how that has affected COVID-19 treatment for patients and overall morbidity and mortality, particularly for Black patients. Are there any initiatives at NIH and NIBIB specifically focused on developing new technologies that can be more accurate for a wider population of people here?
Absolutely. Great question. Did I ask you to ask me that question? We just awarded, and it's up on our website. So we created one of our kind of major areas of focus is in health equity. And we have a number of programs in that. And one of our new R01 awards to help reduce health disparities that we just announced is in exactly that. So in R01, we just awarded to a group that is addressing that observed, there have been a number of reports coming out, and still the full impact of that is not really that well understood. And we probably can get this group together to come up with a variety of different reasons why that may be the case. But for sure, there's observed clinical disparities. And so from a technical point of view, we have a new R01 that we're awarding. We also have, we support design projects by undergraduate students. And our top design award this year, and I know it probably makes everyone feel a little old, but undergrad students came up with a really beautiful approach that involves a very careful calibration and augmentation to the pulse ox itself. And we'll be announcing that at the Biomedical Engineering Society meeting later on this week. But it's on our website. I can just tell you these are very active areas. These are not the only answers out there, so I encourage people to take a look. We have R01 opportunities, R21 opportunities, and even hopefully we're going to have more of these rapid acceleration and prize award opportunities. Very, very important. And it's only one of many challenges that we need to address.
Thank you.
Any
I
other
think
question?
that was one of the most surprising things when I came to the NIH and I met, sort of more traditional directors who felt that technology always drives up the cost of healthcare. And I was thinking, well, that's not generally our experience. The people who develop technologies, we envision how technologies can be more efficient, reduce the cost of healthcare, prevent disease in the first place. And as you know, undergraduate design projects are typically limited in how much they actually can cost in terms of materials and cost of goods. So they're incredibly clever and inexpensive approaches, leveraging the hard materials engineering components that are now widely available and very, very inexpensive. So that's an important point for our community to make and reinforce.
Thank you so much for the really interesting talk. It's exciting to see how much the funding has been growing for BIB, but it's still smaller than NIMH, for example, and other big institutes. So I'm just wondering if you could offer us as a community, from the trainees to their early career to the more senior members, what can we all do as a community to help you get more money so that we can get more money?
Well, I'm not allowed to tell you to lobby. But there's a big gap between the growth of our community. I think you could say in the late 90s, it was a small community for sure, but it's grown in size and impact, in relevance in so many ways. So I think it's time for another kind of surge. I can tell you even more of the stories of how all that happened in the first place. And I think professional societies can make a lot of that happen. We are getting noticed, our community solving these problems at a scale that really nobody thought could be solved, and so quickly. That's extremely important. So keep doing what you're doing and keep talking, because I can tell you that, as you know, we love to talk technology, but it does put a lot of people to sleep when we when we do that. So we have to tighten up our communication, really simplify it, and explain the relevance of it. I have a hard time with my colleagues not backsliding into photons and electrons all the time, but I fight it. I fight the urge. So that's a big part of it as well. I mean, Tony Fauci, if you follow his work, he's the ultimate example. His institute is a $6 billion a year institute. When he first started at the NIH, it was 300 million a year. And he has just been hammering in on this message that there's going to be a pandemic and infectious diseases are something to be afraid of. I had never really considered that before, because I think a lot of our community hadn't really thought that much about infectious diseases. But now it's certainly front of mind, top of mind for me.
Okay. Okay. We should thank again Professor Tromber for this wonderful talk, and we can move to the next session. Thank you.
Thank you.
Okay, so this year award ceremony is bigger and nicer, and we have more award. And the first thing to say is because thank you to NIH, NIBIB, and NHDS. So we got a grant to support part of this conference, and the money is, part of it is for awards. So since we are running late, first of all, we decided it's the president of the society that should give the award and be in the stage. So I have here the past president, Claire, and took over the president-elect. So, and I love that you're in pink. Go. So the first thing we established this year, since we didn't have anymore the GATE award for travel, we established a travel award, much smaller in scale, but very important, especially with the hybrid component. We were able to either have people come here and support not just registration, but also some expenses, or support at least the ability to register and by being in remote. So the first award is with Sando that is here, and he's a PhD student of psychology at Rhodes University, South Africa. So congratulations. And he has two posters, one today, number 53, and one tomorrow, number 80. So please stop by and see his poster. And I forgot to say, this travel award were selected by the Diversity, Equity, Inclusion Committee with Joy Hirsch and Turgut Durand as chairs. And they did a really good job to start everything. Our society was missing this DIE committee, and now it's up and running and doing great. So the next awards, there are a few people that were able to come, mostly are remote. So the next one is Iraj Kanjat. Okay, I will misspell and kill all the names, I'm sorry. She's from Pakistan and at Air University. And she had a virtual poster, so, but you can still see it in other town. The next one is Edgar Guevara. I think he's here and has a poster to day 3. And he's from Mexico. Then we have Asma Gulnaz, a master student again from Pakistan Air University. She also is not here. There is no way to get visa from Pakistan. There is very few people that were able to get it on time. And the next one is Ishara. No, I'm not, I can't read the last name. Sorry, guys. So he's a PhD student from the Bionic Institute in Australia. Thank you for coming here, Ishwara. And then we have another remote one from Brazil, Yumi Ambrojola. And she is from the Federal University ABC of Brazil. And Alejandra Rocha Solache, much easier if it's more Mediterranean name, is a computer scientist from Mexico. And she has a poster on day 3 here. Then we have Jamila Hector, a PhD student again from Islamabad in Pakistan. She had a virtual poster last week, but again, you can see in Gathertown. We have Atif Jacod from the ICFO in Spain, and he has a poster today. And we have 2 more. So we have Parts of Easy from Canada, from Toronto. He's a master studying, and he has a poster on Wednesday. And finally, Huma Adid from Pakistan again, and she was virtual. So thank you again to all of you for applying, and we hope to continue these travel awards. So let me, where is Miriam? So we established a new award. It's called Community Award. This society is all based on service. We have a lot of help for all the members, but we never been able to offer them anything. So this year we established this community award that goes for members that have achieved they have done outstanding service to our community. And we decided to make it regional because while there are people that are able to give their contribution to everyone, everywhere in the world, there are some places where it's more difficult, and just being able to do a community contribution locally is as well important. So we have 3 awards. The first one is Miriam Yushel. Please, Miriam, come over. Where are you? So Miriam has been training research on fNIRS, so she has brought papers. She has brought a lot of paper on standards and how to do NIRS correctly. and she helped a lot on this conference with educational courses. So Miriam, it's my honor to give you the first community award. Thank you, Miriam.
Yeah, thank you. Just a few words. I'm very honored, very humbled by the award. And I would like to take this opportunity to say that it's really great to see old friends, also my Zoom friends that I first met here, and also meeting new people. Just enjoy the rest of the conference.
Thank you, Miriam.
Thank you.
Congratulations. Thank you. So the other community award is in Zoom. These virtual things give me crazy. So we have Felix Schockmann from Switzerland that I hope is in Zoom. It's supposed to be in Zoom. Yay, Felix.
Hello.
Congratulations, Felix. I'm giving you the award. Hopefully there is somebody from Switzerland here that will bring you the award. Yeah, you can't see me, so I'll do it. And Felix, like Miriam, he was one of the founding people of the society, and he had his contribution from the beginning on setting up the web page, the newsletter, and he has this always collect all the fNIRS papers, and so it's a great contribution that he's given over the year. Felix, you want to say a few words?
Yeah, thank you very much. It's an honor to get this award. It's nice to see the fNIRS community flourishing and the research really making great progress. So I'm happy to see this development, and I'm happy to support development in the future as good as possible. Yeah. Thank you very much.
Thank you, Felix. And the third award, you see the name, maybe we will see the face soon. Noman Nasser from Pakistan. Hi, Noman. Great to be here.
Hello.
He must be very late in Pakistan right now, but thank you so much for staying this late. So Noman has established a community in Pakistan. There is one of the first users of NIRS in BCI, and he, despite the difficulty of doing research there and such, he's established a very competitive, very large group, and he has participated to the SfNIRS community by helping with the communication committee. So he always in the newsletter report about his region and his area and development there. So thank you, Noman. Thank you a lot.
Welcome.
Okay, your turn to talk.
Okay, so thank you very much. I would, of course, like to thank the jury and the society for honoring me with this award. It means a lot to me, and getting this award is, it's so encouraging that, it motivates me to do more work. The Society of fNIRS has always been really supporting in my journey. In 2016, I was, waived off the registration because of, lack of fundings. In 2018 in Tokyo, I got the travel award. This time again, I could have traveled, but because of the, visa processing, it's a tough process from here. I do have a representation here in this group. So one of my students, Harun Khan from Oslo Met, he's here. So, if it's possible, he can, receive the award on my behalf. Thank you.
Oh, thank you. I met him 2 days ago. He didn't tell me that. He should have told me, so I would have staged it. But thank you, and I'm glad that I have somebody that can bring this to Paris.
Yeah.
Thank you so much.
Thanks.
Thank you, now. Zoom is here. Yeah.
Thank you.
Thank you, everyone. So I think we're good. We are back in time. I rushed a little. I'm sorry. So now we are back to the Early Investigator Awards. So I have to thank Claire and Elias for establishing this Early Investigator Award for the Society. Elias is so passionate about it and is doing a great job as the committee. This year I made few changes and I don't know if you remember previous conference, there was 3 finalists, they were their talk, and then there was the decision who won by the committee, and the day after there was the celebration. So there was a little bit too much suspense and stress on these early investigators, so we decided let's do it like a career, what they have achieved and everything, not talk about their talk at the conference in a small session. So we had a list of nominees. Elias and the committee pulled out the finalists, and then the whole board decided who are the winners. And this year there were 2 very close ones, so we decided to have a second place and a first place. And Claire will introduce the second place for the Early Investigator Award.
So this is where I need to hold the suspense, right?
No, you can do it.
Oh, okay, well, I'll try and hold some suspense. So it's a real pleasure to really introduce this. Second place is a thing where it's like almost winner. Let's say almost winner. As Mari said, the caliber of the candidates this year were exceptional. And I'd like to echo Mari's thanks to Ilias for managing the process. So the almost winner has spent her career investigating social interactions in infants. Her PhD was involved in advancing and developing methods for looking at functional connectivity in the infant brain. She has spent several years working on the brain imaging for Global Health Project, working on some really complicated data sets, particularly from those in the Gambia. And through that has produced a standardized pipeline for analysis for global health measures with fNIRS, which we expect will go on and really help a range of different groups working in that area. And this is work that she's done with Adam Egerbrecht and Luca Polonini. And in March of this year, she started an early researcher Leverhulme fellowship to work in the world's first toddler lab in London to look at the development of empathy in toddlers. I've had the pleasure of working with her. I can say that she's exceptional from lots of perspectives. She has an incredible attention to detail in the way in which she approaches her work, but is also able to look at the bigger picture of where her work fits into the project. And so I'm really thrilled to announce that the recipient of this award is Chiara Bulgarelli.
Here is your award.
Thank you so much.
And now, Chiara, I'll set up your slides.
Okay.
So she will present about her work. But first, tell us about the award, whatever, you can start.
Yeah, no, I just want to really, well, first of all, thank you, Claire, for the very nice word, the introduction. And I really want to thank Mary and all the board of the fNIRS community for giving me the chance to soon present what I've done so far and hopefully what I will be doing over the next few years.
Find the mouse. Sorry. Oh. Okay, sorry. No, it keeps appearing. I'm a Mac person. This is a PC. Okay, Chiara, sorry I interrupted you.
Yeah, so it's a great honor to receive this award, and I'm so happy I have the chance now to talk to you a little bit about what I did so far and what I hope I'll be able to do over the next few years. If the slides pop up.
Okay, where so as Claire very nicely introduced, what I hope I tried to do so now was to use fNIRS to investigate changes during social interaction from infancy until childhood. So my background is in psychology and neuroscience, and throughout my studies, I've always been interested in developmental changes. So how do we become who we are, but especially like all the variety of changes that happen during the first years of life. I've always been interested also in neuroimaging, but especially in brain connectivity, because I think that represents a bit better how the brain works, and social interactions. And when I came across the NIRS method, I thought, wow, that's an interesting tool that allows me to test all these things, to investigate and test all these things together. So that's why I started to use fNIRS. And while I was thinking about my career so far, I thought about three main avenues, three main topics on which I think I contributed to the field, the neurodevelopmental field. So the first one is like using NIRS to assess the development of core cognitive skills. The second one is using NIRS to assess risk factors and cultural diversity on neurodevelopment. And the third one is some work that I did to improve fNIRS set analysis for infant data. And through the next 20 minutes, I will try to walk you through this aspect of my work. So to start with the first one, this very much covers the work that I did during my PhD under Victoria Southgate, Antonia Hamilton, Karina de Klerk, and Anna Blasi. And here we wanted to start by using fNIRS to assess functional connectivity in awake infants. So as you all know very well, there are several networks several resting state network in our brain that the fMRI literature very nicely already detailed. And the one that I was particularly interested in is the default mode network, because there's quite a lot of fMRI study that, especially in adults, links the network to astral self-processing and mentalization skills. And these were topics that I am interested in, and I was investigating during my PhD. And I always found quite interesting the fact that there are a lot of studies that document the presence of visual and primary sensory network from birth, but there are no traces of the default mode network before the first years of life. So one might think that this gradual development of the default mode network then might be related to the gradual emergence of social cognitive abilities. So we wanted to try to test that. Of course, as you all know, with fNIRS, we couldn't picture the whole default mode network. So we focused on portion of it. So we focused on the frontotemporal parietal connectivity as a proxy of this network. And what we thought was really important, and that we stress out also in this paper, in this work, is that with fNIRS, we have the chance to assess functional connectivity in awake infants to avoid the influence of sleep stages. So the first question we had here was, can we measure the developmental trajectory of frontotemporal parietal connectivity in awake infants? So during my PhD, I was very lucky to be part of a very big longitudinal project where we had 60 infants coming to the lab starting at 11 months and then coming back every 6 months. We assessed connectivity from the frontal and temporal brain region that we co-registered while they were looking at the screensaver-like video, because of course we had to keep them awake, we had to keep them engaged, but without any particular social or cognitive stimulation. And one of the core findings of this work was that indeed this frontotemporal parietal connectivity increased with age. Oh, you can see me. Yeah, well, I found it. So frontotemporal parietal connectivity increased with age. So these developmental trajectories are color-coded like this, this plot here. And most of them do reach a maximum peak between 2.5 years of age. So this was quite pioneering at the time, because this was one of the first evidence of fNIRS as a good tool to explore functional connectivity in awake infants. We showed consistencies with previous finding where the default mode network is shaping over the development with this peak roughly about the 2nd year of life. But as you remember, I started by saying that I was particularly interested in this network because it has been linked to abstract processing in adults. So I was like, okay, what does it mean for infants, right? So I wanted to try to test that. So testing self-processing in infants is very difficult because, of course, they can't talk, they can't tell us what they think. And the gold standard in the literature is what it's a test that is called mirror self-recognition test. So the developmental psychologists might be familiar with that. It's a very old test, but still the gold standard in the field, where basically we covertly place a red dot, normally with lipstick, on the infant cheek, and then we place him or her in front of the mirror, and then we see if they recognize themselves, they touch the dot on their cheek. Otherwise, they normally just bang on the mirror or play with the mirror. Lately, a lot of researchers, a lot of scientists started to think that a successful performance at this test is not just an indicator of mirror self-recognition, but it actually entails a bit more like abstract self-processing in infants too. So what we thought was, okay, then if the default network is a marker of abstract self-processing in adults, we want to see whether we could find any differences in connectivity between infants that pass and those that don't pass the mirror self-recognition task. And we tested this on 18-month-old because at this age, roughly half of them do pass the task and the other don't. And this is what we found indeed. So recognizer showed greater frontotemporal-parietal connectivity compared to non-recognizer, especially over the right hemisphere, which is consistent with what we previous studies have found in adults. And therefore, this study again remarks the fact that not only there is this link between the full mode network and psychological self-processing, but then we found this link in infants too, and this might suggest that already at 18-month-old there is some sort of like broader sense of self, some sort of like self-awareness in infants. So this, as I said, was very much the work that I did during my PhD. And after looking at functional connectivity and the social brain, I wanted to move a bit forward and see how different factors of the environment might affect functional connectivity development. So I was very lucky to be part for my first postdoc of the BRIGHT project. So Anna Blasi already talked about the project this morning for those of you who were there. The Brain Imaging for Global Health project led by Professor Claire Helwell, Dr. Sarah Fox, and Dr. Sophie Moore has the overall game of identifying those risk factors and typical of infants that grow in low and middle income country, and see how these may affect the infant's development. And this project is very unique because, as you see, it has several different time points. So we tested the infants the first time that they were only 1 week old. And then, as you see from this slide, it's very, very rich in terms of measurements that we collected. So a lot like fNIRS and EEG, both for like the neurodevelopment assessment, but we also had like nutrition and biological assessment, loads of tasks, cognitive tasks, and questionnaire. And within the project, I focus on the fNIRS data. And here I'm going to present some preliminary results on the functional connectivity development in Gambian infants that I did within the BRIGHT project in collaboration with Adam Ekbret from Washington University, St. Louis. So why here we thought it was interesting to look at functional connectivity? Well, because of its relationship with structural connectivity, so myelinization, synaptogenesis, which we know are fundamental process for neurodevelopment. Therefore, we think, okay, disrupted pattern of functional connectivity then might reflect greater disruption in brain development. There's quite a lot of literature, especially fMRI literature, that talk about an increase of functional connectivity during the first years of life, with this shift between local short range to a more mature long range, so frontoposterior interhemispheric with age. And we also know that increasing long-range connectivity has been related to greater performance in cognitive tasks, and that risk factors such as blur filtering can have a detrimental impact on neurodevelopment. So the questions we had here were, first of all, how does functional connectivity develops in Gambian infants up to the second year of life? Does poor growth impact functional connectivity at 24-month-old, which is our endpoint here? And does functional connectivity affect cognitive outcomes at preschool age? So we collected fNIRS data while infants, they were just like doing a passive viewing task at several time points, 5, 8, 12, 18, and 24 months. We had 34 channels covering prefrontal, inferior frontal, and temporal brain regions that we have averaged over 6 regions. And as a measure of growth, we collected length and weight at every visit, and then we computed weight for length Z-score. And as a measure of cognitive outcome, we assessed cognitive flexibility, which is a core executive function that we assessed with a tablet task in a follow-up assessment that we did between 3 and when toddlers were 3 to 5 years of age. For several methodological factors, the age of the cognitive flexibility assessment was like the distribution of the age of the cognitive flexibility assessment was bimodal. Therefore, I will present the results split by younger and older preschoolers. So the results from the linear mixed model on 132 infants show that there is an increasing that the frontomiddle bilateral connectivity increase with age, and the long range fronto-posterior connectivity increase with age too, while there is this decreasing long range frontal intrahemispheric connectivity. And then if we look at the distribution of the functional connectivity across age points, so these are all the for connection, we see that there's quite a lot of variability across time points, even across babies, even if the pattern is there. So we thought it was interesting to relate these results to growth and cognitive outcome, as I anticipated earlier. So if we look at growth and functional connectivity, we see that changes in growth between birth and all the subsequent time point and 1 month and some of the subsequent time point positively predict the long-range intrahemispheric functional connectivity over the front. And then we see that the short-range bilateral frontomiddle connectivity are negatively predicted by changes in growth between birth and 1 month and some of the subsequent time point. And generally, like the wrong range fronto-posterior connection over the right was basically negatively predicted by changes between 1 month and the subsequent time point. And then if we look at functional connectivity and cognitive flexibility, we see that generally there's this pattern of like long range connection, so intrahemispheric and fronto-posterior, positively predicting the performance at this cognitive flexibility task in both younger and older preschooler. So generally, these findings are in line with the existing literature, except for the frontal interhemispheric functional connectivity that decrease with age. This looks a bit atypical, first of all, because it's inconsistent with what we were expecting and what previous studies have found. And it is positively predicted by growth and is positively associated with cognitive flexibility, which suggests that this connection should actually increase with age and not decrease. More importantly, I think this is quite interesting from these findings, the fact that positive growth just during the first few months of their birth and not after the 5th month impacted functional connectivity at 24 months. So this really highlights the importance of the first few months of life. And in general, like altogether, the fact that we see this positive growth very early in life affecting functional connectivity and functional connectivity positive predicting performance in cognitive abilities at preschool age emphasize the importance of the timing of early intervention. And these benefits of this intervention might be visible up to preschool age. So unfortunately, I couldn't go into much details of like data analysis and pre-processing, but I have a poster on that on Wednesday, so feel free to come and talk to me about that. The last bit of my work that I'm going to talk to you about is the work that I did to improve fNIRS data analysis for infant data. And the motivation behind this was driven by the fact that, as Claire highlighted earlier, collecting fNIRS data in poor resource setting posed that in front of some challenges. So if you're interested to read a bit more about that, there's this paper from my team that explain all the things that we had to adjust to successfully collect neuroimaging data in the Gambia. One of the first challenge that we had was a score like dealing with dark skin, dark hair, and last but not least, the fact that most of the infants were coming to the clinic with braids or not like this, and you can see immediately, of course, like if the hop dots sit on top of that, that prevents, of course, the light to successfully reach the detector from the source.
So all these challenges motivated us to look a bit more into detail about the data quality of this data. And I was lucky enough not to work only with the BRIGHT data, but also the data collected in the context of the BEAM project, so Bangladeshi Early Adversity Neuroimaging Studies, led by Professor Chuck Nelson. And these are some of the group HRF. So these are group HRF, but every line here is a baby. So here I was processing the data, excluding low quality channel, just a very basic function based on like low signal to noise ratio, and then doing like the classical motion artifacts correction and all the other stuff that probably most of us do. And this is what I was ending up with. And of course, like you can see, I wasn't really happy about, like, there was still quite a lot of variability between infants. Several low quality channels survived the cleaning, and there was still a lot of visual inspection needed, right? But of course, as a field, we try to move towards like a standardization and avoiding like everyone to clean the data manually, and it's quite subjective. So when I looked into more details about these like bad channels, so like looking, for example, at the power spectral density, it was very clear that there was basically like no physiology detected in this channel. So this, for example, the power spectrum density of this bad channel here, and it was completely flat compared to this one, for example, that is like a power spectrum density of a bad channels, and you can clearly see like the peak of the cardiac pulsation. So to not reinvent the wheel, I knew that by then Luca Polonini from University of Houston was working on this very cool tool to assess data quality based on the physiology and cardiac pulsation. So I got in contact with him, and we started to collaborate on applying this tool on our data. And just very briefly, this is a tool that assesses like with two criteria, like the scalp coupling index and the peak power spectral power, assess the presence of the cardiac pulsation in the near signal, both in the temporal and in the spectral domain. This is maybe something that fNIRS researcher working with also, I guess, have been done for quite a while, but in the near infant field, it was still like a relatively new thing to do. And if we look preliminary at the data, analyze both, like, so exactly the same pipeline, but just changing the channel pruning method. So just like a standard signal to noise ratio based method, or like fNIRS. If we look like in this case at the BRIGHT data, so we see that the signal to noise ratio is higher if we prune the channels with using fNIRS compared to the standard method across all time points. And even if we had to exclude a little bit more channels, that in the end, like in the final sample of the analysis, so again, data analyzed exactly with the same pipeline, just changing the channel pruning method. At the end of my, like in my group stats analysis, we end up with more babies when we use fNIRS. And for those of you who do like infant research, I'm sure you appreciate it. Even like a couple of baby mores means a lot. So then if I go back and look at my HRF, I was way happier to see, like even visually to see the results, because of course this represents way lower variability between infants. Very few low quality channels survived this cleaning, and there was basically no need of visual inspection. So to sum up, I showed you so far the front temporal parietal connectivity increase with age up to the second year of life, and it might be considered a marker of self-awareness. With my work in the BRIGHT project, we showed you that the functional connectivity development in Gambian infants seems to show some atypical trajectory, especially for the frontal interhemispheric functional connectivity. And more importantly, that positive growth right after birth supports the development of functional connectivity. And last but not least, the use of fNIRS on our infant global health data increased the quality of the recovered HRF and diminished the need of visual inspection. So now what's going to be next? So I'm super excited to say that I've been awarded of Liberum Trust Early Career Fellowship to look at the development of empathy in toddlers. I think that empathy is particularly important to be studied because it's a crucial social skill to have, and it will affect like social bond, group formation, and failure of empathy might evolve into severe diagnosis of antisocial behavior or callous unemotional trait. Why I want to focus on toddlers? Well, there are quite a lot of evidence, and we contributed to that evidence with one of the PhD work that I showed you earlier, that from the second year of life, toddlers do develop some sort of like self-other differentiation, and this will help them to understand whether, in the case of empathy, for example, emotion originated from themselves or from other. So far, we definitely lack of suitable experimental approaches to test this, because preschoolers don't comply with all our like standard lab setting. And to test empathy in general, it's very hard to elicit empathy if you just ask your participants to look at like just static pictures on a screen. So over the next 3 years, I will be leveraging the newly developed World First Toddler Lab, and especially the K facility in it. And what we plan to do here is to make preschoolers interact with the human-like avatars, and hopefully like being able to feel the first person experience typical of empathy, while we still have all the controls on the experimental variables. Of course, we're collecting NIRS data. So we have a wearable fNIRS Artini system with 48 channels that covers the temporal lobe and the frontal lobe, and we are able to dissect and understand better if there are different networks the response to cognitive and affective empathy, as adult literature show. Of course, this wouldn't be possible without the great support of Paola Pinti and Adine Aburuman, especially for the help in the cave and designing the avatar and integrating fNIRS and all this technology all together, and my mentor, Professor Emily Jones. So just very quickly to show you one of the first pilots that Paola and I tested. So this is the cave. You see all blurry because it's like a 3D environment. So with the glasses, she is able to see like the depth of the scenario, and she's just playing popping bubbles with the avatar. And this is the equipment that she has on, like super wearable, super comfortable, like everything is packed into this little backpack. And as I said, she's able to see 3D in the scenario. So hopefully feel everything way more realistic with these 3D glasses that Paola customized for our toddlers. I just want to finish with thinking that I think that we have had, like for this fellowship, me and my collaborators, 3 years ahead that are very, very challenging, yet probably, hopefully super stimulating, because this is like a completely new lab, and it will give us like the opportunity to rethink about like all the ways in which we test toddlers. So we have to think about new way of designing paradigm, new task, possibly new developmental theories, and hopefully you will hear about us in the next SfNIRS conference. So do come and check my poster about the plan of my fellowship today. So with this, I'm going to finish here. I really want to, again, give a great thanks to all the parents, infants, and toddlers that participated in my studies, and a big thank, of course, to my supervisor, collaborators, and funders that supported my research. And thank you all for your attention.
Okay, so first, I'd just like to echo the theme of community building that Mari and Joy Hirsch mentioned yesterday. While the theme of these awards are early investigators, so they're kind of about the individual, it's also one of the ways that we build the fNIRS community. And so in a way, these awards are for one, but they also increase the profile of fNIRS for all researchers. The other theme I'd like to echo is one that Joy touched on yesterday, which is the exciting cognitive neuroscience that fNIRS can attack is intimately attached to the foundation of engineering innovation spanning instruments, algorithms, and paradigms. And so with that in mind, it is with great pleasure to introduce, he'll come up in a second, winner of the Early Investigator Award, Alex von Luhmann, who it so turns out has an impressive history of building said fNIRS engineering foundations, including instruments, algorithms, and paradigms. Alex earned his PhD at the Berlin Institute of Technology, where he developed an fNIRS EEG accelerometer, kind of a multimodal everything system with the application focused on brain-computer interface. He also developed algorithms, a blind source signal separation approach to denoising data, and then in his spare time, he founded an open source fNIRS project. And kind of collectively based on all that work, David Boas recruited him to Boston, where his first project was denoising, but his main project was building a next generation fNIRS EEG system focused on neuroimaging in everyday life. Shortly after, a highly productive postdoc, he in 2020 moved to NIRS, where he's now Senior Director of Research and Development, leading the development of software, hardware, and also international standards. So now if we zoom out a little bit, Alex has been instrumental at developing ideas kind of at all stages of translational pathway for fNIRS, from visionary high concept projects like real world fNIRS through the nuts and bolts of running a commercial company, and even the more granular nuts and bolts of establishing international standards. So this is a truly impressive career trajectory for somebody just a few years out of having their PhD. So without further ado, please welcome me in introducing the Early Investigator Award to Alex von Lumen.
Oh, that works. Okay. So first of all, thank you. I'm really honored, and it is a real encouragement for the continued pursuit of my research career. So thank you to the award committee, to the whole society. Yes, it makes me very happy. I hope I can convey this in the next few minutes. And I would like to start with a rather personal and hopefully not too philosophical motivation. So I like to see myself and us as humans basically as part of nature that discovers itself through human consciousness. And I'm an engineer, so I try to contribute to that by building tools, as I like to say, for brains to understand how brains work. And that has been a very long motivation for me. And it started actually not on the engineering side, but really more on a introspective side, looking at meditation, mindfulness. This is not so long ago, but you see me with a shaved head in the Buddhist monastery in a Thai rainforest. And so that is where my original interest came from introspection. Of course, on the other side of the world for centuries, science has looked, into observing human nature and how anatomy relates in dead people, mostly relates to behavior that was observed in the living. And of course, luckily, we don't have to do that so much anymore. We can now use technology to relate physiology and brain signals to behavior. And basically, my entrance to this field of neurotechnology was after I finished school. I found an open source EEG project on the internet that I used to build a neurofeedback device, and took part in a science fair in Germany. And that was kind of a good boost, was quite successful, was a good boost to kind of continue in that direction. And this is basically, was originally motivated to link, let's say, subjective internal observance and measuring objective reality. So this is kind of where this all came from, and this is also what motivated me to build tools that I would like to allow us to basically look into the brain while we actually interact. There's a third point that I find very interesting, also due to my background, my PhD, that is artificial intelligence. And this is actually the reason why I used this picture in the beginning. This is an original piece of art that was generated for this conference using an OpenAI DALL-E 2. And I put in this prompt that is nature understanding itself through developing consciousness in the human brain. This is what came out among some other variations. And I found that just very inspiring because, right, we wonder what else will be possible in the future and what is creativity, what is consciousness, and so on. So not going too philosophical, but this is really kind of where my interests came from. And as Joe said already, and also Joy's talk yesterday goes really in this direction. I was always interested in putting something on the desk, sitting down, playing around with it myself, and ideally not so much in a constrained environment, but really in an interaction, in an environment that allows us to naturally interact with other people, also with the outside world. During my PhD, I called this neurotech out of the lab, and with David, we crystallized this into neuroscience in the everyday world. So I'll briefly go a bit, in a timeline in terms of the research contributions. This open neurofeedback thing was way back and inspired me to pursue an engineering degree, bachelor and master's, that I finished in 2014. Did my PhD then at TU Berlin in machine learning and neurotechnology, and then joined David here in Boston in a very productive and very good time as a postdoc had to go back to Germany and join NIRS as a director of research and development. As Joe said, here's a short disclosure. So I'm receiving salary from NIRS, but what I'm presenting here now has basically nothing to do with the work that is going on there. It's solely focusing on the academic work that I have been doing. And then now next month, actually, there's another shift that is basically focusing more of my time back into academia as a head of an independent research group back at TU Berlin. And I'm really looking forward to briefly present to you also what we're planning to do there. So focusing on the first third, basically, this, as Joe said, is basically a my way of thinking. This is very challenging, of course. We all know how challenging it is to measure brain activity in general, but then specifically in more naturalistic environments. And during my PhD, I, for myself, basically put it as a pyramid of three main elements, that is instrumentation, paradigm development, and signal processing, and all of this in the context of multimodality. So using more than just one kind of signal. So to move the whole field forward, the idea was to develop instruments that allow us to walk around, that measures multiple modalities at once. That would allow us to do new experiments, and then we can develop new methods. These methods would then exploit all the information that we have. And I think up till today, we don't really use all the information that we actually have in the fNIRS signal and other brain and body signals. Exploit that to improve contrast to noise ratio, to improve robustness against non-stationarities, basically help us to move this whole thing more towards application. On Wednesday, during the neuroergonomics session, I'm going to talk a bit more in general about hardware. Today, I'm going to talk a bit more about the AI aspect or general signal processing. And during my PhD, I kind of identified for myself a power couple, let's say, of modalities. That's EEG and fNIRS. Many people work with those two modalities. There's good reasons for that. It's because they're both, it can be easily integrated, are rather cost-effective, measure complementary information, and so on. So this is why several of the hardware and also signal processing contributions are focusing on EEG and fNIRS. Okay, so this was basically still in the back of my mind when I was doing my master's, developing my own first fNIRS device. And I realized that there was basically no, there was nothing like this open source EEG project on the web that would have made it easier for me. So I decided when the first peer-reviewed publication, my first peer-reviewed publication came out with this device, to put it online, to put it on a website and open source it. And that was really linked to kind of the good experience of being able to use something on the web, do your own thing with it, modify it. And so this is a very basic device. Was not super proud of it, but I thought maybe, you know, other people can use it and expand on it. I myself used it and expanded on it during my PhD to build basically a more miniaturized and multimodal device that I called M3BA, that integrates EEG, fNIRS, other biosensing signals, and accelerometer. That was published in IEEE Transactions in Biomedical Engineering. Was quite well received in the bioengineering community back then. Was on the front cover and helped me really to kind of use that in collaborations throughout the PhD and afterwards. But it was actually just a means to try to measure brain activity in a more naturalistic setting. And basically, this was the second time. The open NIRS was the first time, the second time that I got in touch with the field of neuroergonomics. So we'll talk about this on Wednesday again. But neuroergonomics, amongst others, tries in one of the application fields to measure cognitive workload in real environments. And I wanted to do the same thing, but I wanted to do it in a more challenging, it's already challenging enough, but in a more challenging because naturalistic environment. And so these modules allowed me to build a custom designed headset that would be really custom for this paradigm, for this experiment, measuring EEG and fNIRS. And I will show you now briefly, very briefly, but what this experiment was about, because it basically inspired most of the methodological development afterwards. So you will see a short fast-forwarded video of a participant pressing buttons. This is based on a modified spatial n-bag task. It doesn't really matter what's happening. I think what's important is that this person focuses on colors and when and where these colors appear and has to remember them. And this is, of course, modulating focus and workload. But you can also see that this participant is doing something that we usually try to really avoid, and that is move, and move quite a lot, go down, stretch, and so on. And so we know for a long time that the fNIRS signal has quite some confounding factors in it. It's not only brain that we measure, but back then I was actually surprised to see that because most studies happen sitting down, moving as little as possible, the artifact rejection methods that I found were mostly to remove optoid shifts. That was not so much a problem here because it was a very fitted and rigid design, so there was not much shifting, but we had a lot of physiological motion artifacts, really motion-induced physiology changes. You can see here low pass filtered fNIRS signal from this experiment. And when I add these button press events, this is really always, correlated to a certain body posture. You can see there's something going on that's probably not brain. And when I add the accelerometer, signal becomes a bit more clear. Accelerometer was measured at the head, that there is some co-modulation going on. And being at the machine learning lab at TU Berlin, the question is, of course, what can you do? There's information in these signals. We can see it. It's not super straightforward because it's not well aligned and these delays differ and so on. So I looked into the toolkit of methods available and chose the CCA, or canonical correlation analysis with temporal embedding, as one of the methods to find these mutual components in both data sets. I will come back to this. Well, this was basically my entrance into this whole field of source decomposition and modeling of fNIRS signals. I think we all know we all have the same goal. Aside from generating new knowledge about how the brain works, we actually want to get from the signal that we measure, that is usually very noisy, to a very beautiful estimate of the brain response. And of course, there's many ways to get there. At the machine learning lab, which is actually focused on EEG, BCI before. The standard approach was the generative linear mixing model, and the GLM that we use is a special case of this. But in the generative linear mixing model, we have our observed data, and we usually have sources that we would like to reconstruct if we don't know them. In the GLM, we assume we know them, and we try to find only the weights for those sources. But basically, in this approach here, the idea was trying to find, let me see if I can put the laser pointer, try to identify sources in the source space that basically depict or represent these slow motion artifacts that were so slow that you can't just low pass filter them out because they're in the same frequency band that the HRF, and that was actually intended, but of course made it a bit harder. There's a lot of challenges in NIRS decomposition that I will not go into because of time constraints, but it's, I think, a very interesting field that will also remain interesting for quite a while. And this was, for me, my entrance, basically just trying to solve this problem. I want to measure and classify brain activity, but there's too much artifact. So to solve this, we developed a method that we call Blizzard that uses TCCA and a specific kind of independent component analysis to reject these slow motion artifacts. And what you see here is basically the average artifact at each button, at each position, basically, at each body position also, across trials and participants. And on the left, you see always the average artifact that is, by the way, in the same order of magnitude and even bigger than the HRF. You can see it before the filtering and after basically you don't see so much. So that's a good sign. We were able to reject these artifacts by roughly 2 orders of magnitude. There was a study published in NeuroImage, and that was, again, actually a means to now the end, which is that we wanted to do cognitive workload classification using only EEG, using NIRS, using EEG and NIRS in a basically standard BCI pipeline. And the results were actually quite promising. So depending on the different conditions that we classified against each other, we achieved performances between 80 and 90%. Pretty comparable to, let's say, the standard NBAC tasks that were published before with fNIRS, but now, pulling this out of data that was before usually considered useless. So this was basically the PhD. So my close circle over all those 3 points of the pyramid, and I was very happy to have and lucky to have David in my committee and join him afterwards here in Boston. That was a great time, and also very thankful that we were able to collaborate and are collaborating to this day. So what I'm talking about now is just basically the cool things that I was involved in here in Boston. This is basically the past now. This is a bit of the ongoing and the future. NINJA-NIRS is something that is spearheaded by David and by Bernard Zimmerman and David's team. That is this basically next generation, much more powerful open source device. And I was very happy to help contribute a bit there, and also with the Ninja cap. That's David's idea of a 3D printed customizable cap. If you're interested in discussing this and the potential idea of finding standard high density layouts, then please come to the poster session tomorrow. There's also some other posters that are all related to this project that is really cool and ongoing. So I just want to basically shout this out. So that's sessions 3 and 2, so tomorrow and day after tomorrow. What you see here then is a concept that we basically developed and that was funded, that was crystallized into neuroscience of the everyday world. This is basically the idea to integrate high density EEG, high density fNIRS wearable with eye tracking and object recognition, and with the vision, which is of course still quite far in the future, but to enable measuring brain activity in naturalistic interaction while keeping quite a good control over what's actually going on, what are the stimuli. So this has been funded, and David and David's lab and bio faculty are working on this, have been working on this, and we have been collaborating. We have described this concept in a paper, and if you would like to see first results, aside from continued hardware development, the posters I showed before, then I would like to also point to these 2 posters that are going into the applications. So that's poster session tomorrow and day after tomorrow. Okay, I will bring this up again on Wednesday because neuroergonomics is a very good example for what makes this whole challenge so interesting, but also so challenging. So the 3 pillars of my pyramid, they all try to achieve the same thing, and that is basically getting more robust brain estimates, reduce the variance, increase the contrast to noise ratio, and specifically in brain-computer interface or neuroergonomics applications. But generally, it gets more challenging when we do real-time analysis or single trial analysis. And the whole point here is that I think that if we include more knowledge and more measurements of peripheral physiology, then we will be better at modeling the physiological signal, and that needs to be included into single trial analysis. I will talk about that in the remaining minutes. There's a paper that is recent. I'm sad that Felix Schörkmann is not here in person, from him and the ETH Zurich group in Bern, that is basically giving an outlook again on how systemic physiology is kind of really relevant to explain fNIRS signals and how that can be used. So basically the work that we then did in the last years basically focused on trying to improve these models and the modeling of physiology. This is where TCCA comes in again. So what you see here is just my approach of trying to depict a general linear model, all the things that go in there. Usually nowadays it's long channels and some short channels and some other regressors. And basically what we did is something that is not very complicated. It is that we said we don't put in the short channels as regressors right away. We actually put these short channel signals among any other auxiliary signals that we measure, so blood pressure or accelerometer. We put this into the TCCA pipeline to identify regressors that are more optimal to model the noise. And those regressors we then put into the GLM. So the GLM actually looks pretty much the same as always, but uses more optimal regressors. And that's a study this method actually we published in NeuroImage, and we were able to show that depending, of course, on the conditions and so on, but the contrast to noise ratio measured via correlation, root mean square error, and then also F-score went up dramatically, or could go up dramatically depending on the HRF magnitude. So that's basically trying to improve our current models for block design analysis. There's, of course, a lot of other great work that is exactly trying to do the same. And I think the idea is to merge all of this into something that at some point can be called a gold standard. Then I would like to just briefly point out ongoing or like recent work. Antonio, David's postdoc, recently published in Neurophotonics, basically the real-time implementation of this. So the GLM with TCCA in a Kalman filter for single trial analysis of finger tapping. And there's another poster tomorrow that looks at fast and slow motion artifacts, how to basically try to remove everything that we don't want in the signal. So that was block design. And then there's also single trial analysis. So this before was really the, let's say, standard neuroscience approach. We have a lot of trials. We try to find which brain area gets active, and we can use all our trials for this. If we want to do this in real time and in single trial, then it becomes a bit interesting, because we found that in literature, basically the majority of papers out there that are doing BCI or single trial analysis are not really doing any physiological modeling or GLM. And so basically this is just where we proposed how to do this. It's not that complicated, but the GLM is usually used for, using all the trials. And if you want to do proper data science and no information leakage, of course, needs to be considered how this GLM is trained, how one can use it actually for single trial analysis, and how that can lead to improved classification accuracy. So that was a fNIRS paper that we published, I think 21. Okay, so this is kind of the rough overview, and now I would like to really emphasize again why this reward means so much to me, because basically next month, if nothing goes wrong, I think nothing can really go wrong anymore, but it might, I'm going to start as head of independent research group. I will also stay with NIRS, but the majority of my time will go back into academic work, and of course it helps tremendously to receive this award. So a short outlook into what I'm planning to do. You've seen this. This is basically the whole point of trying to measure brain activity in interaction and with the environment, with other people. And I think it's just worthwhile. And again, I want to point to Felix Schorkmann's paper, pointing out that brain and body are not separate entities. And it might be very interesting to look at how they interact, how the signals that we can measure from both can help us understanding what happens in the brain. Felix calls this the embodied brain. I really like that. I have called it holistic understanding of brain and body function. In this group, I would like to do context-sensitive analysis, so environment context or wearable multimodal data. That's kind of my old thing that I've always liked to do from brain and body, and then now using machine learning and AI, which leads us back to the initial picture. I think AI can do a lot in the future. We'll develop open source tools, and we will develop so open source hardware and software. And I will be looking for postdocs and PhDs. So if you're interested or you know somebody who might be, please contact me. So this is basically it. I have one last slide that is, again, emphasizing this general, endeavor, I would say, that was ramped up greatly by David. So I had the fNIRS idea that was kind of the small first modular device, and this now is much bigger. And I would like to, I will include it there, and it's the fNIRS project. There's already quite some hardware and software, amongst others, available. There is going to be the BITS fNIRS cloud server that allows you to basically look at your data in the BITS format. And this is basically just asking everyone, if you have hardware projects, software toolboxes, this is a community effort. If you would like to contribute this, then please reach out to David or to me. And I think the idea is really just to make it very easy to have accessible hardware and analysis suites on an open source domain in the internet. With that, I would really like to thank all the people I worked with, specifically now most recently David and Miriam and the group at BU, but also my former supervisor. All the faces on here and the names, but also all the people that were otherwise involved. It has been a great fun and pleasure trip, and I hope that it continues to be one. And I would like to thank you for your attention.
Let's just give an extended hand of appreciation for Alex. We're only running like 30 minutes behind, so go have lunch, everybody.
Good afternoon, everyone. Welcome to the postprandial session here. Our job is to wake you up with some great talks. First up, we have Professor Gregory Fisher from Memorial Sloan Kettering University in New York. He's practiced in New York at Mount Sinai for several years, and he's now the chief at Memorial. He's going to be talking to us about the clinical applications of oximetry. Gregory, the stage is yours. Thank you.
Thank you very much for that kind introduction, and thank you for having me. Just a quick question. How many of you are medical doctors in here right now? How many are MDs? Very few. Yeah, there are a couple, very few. Okay, that's good. So, what I'm going to try and do today is I'm going to try and give you an overview of how NIRS is used in medicine. Okay, so what are we doing right now in medicine with near-infrared spectroscopy? You'll often hear us use the term cerebral oximetry because the primary focus where we use it is for the brain. There are some places where they're also using tissue oximetry, so looking at oxygen saturations in the musculature. But my talk today will just focus on cerebral oximetry because that has been around longer. So I do have a couple of one conflict of interest to declare, and that is I'm in the Speakers Bureau for Edwards Life Sciences. They are manufacturer of one of the cerebral oximeters. So what are the objectives of my talk today? Well, first, I want to give you the clinician's perspective. So in other words, the medical doctor's perspective of how we use NIRS. What do we use it for? And how can it assist in patient management? Remember, I'm an anesthesiologist. Probably the most important organ that I take care of is the brain. I mean, I take care of all of the organs during an operation, but the brain is the one that I probably have the greatest influence on because the drugs that I give you to put you asleep, motionless, make sure you're amnestic, they all interact with the brain. However, there are very, very few monitoring techniques that I can use to really see how is the brain doing. The best monitoring technique is when you're awake and you're conversing with me, but you know, you wouldn't want that. The surgeons wouldn't be happy if that would be happening during an operation. All right. What are the shortcomings? And with any type of monitoring modality, I find it's more important to know what are the shortcomings of a technique than actually what are the advantages. Because as you'll see in a second, doctors have, and when I use the term doctor, apologies to the PhDs. I know you guys are the real doctors. I'm only an MD, but make that mistake often. You're going to see that a lot of medical doctors don't understand the shortcomings of the technique of the monitoring modality, and thus they don't understand or they don't interpret the data correctly. And then what is the future of cerebral oximetry in medicine? And this will just be my humble opinion of where I think it should go and what I would be interested in. So all of the engineers here, the gauntlet is down. This is what I want you guys to work on. Okay, so cerebral oximetry in cardiac anesthesia really took off in 2007 when this paper was published. This is probably the seminal landmark paper by a colleague of mine, John Merkin in Canada. And this paper was interesting. What John did was he took 200 subjects. They were all coming for cabbage. Cabbage is not a vegetable for me. It means coronary artery bypass grafting. So bypass surgery, and he took 200 and he randomized them 100 each. Both of the groups got cerebral oximetry monitoring. The way we monitor cerebral oximetry in medicine is we put monitors on the forehead here, and it's just one channel, and what it does is it monitors the cerebral cortex between the frontal and the parietal lobes, which is a watershed distribution. So it's a really good place to monitor to see if the brain is being malperfused. Okay, that's the place where we monitor. So he put them on all the patients, but half the patients, 100, were masked, which means the clinicians did not know what the values were. The other 100, he got a baseline value. So let's just say the baseline value of the patient was 60 or 70. And what they did then during the case is they would intervene to keep that patient's cerebral oxygenation value within 25% of the baseline value. And they used this intervention protocol that you can see here. There's an intervention protocol when they were on pump and when they were off pump. The things that they used were quite straightforward. They would correct oxygenation. They would correct blood pressure if it were low. They would correct blood flow if it were too low. All right. And you could see then lower down here that the interventions, the efficacies were quite high. Now, they followed those 200 patients and they looked for typical outcomes, adverse outcomes like we see in medicine. For us, an adverse outcome, clearly the worst outcome is death, right? But there are other adverse outcomes there as well, being on a ventilator prolonged, which means more than 48 hours, having an infection like a mediastinitis, requiring renal replacement therapy. Now, obviously, for a power analysis, if you only have 200 patients, you're not going to be able to look at each one of these side effects. So what John did was he threw them into a bucket, made sort of like a basket of them. So there was a composite outcome. And when he looked at that, what he was able to show here quite nicely is that the group that they intervened upon had much fewer adverse outcomes than the group that did not, which was masked. This was sort of like the birth, and people got really excited about this and said, oh my God, we finally have found a monitoring technology that can actually help improve patient outcomes if we follow it. Okay. And this is exciting because most of our monitoring techniques that we use in anesthesia, believe it or not, have never really been shown to influence outcome, even though we all do them. I also got involved here, and at Mount Sinai, I looked at 36 patients going for major aortic surgery, and I was also able to show similar things. We also created a basket outcome, and you can see here, depending on the thresholds we used, whether we used 55%, 60 or 65%, and the longer that the patient spent under that threshold, the higher the likelihood was for them to have an adverse outcome. And you can see the lower that I chose the threshold, the worse the outcomes were when they spent a long time under it. There was a German group, Matthias Herrenlocke from, I believe it is Kiel. He looked at patients preoperatively, so in other words, in the preoperative clinic before they even came to the operating room. And what he was able to show was in almost 1,200 patients coming for cardiac surgery, that patients who had a low cerebral tissue saturation oxygenation number under 50% preoperatively, that they did much worse. You can see how many times higher that was than compared. It was almost as bad as having a very high Euro score, which is sort of a frailty index for patients going for cardiac surgery. These two Kaplan-Meier curves show here patients with preoperative cerebral tissue oxygen saturations above 50 and below, and you can see the difference. And if you have here a combination of a low cerebral tissue oxygen saturation with a Euro score greater than 10, you're almost practically inoperable. You will not be alive in a year. Okay, so also stuff that showed we can also use NIRS as a predictive for outcome for our patients. So this is all interesting, but now the questions, of course, that the opponents were asking, well, where are the randomized controlled trials? In medicine, we love randomized controlled trials. We look at that as the gold standard. This is how we prove that an intervention or a drug is better than the standard therapy or placebo, right? So they were calling for randomized controlled trials. They tried to do this in Canada. Why not in the United States? I actually did try to do something like this. I wrote a protocol. I sent it to the Department of Defense. I sent it to the NIH, and unfortunately, it never got approved. Most of these larger funding agencies said this is something that the company should support and not the government. So that was a problem. So the Canadians actually were able to put something like this together, and they tried to do this, and they published a feasibility trial in 2016 where they were able to show that desaturations happened often in 70%, and in almost all cases, 97% of the time, the clinicians are able to use an algorithm to get the cerebral oxygen saturation back to the norm. The problem was they were never able to really complete the trial looking at different outcomes. And one of the reasons why this was so different is because they required numerous institutions, because you needed thousands of patients in order to get an adequate power analysis. And there are different types of technologies on the market right now. And you can see that the problem is these different devices use different techniques, and they wind up having different degrees of precision. So they are all very good when it comes to change from a baseline, but they're very different when it comes to their precision from an absolute number. This confuses medical doctors dramatically. So if you look at one device, like I'm going to pick the INVOS that has a precision of almost 10%, that means if you take two standard deviations, you can have anywhere between 40 to 80% as your baseline number, right? And that just drives doctors crazy. If I put it on one person and it reads 80 and on the next person it reads 40, even though they're both awake and talking with you, it gets the reputation of being a random number generator. So the precision of the device, and this is again for the engineers under you, has to be tight, right? So the Foresight, for example, the Elite, that's the most modern one. This is the older generation of Foresight because the Elite wasn't around yet when Bickler published this, is about 3%, which is about at the realm of where a pulse oximeter is. All right, so that has to be the gold standard, because otherwise you will confuse doctors and they'll have a hard time believing the number that they're seeing. And this was an issue where the Canadians had a difficulty designing the trial. Should we use baseline and then change from baseline? Should we use the absolute number? So they could never really get the trial off the ground. There are other limitations to cerebral oximetry. Doctors will do everything with a device if you give it to them and they have enough time. They put the device on dead bodies. They put the device on vegetables, okay? And guess what? They got a reading. Now, for the engineers under you, you're going to go, well, of course, cerebral oximetry works with chromophores, and it just shows you different absorption. Of course, it's going to work. Not medical doctors. They're not going to believe that, right? They're going to have a hard time understanding what is going on here. This was highlighted in a publication from some of my friends at Mount Sinai. Ron Kahn and Annie Anyanwu published this paper in 2018. And this is probably the funniest paper that I've ever read. If you guys really want a good laugh, download this paper. It came out in the Christmas edition. It was where they actually show jokes. The science behind this paper is perfect, okay? But it highlights the deficiency of the technology, all right? And you can see they write jokes such as a world-class medical institution. They're talking about yams and about squash and stuff like that. And of course, the control people were cardiac surgeons and cardiac anesthesiologists. And it just goes on. And they wanted to use carrots, but one of the investigators ate a carrot, so the whole group had to be discarded, right? And stuff like this goes in here. It's a very, very funny paper. The science behind it, however, is that commonly used vegetables had similar NIRS to the other people, which of course makes sense because there are carotenes in these types of vegetables. And this is probably the funniest thing when you see a yam there. And I like that they were HIPAA compliant and they put the little black bar so that, you know, they wouldn't know who the yam was, who the squash was exactly. But you can see the values are very similar. And again, you're freaking doctors out with this when you build a device that they can put on a vegetable. Now, this says a lot to my colleagues. You might be asking, why do anesthesiologists have enough time to get a hold of vegetables and put their device onto it? But, you know, this is what we do sometimes. It's late at night, we're on call and nothing's happening. All right. So because of some of these side effects that we have, one of the things that I like to do in my practice is use multimodal neurological monitoring. So we don't only monitor cerebral oximetry, we also use processed EEG because the two modalities complement each other actually quite well. Processed EEG, the BIS was a device that was actually developed up here at Mass General. And it's just like the cerebral oximetry devices on the market. It's very reliable. They're very easy to use. And they can show you, or they can help you avoid a catastrophic event if you follow them. And they are quite sensitive to cerebral ischemia when it is extreme. The limitations is they only become isoelectric when there's practically no flow through the brain anymore. It really has to drop down to like 5 to 7 milliliters per 100 gram. That's very, very low cerebral perfusion before they become isoelectric. The other problem is if we're not paying attention in the operating room and we let patients get colder, when we're on the heart-lung machine and the surgeons cool the patients down, sometimes patients will be cooled down to 20, 25 degrees, sometimes even 15 degrees Celsius before we turn off the pump to do some surgery on a patient without blood flow. That will give you an isoelectric EEG. And also, if we dial up our anesthetics too high, you'll also get an isoelectric EEG. So again, you can confuse physicians very easily with these devices. But if you use them together, they actually do work quite well. This is a case I'm showing here where you can see the blood pressure. We went on bypass shortly afterwards. There was this acute decline in the BIS down to 0. The interesting thing is the NIRS, as you can see, stayed almost constant. And the reason why we think this is afterwards we knew that the patient was brain dead, had a massive, embolic event that basically embolized the entire brain. But if you're not consuming any oxygen, then your NIRS doesn't really decline, right? And that's one of the things where the two work together quite well. So where the future, and this is like my throwing down the gauntlet to the engineers here, where do I think the future is going to go? I think the future of NIRS, where it's going to have a much larger impact on medicine past just cardiac anesthesia. Right now, it's used primarily, I would say 95% of the cases of NIRS is used in cardiac anesthesia. But if we could use NIRS as a device to help us detect autoregulation, and especially I'm talking about the lower and the upper thresholds of the autoregulation, that would open the doors not only to cardiac, but it would also open the doors in general anesthesia with older and sicker patients especially. And we noticed that there was something here in my group when we were doing beach chair procedures, and we had patients, beach chairs like the term says, they sit up like in a beach chair and they get their shoulder operated on. And we noticed that every time we'd give the patient some phenylephrine, which is a drug that helps your blood pressure go up, we would also see the cerebral saturations increase. So there seemed to be some correlation going on there. And sure enough, about 10 years ago, Chuck Hoag's group at Johns Hopkins was doing in the lab some work on this, and he showed that he could use NIRS to find out where that lower inflection point is. And what's interesting about it is in medical school, you learn in the textbooks that usually cerebral autoregulation is constant, or the lower inflection point is like at 50, and then between 50 and 150, cerebral blood flow is independent of systemic blood pressure. But what Chuck Hoag's group showed here was that that lower inflection point isn't at 50, it's really higher, especially in patients with cardiovascular disease. It might be 70, it might be 80, and nobody really knows where it is in the individual patient. Now, some background for that. I'm trying to make the case here why this is important. We do know that intraoperative low blood pressure is bad, but when does it really start? So this is a paper from Terry Monk's group, and she showed quite nicely when she followed low blood pressure intraoperatively patients that had quite low blood pressure, systolics of 70, MAPs in the 50s, that these patients, you can see, had much worse outcomes than patients who were normotensive. So having low blood pressure is bad. So we do know that that's true, but what we also know is we're not all the same. All right. And I think that's a really important thing to realize, right? So one patient might have low blood pressure and nothing happens, but another patient with numerous morbidities will not tolerate such low blood pressure. There are some devices like the Edwards device that I was talking about before, the Hemosphere, where they have the Foresight technology incorporated into it. They have this HPI module. This is something that predicts a patient who will be hypotensive. I personally thought that this is useless. And I've told them at Edwards this as well. They weren't that happy with me saying this, but the point is, how do I know what hypotension is for the patient that I'm treating? If I look at a population, I see the population health, like Terry Monk's paper. Okay, that's fine. But I'm not treating a population. I treat one patient at a time. So how do I know that the patient I'm treating right now is going to be hypotensive? Here's an example. I can be doing a surgical procedure. It can be the same surgical procedure. Let's say you're having your colon removed, for example. But the one patient might be a completely healthy 20-year-old. ASA1, that's a grading system we anesthesiologists use to show you how healthy a patient is. But then on the other hand, you might have an 87-year-old patient with, sorry for the acronyms, but doctors love acronyms, it just confuses people. That's why we use them. Coronary artery disease, congestive heart failure, severe aortic stenosis, insulin-dependent diabetes mellitus, chronic obstructive pulmonary disease, and chronic renal failure, right? Same surgical procedure, but two entirely different patients, right? The one patient will probably tolerate low blood pressures quite well. Let you know a secret, it's the 20-year-old. The other one probably won't. So all generalizations are false, including this one. All right. So how can cerebral help us? So back to cerebral autoregulation, you can see where the lower limit of autoregulation. This is a nice review from John Drummond, who's a neuroanesthesiologist in San Diego. And you can see that these papers, he put this very nice together. There's a lot of variability where that lower inflection point is. It ranges everywhere from like in the 40s all the way up into the 80s, right? So you're seeing right now the individual patient can have completely different lower inflection points. And it's important that we could pick that up. And I think that cerebral oximetry could do this. And how can it do this? Well, there comes this autoregulation index. So we know that the value that we're seeing is basically a product between cerebral blood flow, hemoglobin concentration, and PaO2. Now, if we use high temporal resolution here, like we are measuring this really a couple times a second, all right. And I apologize to the engineers if I'm butchering your language anyway. Okay. But this is sort of like as an MD, how I have a simplistic understanding of this. If you look at this at a really high resolution, you know hemoglobin's not going to change, right? And you also know that the PO2 is not going to change. So really SCO2 is a function of cerebral blood flow. Now, remember what we talked about before with the autoregulation. If you're in the autoregulation realm, if you're in that area, you should not have changes in cerebral blood flow based on systemic blood pressure. So there should be no correlation between systemic blood pressure and SCO2 values. I hope I'm making that somewhat clear. So that's the concept of the autoregulation index, that there's a correlation between MAP and cerebral oxygenation. And if the cerebral autoregulation is intact, there should be no correlation. So that index should be under 0.3. We've done some of these trials at Memorial Sloan Kettering, and we were able to show this on a real live patient. And you can see right here that when you let this run long enough, how the autoregulation index is in the autoregulation phase is very low. It's under 0.3. And as soon as you go lower with the blood pressures, it increases. In other words, blood flow and cerebral oximetry, they start correlating with each other. And when you go over the upper limit, they start correlating with each other as well. Now that tells me, as an anesthesiologist, I want to keep my patient's blood pressure in that range where the autoregulation index is very low. So conclusion to my talk, cerebral oximetry used primarily in cardiac anesthesia. We know that, but it would be nice if we could find a way to bring it to the masses, right? Not only have it used by cardiac anesthesiologists, but have it used in general anesthesia by general anesthesiologists. There are numerous devices with different technologies that has certainly harmed the development or the making it available for the masses. It's still too confusing for physicians to really get behind it completely. And autoregulation, I think, in my view, is the way of the future. For that, I thank you very much for your attention, and I'll be available for questions.
Thanks very much, Greg. Really enlightening and good to hear from a fellow clinician. Have we got any questions from the audience? Greg, in terms of the clinical uptake of these monitors working in the same space, the last 10 years or so has, you had Merkin's paper and so on. The last 10 years has seen us using instruments which the gathered audience over here would regard as very crude, old pieces of engineering that simply are not relevant to a group of engineers and physicists in 2022. But the problem is that those clinical instruments have changed the way doctors think about cerebral oximetry. And there's a big group of skeptics out there going, why do we need to even look at this tech anymore? We've proved that it doesn't work. How do you deal with that as a clinician teaching younger people?
So it's kind of like what I said before. When you develop a device which you as an engineer, you understand. But you have to understand the way an anesthesiologist thinks. We are like, often you'll hear the analogy of pilots. So you're there and you have 20 instruments ahead of you. You're watching the patient, you're watching the surgeon, and you have to integrate all this data. You cannot have a device which is complicated where you have to know too many limitations on it. Otherwise, clinicians will quickly go, it's not worth it, and it has to go away. Now, why our devices are so crude is because the technology that you guys develop today has to pass the FDA in this country, right? And that's one of the big holdbacks. So it has to become FDA approved before we can start using it in the clinical setting. And that's one of the problems that we have. Now, what I try and do with my colleagues is I try and educate them as well as I can, but that's often one of the bigger problems, of that once people have made up their mind, once they've had a bad experience with it, they sort of just push it aside and it doesn't get used anymore. So it is very important that when you design something, that it might be good if you do partner with a physician and you sort of get the physician's view on it, if you want to bring it to market and you want it to be successful and used on patients.
Great, thanks.
So I just want to ask, so the cerebral oximeter took a good long time to become well established, at least for cardiac surgery. The autoregulatory index looks really quite promising. So what do you think that pathway towards widespread adoption utility will look like?
I think it's,
what limitations do you see, if any?
Yeah, I do think it's moving quite rapidly now. The problem is that it would be naive to think that the autoregulation index is a static physiological phenomenon. And you have to understand, when we provide care for a patient, there are numerous things that are taking effect that can alter the autoregulation index. So there's temperature of the patient, there's the hemoglobin. So if you go on the heart-lung machine, for example, that heart-lung machine is primed with a crystalloid solution. So as soon as you go on it, you immediately have a 10 to 15% hematocrit drop. right? So that would be 3 grams per deciliter hemoglobin drop, right? So you're changing hemoglobin very dramatically there. That can have an effect on the autoregulation. What's your CO2? What's your PO2 doing? Glucose management. So there are many, many things. Then our gases that we give, they all interact as well. So it's something that's very complicated, and it needs to be looked at, and it needs to be used in real time. But that precisely is one of the strengths of the cerebral oximeter, that it is readily available. It's easy to employ in the operating room, and it would, whatever you're using for a technique like what I just showed you there, where they use these baskets of constantly measuring the autoregulation index, you would need to have a device that's constantly updating, because I could imagine if you're doing a long anesthetic, 6, 8, 10 hours of anesthesia time, your autoregulation index might be very dynamic, and that lower inflection point might move.
Just looking to the future, would you think that one of these very high node count, I mean, how many points would you need to know the oxygenation in the brain ultimately? Would you want to know it at all over the brain, or would you only need to know it left, right, or would you only need to know it in one spot?
Yeah, no, what you're bringing up is one of the main problems that clinicians have with it as well. So in cardiac surgery, there are 2 types of stroke that we talk about. One is embolic and one is low flow. The cerebral oximeter is really good for low flow types of stroke, watershed strokes. It picks that up really well because that's more of a global problem. What it's not good at is embolic strokes, because you're only measuring with our 1-channel system here on the forehead. You're only measuring like a cubic millimeter of tissue, right? So if the stroke's not hitting that area, you'll never know. And I've had numerous patients where the cerebral oximetry has been normal, but they've had a big embolic stroke where it went up, usually goes up the right, the right side goes into the middle carotid artery. Patient's left-sided hemiplegic, and nothing happened to the cerebral oximeter, but the patient wakes up in the ICU with a dense left-sided stroke. And that's the problem. For that, you would need to have numerous channels. And I was sort of interested in looking at some of the things that were being displayed over in your vendors here. And it is interesting how they have these multiple channels. But now the difference becomes here is as soon as you start using different distances, are you really measuring now the cortex? Because the depth is, of course, half the distance between the emitter and the receiver, right? So if you can put the emitter and receiver wherever you want, you might be going too deep, you might be going too superficial. So now you're going to really start confusing people. And I think that's one of the reasons why cerebral oximetry in the clinical setting has remained just on the forehead. But ideal, it would be lovely if we knew the whole brain. Did you turn him off? I think my time's up.
I'll try.
Yeah.
Okay, now it works. I have a question too. So you put a slide up that says saturation is equal to flow, which, I mean, not entirely true, right? It's, I mean, of course, it's driven by flow, but some of us are measuring flow directly, and we're wondering from your perspective, do you see more information? Is information more useful if you're monitoring blood flow directly as opposed to its impact on saturation?
No, I think flow would be ideal. And again, I'm an MD, so you got to give me a pass here, right? When it comes to things like this. It's in my brain. That's how I try to explain this to myself, how it works. Would it be better to see flow directly? Yes, it would. But flow is a problem that we have in medicine in general. What do we measure when you go to the doctor's office? They measure your blood pressure. It's not the blood pressure that's relevant. It's really your cardiac output that people want to know, but it's hard to measure the cardiac output. You're not going to be swanning people. You're not going to be putting in a pulmonary artery catheter to measure your cardiac output, right? So that's one of the reasons why even back then we used blood pressure as a surrogate marker, even though it's not necessarily flow either. Yes, there's a correlation over SVR, but it just makes it simpler for us. If you could get me flow, I'd want flow.
All right. That's great. Thank you very much, Greg, for an enlightening talk. And the challenge is laid down to the engineers. Give us stuff we can use in the OR. So we're going to move on to our standard length presentations. And because we had a slightly late start to the session. We're going to be ruthlessly keeping to time. So each presentation cannot be more than 15 minutes. So we're going to ask presenters to keep to 11 to 12 minutes. And then you'll hear a certain cough and either Stefan or myself will stand up. That's your cue to wrap up. So you've got time for questions, because questions is where the cool stuff happens. So I'd like to invite Luis Bortoleto up to the stage to give the first of the shorter talks. Luis. Right. Yeah, I'm going to need some tech help here rapidly. Oh, there we go. Marie, help. There's no cursor. There's no nothing here. No, that's. That one's too. Is there? I'd say other.
So you found it.
Can you find the actual cursor?
Too close is here.
No, the cursor. Show me the mouse.
Oh, the mouse. I had the same problem earlier, and okay,
No, that was the.
Oh, plus this one too. So click again. I'm sorry.
So maybe it doesn't have a screen, right?
And here is to get to the screen.
And that one.
Yeah, the mouse, you have to do this. I don't know why.
All right, that'll teach you to put a doctor in charge of tech.
All right. Good afternoon, everyone. It's a huge pleasure to be here at the fNIRS Congress. And today I'll be presenting to you guys the work that we've been doing where we measure patients with carotid stenosis using fNIRS on a resting state protocol. So to start off this presentation, it's really important to understand the brain vascular supply system. The brain is mainly vascularized by 3 arteries, which are 2 of which are the internal carotid, whereas the focus of our study here. The internal carotid is going to supply mainly the anterior and also the middle left and right hemisphere regions. And therefore, if we have any impacts on the hemodynamics of the carotid, it's going to directly impair the cerebral hemodynamics. So our study is consistently based on the measurements of patients with internal carotid stenosis, which is the partial or total clog of the internal carotid artery by a plaque that is consisted of fat and cholesterol and atherosclerotic plaque. And the symptoms of this disease are very dangerous. They can lead to brain damage and also death in most cases. And they might happen when fragments of this built-up plaque go to the vascular system. It then can interrupt the blood flow on the blood vessels of the brain, leading to hemorrhagic and also ischemic attacks. So it's very important to understand how these vascular supply deficits are caused by this disease. And we often use vasoreactivity tests to measure the vascular ability to respond to vasodilatory challenges. And with this vasoreactivity test, we use stimulus to induce vasodilation, normally inhalation of CO2 and also administrations of certain drugs. And breath holding is also a technique used that is cheap and also easy to perform. And in the clinic, it's mostly used the TCD, the transcranial Doppler ultrasound, to measure this vascular supply deficits. However, this technique only measured the impairments of the large arteries because we're measuring the mean blood flow velocity on the larger arteries. Therefore, we don't have a measurements of the impacts of the microvasculature of the brain. Therefore, we previously did this study on our lab where we measure patients of stenosis patients using a breath holding test with fNIRS. And what we can see here is that the patients have a very increased time delay response and when we do the breath holding tests when compared to the controls, which states that the fNIRS is sensitive to the carotid stenosis. But this protocol imposes a lot of challenges for us because most of the patients already have a vasodilatory problem. So it's going to be hard for them to hold their breath for 10 seconds or longer. So we had to think of a more accessible and more flexible protocol that could be used as for the elderly population. And there's no better way than the resting state. So our hypothesis behind the study is that the impairments of the cerebral hemodynamics could be seen on the spontaneous fluctuations of the brain hemodynamics. And our goal here is to understand how these hemodynamic impairments might change the cerebral connectivity. So to do that, we use a NIRS system, and we try to build our probe mainly focusing on the internal carotid supply region with 16 sources and 32 detectors, using 48 channels and also with a temporal resolution of 8 Hz. And then we performed one session of resting state. This resting state is just a 6-minute resting with the eyes closed, seated down in a silent environment. And we did that for 50 patients. All these patients underwent magnetic resonance angiography to detect the levels of the stenosis. And then we also gathered data from 20 controls. From our participants' demographic info, we divided these patients into 2 groups, the bilateral and the unilateral patients. Being the unilateral, the patients that only have one carotid being obstructed by more than 50%, and the bilateral when the patients have both of their arteries being obstructed by more than 50%. And we did that because, well, dividing in degrees of stenosis, we have a low amount of participants that could be divided in the degrees of stenosis groups. So we decided to reduce the complexity and divide into unilateral and also bilateral groups. Also, for the sake of the analysis, we defined the left hemisphere as the higher occlusion side and the right hemisphere as the lower occlusion side. For our data preprocessing fluxogram, we use the usual preprocessing routine that we do on our lab. So at first, we discard the channels that have a bad signal to noise ratio, lower than 8. And then we convert the raw data into optical density. And with the optical density, we correct it for motion artifacts. And this motion artifacts, we use a hybrid approach that is the combination of spline followed by wavelet transform. And then we convert it to hemoglobin changes in concentration. And with that, we use a bandpass filtering to focus on the low frequency range. And then we try to remove the global signal. Here, I like to emphasize that we don't have any short channels, neither physiological measurements, auxiliary for our measurements. So we try to use a statistical approach, which is the PCA, the principal component analysis, to regress the first principal component. However, when we did that, we saw that the covariances for the first principal component varied across groups, increasing the covariance as the severity of the stenosis increases, which might be related to removing some information that might be useful for us to show that there is cerebral hemodynamics impairments on the microvasculature. All right, so then we divided this preprocessing fluxogram into 2, one with global hemodynamics and the other one without the global hemodynamics. And then we use a pre-whitening filter to remove the autocorrelation from our data. And then we computed the weighted graphs. We just did the Pearson correlation values for all the combinations of channels. And after that, we use a Fisher transform to convert to z-values. And with the z-value matrices, we build our weighted graphs. And with the weighted graphs, we computed the functional connectivity parameters. Here also, I like to emphasize that for this presentation, I'll be only showing the average node strength for the parameters, but we're also looking at the clustering and also the efficiency. But that's a future step for our study, to looking further into the functional connectivity parameters. So first, we analyze our data using the usual preprocessing routine without the global hemodynamics. And I'll show both of the chromophores here. They're not really here on the left-hand side. We have the average networks, and there are no differences in the local, neither in the global connectivity without the global hemodynamics. And also when we compute the network strength, we don't see much differences in the mean value, neither in the variability inside each group. But And then we introduced the global hemodynamics into our data. So the first thing that we see for the oxyhemoglobin signal is that there is an increase in connectivity that is expected since we are introducing more of the system component on our data. However, we start to see some tendencies different the groups. The first one being that there is a decrease in the connectivity when we increase the severity of the stenosis. But then we computed the deoxyhemoglobin signal, and that's where we found our most unexpected result. At first for the unilateral group, we have a more dense and more connected network for the anterior circulation, and for the bilateral group, we have a more global partner, increasing the connectivity. Here, also on the right-hand side, we have the network's strength for either the oxyhemoglobin and the deoxyhemoglobin. And we see that the unilateral group is in the middle, following the pattern of increasing the connectivity as the severity of the stenosis increases. So we decided to divide this study into 6 regions of interest, following the main vascular pathways, basically the anterior cerebral artery, the middle cerebral artery, and also the posterior cerebral artery. And we computed the average node strength for all the regions of interest. Here I call it the ROI strength. And well, for this graph, it's very hard for us to see some differences, but we start to see some differences in the local connectivity. So to get a better view, what we did is we computed the relative to control connectivity. So we divided the patient's connectivity average node strength with the relative to the control average node strength. On right-hand side, we have this graph that's easier to observe. We don't see much differences, as I said before, for the oxyhemoglobin. However, we start to see some tendencies for the different hemispherical sites. And again, when we compute the deoxyhemoglobin local connectivity, we see a more dense and more connected network, mainly focusing on the anterior circulation for these patients. And also, once again, the unilateral group being the middle and the bilateral group with a more highly dense connected network. So in summary, we introduced a new protocol with this study that has potential to measure the carotid stenosis impact in the cerebral microvasculature. And because we see differences in the hemodynamics, that seems to scale with the severity of stenosis and mainly focusing on the deoxyhemoglobin signal. And also that the global hemodynamics might contain some valuable information that's often discarded by us, but that could be used as a biomarker for this disease. And also that the connectivity profile may vary across brain regions with a higher preeminence on the anterior circulation. And here, I'd like to thank everybody that support to this study, my Professor Rickson, and all of the students made part of this study. Thank you.
Thanks very much, Luis, and impressively kept to time, very succinct talk. Have we got some questions there, Elias?
Yeah. Hi, very interesting work. Can I just ask you, have you investigated the effect that your bandpass filter will have? Because, you know, it's quite a heavy one, and then you apply the global removal of the hemodynamic. Might be interesting to see whether you get more information on the global hemodynamic before you done the bandpass filter.
Yeah, that could be a very interesting approach too. We didn't think about that at first. We mainly focused our bandpass filtering on the low frequencies that is already established in the literature, but that's quite interesting. Yeah. I'll be looking at that further.
Because most of that had to do with neuronal function. But if you're interested in the hemodynamics, you know, including the systemic, you might want to remove that bandpass filter and look at other frequencies.
Sure.
Luis, quick question from myself. You chose controls who were obviously similar age, but how did you make sure they didn't have asymptomatic carotid stenosis? Did you scan them as well?
Right. We didn't scan the participants that made part of the control group. That's a very interesting point too, because will increase the variability, and I think that gives us a more robust approach for the study, since we don't know if the control participants have the stenosis or not. But in the future, when we do more collecting data, we will probably be checking that too.
Great. Thank you very much for an excellent talk.
Thank you.
Okay, next up we've got Deep Shikhar Acharya, who is coming up right now to give us her talk, and I'll see if I can get the audio visual to work a bit better this time.
Perfect. Hello. Hi. Am I audible? Yeah, perfect. Okay. Hi. Thanks. My name is Deep Shikhar Acharya, and I'll be talking about the influence of cerebral perfusion pressure on neurovascular coupling and linking it to autoregulatory health. So we all know that the brain is a metabolically expensive organ. It actually consumes an enormous amount of oxygen to keep up healthy functioning. And this is facilitated by cerebral blood flow. And this cerebral blood flow, in turn, is modulated and regulated by something called the cerebral perfusion pressure, essentially the pressure that perfuses the brain. Given how important it is to get an adequate amount of oxygen for healthy brain functioning, our brain actually has a mechanism in place called cerebral autoregulation that ensures that even when the perfusion pressure changes within a certain range, our cerebral blood flow remains largely constant. And one of the ways it achieves it is by changing the diameter of the arteries in the brain. However, there are a host of diseases where this ability breaks down. Some of them are stroke, hydrocephalus, traumatic brain injury, hypertension. And in these cases, even a small change in perfusion pressure can lead to drastic changes in blood flow because our cerebral arteries can no longer further constrict or dilate to compensate for pressure changes. That is why one of the primary clinical techniques is to monitor cerebral autoregulatory health for such neurocritical patients and monitor cerebral perfusion pressure. Unfortunately, the current methods are either invasive, not bedside compatible, and they usually lack a simultaneous measure of neuronal health. This is where we feel neurovascular coupling might come in and be the answer. So we know that neurovascular coupling is essentially changes in local cerebral perfusion in response to local neuronal activity. It has been hypothesized that neurovascular coupling and cerebral autoregulation share common pathways. That is because they both affect cerebrovascular diameter. If the changes in diameter happen in response to cerebral metabolism, it's neurovascular coupling. If it's happening in response to cerebral perfusion pressure, it's cerebral autoregulation. Which is why if your blood vessels can no longer further constrict or dilate because you are not autoregulating anymore, it would affect neurovascular coupling. It's also known that too much or too little blood in the brain, ischemia or hyperemia, can affect neuronal function, hence affecting neurovascular coupling. This is why we believe that studying neurovascular coupling as a function of autoregulation could provide a non-invasive bedside compatible metric for looking at autoregulatory health in the clinic. So what we want to know is, do changes in cerebral perfusion pressure causing changes in autoregulation affect neurovascular coupling, and can then neurovascular coupling be used as a bedside compatible monitoring technique for patients? So let's first look at how we look at cerebral autoregulation. How do we create a control model for it? So we worked with non-human primates. I'll be showing you data across 4 rhesus macaques today. They were all anesthetized with fentanyl and isoflurane anesthesia. We kept the isoflurane percentage under 1%. We placed an A line in the carotid artery to measure mean arterial pressure. In addition, we had an intracranial pressure sensor in the parenchyma. And in addition, we had a saline reservoir going into the ventricle of the non-human primate to change the intracranial pressure in the head. Over an extended period of time, what you see here is an example data from about 20 hours of continuous experiment. We altered intracranial pressure in very slow steps. Each step of intracranial pressure change was about 2 hours long. And in these periods of slow changes in intracranial pressure, we observed changes in perfusion pressure because they're related to each other. So this is how we created a control model for changing autoregulation. So now that we have a model ready, let's look at how we studied neurovascular coupling in these experiments, specifically using tools that are clinically translatable. We studied neurovascular coupling in two ways. One was stimulus-evoked neurovascular coupling, and the other one was resting state. So let's first talk about the stimulus-evoked neurovascular coupling. So it's known that in the presence of an external stimulus, there will be local neural activity, and that causes changes in local hemodynamics. These two are together related by something called the hemodynamic response function, such that when you convolve the HRF with local neural activity, it should be able to predict local changes in hemodynamics. That is why the HRF acts as a measure of neurovascular coupling. So in our experiments, we presented the non-human primates with an external visual stimulus, which was a radial checkerboard. And we simultaneously measured EEG in the visual cortex to measure local changes in neural activity in the visual cortex in response to the stimulus. And in addition, we also had a NIRS device measuring changes in oxygenated hemoglobin in the same region. We used an ISS oxyplex system at 2 different wavelengths, and then we used the modified Beer-Lambert law to calculate changes in oxyhemoglobin. So we essentially have a measure of neural activity and a measure of oxygenated hemoglobin or hemodynamics. And the hypothesis is that the HRF that we would then evaluate at different perfusion pressure values would change as a function of autoregulation. So this is an example data from one of the non-human primates at a healthy perfusion pressure value. What you see here is a continuous time trace of square of EEG amplitude, so roughly EEG power, over about 3.5 minutes of data. And also the simultaneously recorded changes in oxygenated hemoglobin. Each dashed line, each dashed vertical line represents presentation of an external stimulus. Then we define the HRF as a 6-variable double gamma function, which we iteratively convolved with the EEG power till we found an HRF that best predicted the oxygenated hemoglobin signal. And the HRF that we thus found was the HRF corresponding to that CPP value. So for the same non-human primate, this is what the HRF looked like at a CPP of about 40 millimeter mercury. This is what it looked like in the range of about 70 to 90. And this is what it looked like at 100 millimeter mercury. To put it in the context of autoregulation, you would see that the shape of the HRF does not look like what we usually expect outside the regions of autoregulation at very low or very high perfusion pressure. This was seen across 3 non-human primates, and we saw that the healthy shape of the HRF started looking less like the healthy shape as we moved to extreme perfusion pressure values. So what this indicates is that stimulus-evoked neurovascular coupling, specifically the one calculated using HRF, changes as a function of CPP and could act as an autoregulatory indicator. So we looked at stimulus-evoked neurovascular coupling. Now let's look at how we studied resting state neurovascular coupling. Often it is not possible to involve patients in external stimulus-driven tasks. And in those cases, relying on spontaneous changes in neural activity or cerebral metabolism and hemodynamics works better because that involves minimal patient involvement and then also allows for continuous monitoring. One of the ways to study it is to look at the changes in the demand and supply for oxygen in an area in the brain. One of the ways of doing that is using something called the coupling ratio, which is looking at the ratio between CMRO2, or the demand for oxygen, and CBF, or the supply of oxygenated blood in that area. It is also known that this coupling between CMRO2 and CBF changes as a function of autoregulation, specifically when autoregulation is impaired. While measuring CBF is something, or cerebral blood flow is something that we could do with diffuse spectroscopy, estimating CMRO2 itself requires mathematical modeling. We know that CMRO2 is associated with need for oxygen, usually associated with local neural activity, is related to the cerebral blood flow in that region. It's related to the amount of oxygen in the blood and the ability of the tissue to extract oxygen from that blood, or oxygen extraction fraction. This oxygen extraction fraction in turn depends on tissue oxygen saturation and the relative ratio of arterial and venous volume, something known as gamma, or arteriovenous fraction. Now, people who are familiar with CMRO2 literature would know that this gamma is usually assumed to be constant. However, this becomes an issue when studying patients with cerebral perfusion pressure disorders, because we know that to maintain a constant amount of blood flow, arterial volume changes drastically. And once you're outside the region of autoregulation, blood flow changes, but arterial volume can no longer remain constant. And this is actually what we saw across 4 non-human primates. So the data you see in gray is plotted across 4 non-human primates, the arterial volume and blood flow changes. So we used a model which was proposed by Coxis et al. In their 2006 paper, which allows to incorporate these changes in gamma and blood flow. The way we analyzed this in our non-human primates was by using simultaneous measurements of NIRS and DCS. We had a frequency domain NIRS system to calculate tissue oxygen saturation and arterial saturation, and a DCS system that was built in-house for measuring blood flow. And using these simultaneously with the model proposed, we found that our oxygen extraction fraction in CMRO2 largely remained constant in the region of autoregulation, but varied widely outside of it. Now, taking it back to the coupling ratio, which is where we started, you can see that blood flow and CMRO2 are very well coupled across perfusion pressure values. However, they start straying away from linearity as we move to really high perfusion pressure values. This is also observed in the ratio when you plot it, as they're significantly lower at very low and very high perfusion pressure values. What this indicates is that resting state neurovascular coupling, specifically the one indicated by the coupling ratio, changes as a function of perfusion pressure, again linking it to autoregulatory health. So in summary, we showed that neurovascular coupling changes, both stimulus evoked and at rest, change as a function of autoregulation and could act as a non-invasive bedside compatible biomarker for autoregulatory health. In future, we do want to add EEG to our CMRO2 analysis to actually see the origin of the changes of CMRO2. In addition, we are currently looking at clinical translation of this work. Finally, if you're interested to discuss more models of CMRO2 and how they break down, my colleague has a poster in poster session 3, where we're looking at how the current models break down in free divers during breath holds. So feel free to visit us on Wednesday. Finally, I would like to thank the labs that I'm a part of, and thank you all for listening.
Thanks. Microphone at the top here. Thank you. Thanks a lot, Deepshika. Has anyone got any questions about a fascinating topic, keeping the brain healthy? Unfortunately, Greg's had to go off and get a train ride, but I'm sure that stuff would be interesting to him. Can I ask you a quick question about your experimental model? You mentioned quite specifically that you used low doses of isoflurane, but can you just explain to us what the possible problem would be with the anesthetics that you're choosing in that model?
Yeah, that's a good question. So isoflurane is supposed to be a vasodilator, which will affect autoregulation. Given that we're studying autoregulation, we can no longer predict vessel diameter changes if we use high doses of isoflurane. And that's why we kept the levels low and added fentanyl in addition. We actually published a paper where we looked at how our anesthetic protocol actually shows intact autoregulation and only changes autoregulation when we alter it. Yeah, so we did look a lot into anesthesia and autoregulation.
Great, that's really excellent, because some of the early data is from the 1960s, where you have to reference papers using isoflurane of 3 or 5%,
exactly,
which messes things up. All right, thanks again for your talk. Oh, sorry, Stefan is now going to take it from here, including a question.
So I was just wondering, if I understand correctly, what you saw is that CMRO2 was increasing with the blood pressure. I mean, you had essentially you said you had autoregulation of, it almost looked like the same as the expected autoregulation curve. So do you have any reason to expect that?
That is one of the primary reasons why we're looking at EEG, to actually understand it and see if there were any seizure-like activities which might have caused it. That is one of the hypotheses we have, but we do need to look at the EEG data to actually understand why CMRO2 increased so drastically. Yeah. Thank you.
Thank you very much. If there's no other question, let's thank the speaker. I'd like to invite Kyle Caldrick next. talk about the agreement among experimental cerebrovascular activity paradigms assessed with DCS with short separation regression. Yeah.
Does it come out?
Yes.
Awesome. Thanks.
Great.
Well, thanks for the intro. I'm excited to be with you all to talk about cerebrovascular reactivity. So we're going to just jump right in here in the interest of time. CVR is defined as the ability of the cerebrovasculature to respond to a vasoactive stimulus. So specifically in this presentation, I'm going to be referring to the changes in arterial CO2 concentration. Now, compared to other biomarkers we are familiar with in our community, such as tissue oxygenation, CVR is thought to be a more specific indicator of the brain's vascular health. And quantification of this biomarker is of growing interest in the research community, especially over the past 2 decades, as you can see here, especially as new methods to assess it have been developed. It's known to be impaired in all of these disease states you see. And you may know that cerebrovascular dysfunction in particular can lead to brain injury that's secondary to these primary disease states, adversely impacting patient outcomes and quality of life. So quantifying CVR is an unmet clinical need, where clinicians need feasible and validated methods to perform these assessments at the bedside on demand. If we were to address this need, it would be of immense help for any type of brain injured or hemodynamically compromised individual, because clinicians would have a viable tool to inform their medical management that are aimed at restoring abnormal CVR and preventing any type of secondary brain injury. I have to thank Luis. He set me up really well here to talk about this topic. CVR is measurable at different length scales. A lot of this will be familiar now, both at the macro and the microvascular scale, and it exerts its effect at any vascular location with smooth muscle. So as Luis mentioned, in the macrovascular length scale, this is commonly assessed at the middle cerebral arteries, and the microvasculature, we're looking at the peel artery network and the precapillary sphincters as our primary drivers of localized blood flow regulation. The macrovascular side, we're looking at transcranial Doppler as the preferred tool of choice for most researchers in the clinical space. And on the microvascular side, a bold MRI. With respect to CVR being a clinical need, these tools are primarily used in research context today. They do have their limitations, but they also have utility as well. Some of the limitations are the need for experienced personnel and the obvious lack of bedside capability like MRI. And we know that when the neurovascular unit in particular is damaged due to these primary disease states I just mentioned to you all, the macro and microvasculature kind of decouple with their blood flow regulation. So there is a strong motivation here to do CVR quantification at the microvascular level to prevent these adverse outcomes. And given that we can assess CVR at this length scale, there's considerable potential for optics to play a role in addressing this unmet clinical need. You all are familiar with the benefits. In particular, because CVR is examining the blood flow response to a perturbation, there's an exciting potential for diffuse correlation spectroscopy to do this assessment, given that this optical modality in particular provides an index of microvascular blood flow. And this may be advantageous over existing NIRS-based platforms like the one we just heard about with cerebral oximetry, because these devices could provide more indirect assessments. So regardless of whatever imaging modality you use to assess this, you also need a corresponding experimental stimulus. So in the present day, the hypercapnic gas challenge is the gold standard, where we we inhale medical grade CO2 at a higher concentration that's found in room air. And the blood flow response to the changes in arterial CO2 is quantified. So when I'm referring to CVR in this presentation, I'm specifically referring to here the percent of blood flow index change to any millimeter change of end tidal CO2, which is a surrogate marker of CO2 changes. So this gold standard in particular has its advantages. It's a potent vasodilator. It's relatively quick as far as its onset time for the stimulus. Yet, despite this, it's really clinically infeasible to perform in a wide range of settings. There's a lot of equipment you need, and for certain individuals who are hemodynamically compromised, it may be contraindicated. So as Luis said, there's a lot of other more tolerable paradigms that could help you make this assessment. The breath hold challenge. Anyone in this room can hold their breath for 20 seconds, and that's far easier than inhaling CO2 gas for 6 minutes. So while that's advantageous in particular, you can imagine it'd be difficult in young children and impossible with sedated individuals. So the holy grail here would be to do a resting state assessment, since this procedure could be virtually adopted in any patient cohort. Now, if you look at the literature, it's really interesting that CVR tends to be used interchangeably among all these methods listed here, yet there's a lack of rigorous studies that truly quantify and validate that agreement. And so you could say there's a research need to validate that CVR measured with these more tolerable experimental paradigms are substantially equivalent, to use that FDA language, to the hypercapnic gold standard. So if you were to design a medical device for a CVR quantification, you would want to show that these more tolerable stimuluses are comparable to that gold standard. So we set out to address this need by using diffuse correlation spectroscopy, where we hypothesize that CVR assessed during hypercapnia agrees well with that during breath hold and resting state exam. So we enrolled in this study 27 healthy adults. We had some repeated measures yielding 31 data sets. And we subject our volunteers to this experimental protocol you see here: a 10-minute resting state observation, a breath hold examination consisting of 5 repeated epochs of paced breathing for a minute and 20-30 seconds of holding your breath, and a hypercapnic gas challenge consisting of a baseline, a 6-minute CO2 inhalation, and a recovery period. I'm going to bring the lab to you just to give you a sense of our experimental overview here. We acquire our DCS measured signal at 20 hertz, downsampled to 1 hertz, and we acquire this blood flow index at 1 and 2.5 centimeters. We use standard respiratory circuitry, fairly easy to deliver this 5% CO2 gas to our volunteer. We monitor end-tidal CO2 using capnography. We use pulse ox and blood pressure monitoring to keep our eyes on any systemic changes that may be occurring. So now, using this hypercapnia data here, I'm going to just show you a representative timeline to show you our analysis process briefly. We have our blood flow index measured with DCS at the short separation in red and the long separation in green for our baseline CO2 inhalation and recovery period mapped against a CO2 trace here. We calculate a relative blood flow index trend. We extract our end-tidal CO2. We make sure all of our axes are sampled correctly and time-aligned with cross-correlation to account for gas sampling and physiological delays for oxygen exchange. And finally, we take this green long separation relative blood flow index and this blue end-tidal CO2 trace, and you can simply quantify CVR, as most people in the TCD community do, by a simple linear regression. You can see the representative time series and breath hold here, noting the 5 epochs of breath holding, and our resting state period, noting considerably less variation in these metrics. So now, if we wanted to look at the agreement between hypercapnia and breath hold, you can see that plot here, and the agreement between hypercapnia and resting state. Right away, you'll see the agreement is pretty poor. And this finding was not only interesting, perhaps unexpected, but also it was novel for us because, to the best of our knowledge, a direct comparison among these experimental paradigms has not been shown robustly when using any optical modality that I'm aware of. And on the surface, it suggests that using a simple linear approach with a DCS measured cerebrovascular reactivity biomarker does not allow us to claim that a medical device could be substantially equivalent for using these more tolerable paradigms. If we drill into the time series using breath hold as an example, you'll notice that our data shows systemic confounding contributions to our DCS measured blood flow signal. And you can see this by the mean arterial blood pressure trace here. So perhaps this lack of agreement I just showed you is explained by these confounding systemic influences, which we need to be accounting for in our optical signal. And we're, as an optical community, in luck because we have access to that information, especially anyone who does fNIRS is very well aware of that. And that's a huge asset over other techniques such as MRI, where access to some sort of systemic blood pressure signal, for example, is practically non-existent. So what if we took this basic numerical approach and just took our cue from fNIRS and applied a short separation regression in the context of a general linear model to quantify the CVR of interest, which is hopefully a more faithful representation of the brain's blood vessel response to changes in end-tidal CO2. Perhaps that would help our agreement here, and in fact it did. I'm showing you for hypercapnia and breath hold the same data on the left using the standard linear regression approach, and then you can see the short separation regression here. A very strong agreement noted by all these correlations and this concordance correlation coefficient, which is commonly used as a way of comparing a new method to a gold standard, and any value greater than 0.7 is considered quite strong or close to substantially equivalent, if you will. If we look at the same data now between hypercapnia and resting state with our standard linear approach and hypercapnia and our short separation regression, you can see the very strong agreement. which is actually really exciting for a resting state exam. To be able to do a cerebrovascular reactivity assessment with resting state using DCS for a blood flow index measure would be immensely valuable clinically, in addition to some of these metrics we talked about, like autoregulation. So we were curious if any of these agreement comparisons that I just showed you were improved using a 3-layer model, for example. And so while we did use a 3-layer analytical technique for diffuse correlation spectroscopy, oh, hey, slides, that's fun. I saw some faces over here. I was wondering what's going on. There we go. Okay, so we did look at the 3-layer analytical technique to see if we can improve upon the agreement that I just showed you with the semi-infinite standard process for analyzing our DCS signal. And we actually did find that using the semi-infinite approach with the short separation regression demonstrated our strongest agreement here. So that was really nice. There's one caveat when you compare our results to the literature that I'd like to share with you. We observed that our CVR values are pretty blunted, I guess, compared to the reference range. So a lot of our values shown here fall within less than 3% per millimeter of mercury change, where CVR measured with other modalities like ASL MRI and one recent DCS study kind of show this 4 to 8% range as noted on the slide. So we observed this blunting effect with our short separation regression. We didn't see it as much with the linear regression approach or any of the 3-layer techniques. So to explain this, it's certainly possible that the systemic influences we're accounting for and that we are regressing out are caused by other factors that change cerebral blood flow in tandem with the effects that dissolve CO2. So in these results, we may be regressing out too much of that desired brain layer signal that's indicative of the cerebrovascular reactivity response. So this blunting effect could be caused by a suboptimal source detector separation. Here we're using 1 centimeter. It's common to use less than that in fNIRS, for example, or not having sufficient brain sensitivity, which we're using a 2.5 centimeter separation here. And we saw for a lot of our subjects that there may be evidence that we're not getting enough brain sensitivity at that long separation. But another thing worth pointing out is subject positioning. So some of our blunting may be due to this. So in our studies were performed with subjects seated, but studies were performed in the MRI community, primarily done supine or laying on your back. And we know from the TCD literature, some recent work, that a seated posture decreases the magnitude of CVR. So that's something to keep in mind here. So to wrap up our discussion, I have a few key points. DCS-based neuron monitoring for a cerebrovascular activity assessment with using these more tolerable, clinically feasible paradigms are extremely feasible to implement experimentally. If you use our standard processes for demonstrating agreement among any of these experimental paradigms using a simple linear approach, we don't demonstrate strong agreement, but if you use a short separation regression approach, we do demonstrate a very strong agreement. And that is really critical for translating any type of medical device that assesses CVR for clinical care. So our next step in this work would actually be to demonstrate that we are sensitive to changes in CVR impairment between healthy controls and children with sickle cell disease in our case, to be able to validate this approach a little bit further. So if you're interested in this work, we have some 3-layer modeling work in DCS that I would encourage you to check out with my colleague Hong Ting, and we're also hiring a postdoctoral position. So we'd love to meet you at this conference if you're interested in our work. I'm thanking the team at the lab and at home, and I look forward to this next 2 minutes of questions. Thanks.
Thank you. And do we have any questions? Okay.
I was interested with when you did the comparison with the breath holding and the hypercapnia, the short regression separation, you done it only for the breath holding. Have you also done it for the hypercapnia one?
The short separation regression?
Yes.
Done for both hypercapnia breath hold comparisons.
So both.
And also for the resting state comparisons.
Yeah. And when you did it both, then you actually get this very.
Yes. Yeah, absolutely. And if, of course, this isn't the perfect experiment too. This is done in a lot of healthy adults. We'd like to see how this varies across a given age range as well. But it was remarkably great.
I have a question too. So that agreement is, kind of exciting. But I guess people maybe should be doing more source separation regression with DCS. But the range of reactivity values was quite wide. I mean, if he's trying to make a diagnostic, is that precision good enough?
Yeah, is the precision good enough? Well, if you were to bring this to FDA, I would say no. You definitely need a lot more work on that. those confidence intervals are a little bit wider than you would like to say. So I think, but I think per your point, I think a lot of people in our optical community who do diffuse correlation spectroscopy, these interventions are fairly like feasible to perform. And I think there's a lot of interesting space here to unpack. How does the cerebrovascular reactivity assessment relate to autoregulation indexes? How does the hemodynamic, like a hemodynamic response function captured by DCS, for instance, change in various ages and autoregulatory states? So I think there's a lot to unpack here, and I really want to emphasize that more people could be doing this work to try to make the precision on those confidence intervals a little bit stronger. Thanks for the question.
Okay, any other questions? Well, let's thank the speaker.
Thank you.
Switch. I'd like to invite Matthew Kolesnik from University of Western Ontario to talk about detecting covert consciousness using movies in the intensive care unit using fNIRS.
Right, thank you for the introduction. Hello, everybody. Today I'll be discussing how we use movies and fNIRS to detect covert consciousness in acutely brain injured patients in the intensive care unit. So what is covert consciousness? It's probably best understood through an example. So suppose, again, there's not many clinicians in the room, but suppose you're a clinician and you're presented with two different patients, both of whom have experienced a stroke and are now in a coma, and both are completely behaviorally unresponsive. Regardless of asking them to follow a command or looking at pupillary response, you can't seem to find any sort of behavioral indication that there's somebody in there. And it's typically what the diagnosis would be in these situations. When there's no evidence, behavioral evidence that a patient is conscious, well, why would we assume that there's any consciousness there? Well, that is entirely what covert consciousness is. It's just the fact that these patients cannot produce the correct behavioral response to show that they are truly conscious. And this is far from an isolated issue, where research has consistently pointed towards approximately 15% of these patients retaining some type of covert consciousness. And this is, I think, crucial because it really draws implications in terms of withdrawal of life support, recovery prospects, decisions of care, interactions between loved ones, patients, and clinicians, and also just the fact that we're now trying to detect the one thing that makes us, I guess, processing and beautiful experiencing creatures. Why wouldn't we want to explore this? Well, how can we know that these patients are actually conscious? Well, I think the seminal example is using, instead of like trying to have the patients respond behaviorally, why not have the patients respond mentally? And this was exactly what this study did. This patient who was previously diagnosed as being in a vegetative state was asked to engage in two different mental activities. One to imagine playing a game of tennis, and one to imagine walking through their home. And it doesn't take a clinician to see that the patterns of activity, here shown in fMRI, were indistinguishable between the patient and the controls, suggesting that the patient just can't create the correct behavioral response whenever we tend to ask them to blink or command follow, but they can produce the correct mental response. And by galvanizing the correct neural activity, these patients can show that they truly are conscious. So how do movies come into this? Well, movies are actually amazing experimental stimuli. They can index a series of higher order cognitive functions, such as language comprehension, plot following, feelings of suspense. They're also easy to administer and naturally attention grabbing, which might be useful for a patient who has sustained an acute brain injury. And they also, these higher order, specifically these higher order cognitive functions map onto this frontal parietal network. And again, when you look at these sort of frontal parietal networks and healthy controls as they watch the movie, you see that there's high amounts of synchronization in healthy controls when they're engaged with the higher order cognitive processes of the movie. And when you look at patient 2, again, you see that this patient also synchronizes in frontal and parietal regions, whereas patient 1 is not. Again, suggesting that patient 2 might be indeed covertly conscious, whereas patient 1 is not. So really, all this research before has been done using fMRI. And now we want to, the question is quite simple. We want to move to fNIRS and see if we can use movies and fNIRS to detect covert consciousness in acutely brain injured patients. So now we have, what I'm going to be presenting to you is the results from 2 separate patients. both of whom were equally unresponsive, the lowest possible score you can get on our ratings of responsiveness. But there's a critical distinction between these patients. So one patient sustained a cardiac arrest, whereas a second patient suffered from this rare condition known as Guillain-Barré syndrome. Now, Guillain-Barré syndrome is quite special in that it affects specifically the peripheral nervous system. So often you have patients who are cognitively intact, but unable to move any sort of aspect of the peripheral nervous system, anything from blinking. So they basically appear as equivalent to a regular patient who has sustained an acute brain injury, but really are consciously aware. So this represents an excellent opportunity to test whether or not fNIRS and movies can detect covert consciousness in this patient. So the task structure is amazingly simple. All we do is present patients with a 5.5 audio clip from the movie Taken. This has been shown to produce, again, frontoparietal activation, been related to higher order cognitive functions. And again, all we do is put the headphones into the patient's ear and they just listen to this Taken audio clip. The data acquisition, we used a NIRS Scout XP system, 129 channels, 8 short channels. Essentially, it's full brain coverage, essentially minus the occipital regions. I won't go to the analysis pipeline in great detail, but if you have questions, please let me know. The idea is that we perform, I guess, 2 stages of motion correction, so spline interpolation and wavelet correction, run a high pass filter, and we also try to remove systemic physiology using short channel regression. So let's start with the results from controls. So I didn't talk about this just yet. Essentially, we ran separate, in the same way that we're constantly comparing these patients to controls. We ran a fNIRS replication of the original fMRI studies, and the results are quite robust. We saw that these healthy controls were able to produce frontal and parietal activation. And importantly, they were able to do that not just in response to any movie clip. They made that frontal parietal response selectively to when that movie clip contains some type of higher order cognitive function. And we found that this frontal parietal activation was observed in 14 out of 15 healthy controls, giving us confidence that if we were to observe it in a patient, then that would be suggestive that the patient was indeed processing the movie in the same way a healthy control would. We wanted to be very careful, though, and create a straw man for understanding when is the patient's frontal parietal activity similar to healthy controls. So again, we created an ROI, including significant regions, specifically to the frontal parietal regions, and only when those regions were significant in the intact clip rather than this sort of scrambled control condition. So here are the results. We have both patients, and then so blue scores indicate low levels of synchronization, so low levels of, I guess, correlation between the neural time series and frontal parietal regions between the patients and the controls. So here we have the results for patient 1, and we don't see too much evidence at all that this patient is synchronizing with healthy controls. And when we look at patient 2, we see a little bit more evidence that this patient is synchronizing with healthy controls, including significant activation in the pre and post central gyrus and the middle frontal gyrus. But this is still a little bit wishy-washy. We're still seeing that there's some low levels of synchronization in some areas. Well, how can we be slightly more robust about comparing our patients and our healthy controls? So the approach that we took was to take each of the patients in neuroactivation within the ROI and frontal parietal regions. And then we wanted to compare each of those different channels. And you wanted to compare it between each patient, so the channels within the ROI for the patient between each of the different healthy controls. And then from doing this approach and computing Euclidean distance between these, I guess, frontal parietal maps, you can compute a dissimilarity score between the patient and each healthy control. And then from there, you want to run a statistical test to see whether or not that dissimilarity produced in frontal parietal regions is greater than what we would expect. So here are the results for the healthy control group. So again, x-axis would be healthy control, y-axis would be dissimilarity. So more dissimilarity is bad, means that they're not synchronizing with healthy controls. We see the distribution in this sense. And now when we plot both patient 1 and patient 2, patient 1 seems to have higher levels of dissimilarity compared to healthy controls, whereas patient 2 shows no such evidence of that. And that was verified with a statistical test. Another way of presenting this information is, again, on the x-axis, we have each of our different participants, and the y-axis, we have the dissimilarity attributed to that healthy control or patient. So where does our patient 1 fall within these results? Well, they appear up here. They're clearly different from, their frontal parietal synchronization is clearly different from that of healthy controls. And where do we find patient 2? Well, they're just right here. There's no evidence to suggest, again, that this patient frontal parietal synchronization is different from the healthy controls. So just to, again, we can rely on neural results, but this is the fact that this patient had this condition represented a rare opportunity where they tend to recover from this condition miraculously. And then when we ask them whether or not they remember participating in this task, they confirm their participation in this task. Again, lending credence to this idea that it's not just spurious correlations. This person was indeed covertly conscious, and our tests were sensitive enough to pick that up. Unfortunately for patient 1, they passed away. We can neither confirm or deny that they were conscious. And this study really reflects so much of the work that we conduct within our lab. It wasn't just that we confirmed, I guess, neurally using fNIRS and confirmed with behavioral testing after the fact. We also confirmed these results in a multimodal sense. EEG results corroborated our results where they showed, I guess, across both fNIRS and EEG, high levels of synchronization in the taken task that were comparable to healthy controls. And not only that, we also run the patient through several different tasks, including the standard task of motor imagery. And again, across both EEG and fNIRS, this patient produced reliable activity within the motor cortex. There are also some advanced data analysis techniques that we're working on to try to predict who is in fact conscious and who isn't. If you're curious about that, I'm happy to show those results. And then ultimately, we're moving towards this position where we're going to collect a large enough patient population that we can start to track not just whether patients are conscious or not, but also whether or not they will recover from their illness and to what extent their recovery will be. And that's everything. I have so many people to thank, but I want to especially thank all the nurses, clinicians, and clinical staff at the University Hospital, and of course, the patients and their families for participation. And of course, thank you all for your time.
Thank you for a very interesting talk. Do we have any questions from the audience? Okay.
You said that one of your health controls did not show any sort of significant response. Given that, how much would a negative result in a patient actually tell you if healthy people may not have such a response?
Right. So I'd compare this against like the typical results that we expect within fNIRS, where I guess like reproducibility of results even within labs can sometimes be quite difficult. So getting 14 out of 15 participants is quite significant. Again, this really highlights the importance of doing our multimodal and multitask technique, where perhaps if one task cannot pick up the results, you know, for any number of reasons, then perhaps another test will, and another test will, and another test will. And we don't seek to replace some of the existing decision-making processes that clinicians have. We just want to add this additional tool that clinicians can have to make decisions about, I guess, patient outcome, but also whether or not the patient has covert consciousness. So yeah, multimodal, multitask, don't rely on a single task and continue along that path. Yeah.
Hi, cool talk. What about other forms of altered consciousness other than comatose, like light anesthesia or sleep? Have you guys looked at it or thought about it?
That'd be really fun to do. Yeah. We're trying to run a study where we put patients in the scanner or even healthy controls in the scanner after having some type of psychedelic. But there's a lot of work that's already been done with anesthesia. But the idea is to track, I guess, seeing how the metrics in response to movie watching adjust across different, I guess, even ages or different populations or anything along those lines. And once you can get sort of a quantifiable measure of how this sort of measure changes across different populations, you can start to really quantify the extent of consciousness of these patients, right? So I think it's a fantastic idea, and hopefully we can do it.
Wes.
Yeah, very, very interesting talk. So in light of some of the other talks in this session where, like, if you have maximal vasodilation and critically injured patients and they lose their responsiveness. Do you think that could be like a false positive if you're using like this technique for, assessing, comatose? Or...
All right. So like systemic physiology, all these other types of concerns are hugely concerning to us. We do collect a series of other systemic physiological measures that we use to regress out the data, including the use of short channels. But yeah, certainly like the best scenario would be to extract out all these different types of regressors and see whether or not our results hold. You can't rule out the fact that this patient could have some sort of physiology that's inducing these results. But I think that one thing that's would be quite remarkable is that the time course of the patient synchronized also with the time course of the controls. So if it's just some sort of, I guess, something to do with like the disease itself that's compromising the cerebrovasculature, then why would we see this sort of synchronization from healthy controls to patients? Right? It doesn't seem to be the most parsimonious explanation.
Very quick question too. There's also EEG monitoring on most of these patients, and there are standardized protocols for EEG reactivity. Right. Has that been done in your patients, and how does that compare to doing this with optics?
That's a great question. We do have like a subset of patients who also have the EEG reactivity protocol. None of these patients also had that currently, but it's a study that we hope to be running quite soon to see the differences between, again, using multiple different techniques to try to predict outcome and see whether they're conscious or not. But it's a good idea.
Okay, one last quick question.
Yeah, you list on your future goals is to maybe predict outcomes. I'm thinking about like Guillain-Barré being sort of a complex immune reactive and reaction in something like, I don't know, like a basilar stroke where you get locked-in syndrome. What do you have some sort of case or ideal pathological process where you could foresee future outcomes, or what are your thoughts there on how to do that?
Yeah, like it's always, I guess, easy when the patients are surviving. Then we can sort of have certain types of measures that directly measure the extent of their recovery, so stuff like GOS score or CRS. We also have within our lab a series of cognitive tests that we run, I guess, 1 year afterwards. There's a series of cognitive tasks that measure things like spatial reasoning and the rest of spatial reasoning and language abilities and stuff like that. So the idea is to get a large enough cohort that we can start to make decisions about these things. But certainly these current results cannot, I guess, predict exactly that.
So would you be looking at like fNIRS like across multiple neurocognitive batteries?
Right.
Okay.
Right.
Got it. Thank you.
Let's thank the speaker. Very interesting talk. I'd like to invite the last speaker of the session, Farah Kamar from Western University, talk about assessing the sensitivity of a high-density time result near-infrared spectroscopy device for neuromonitoring.
All right, thank you for that introduction. I'd like to start by acknowledging my supervisors, my lab members, collaborators, sources of funding, and of course, the kernel team for loaning us the device this past year. Perioperative stroke is when the blood supply to part of the brain is interrupted, either during surgery or within 30 days of surgery. And this decrease in blood flow leads to downstream decreases in oxygen and nutrients that can have lasting physical, cognitive, and mental damages for patients. Neuromonitoring during surgery can actually detect these strokes early to help us guide subsequent interventions. As you all know, SfNIRS is a great tool for monitoring oxygenation. So with continuous wave or traditional SfNIRS, what we do is we shine light in the near infrared range from a source on the head, and it travels through the tissue, and we can detect the photons that return to the detector and estimate oxy and deoxy hemoglobin concentrations. One of the challenges of SfNIRS, as you're all aware, is the depth sensitivity. And so one potential solution is using time-resolved SfNIRS. With this type of SfNIRS, instead of a continuous light source, what we do is we shine a short light impulse into the tissue, And then we can detect the photons that return to the detector across time. And so we have this time of flight distribution. And what we could do to improve our depth sensitivity is focus our analysis on these late arriving photons because these travel deeper into the tissue. And so returning to our clinical problem, cerebral oximeters are a common way for monitoring oxygenation at the bedside during surgery. However, as was discussed previously in this session, the limitation of these cerebral oximeters is that they really only monitor a single brain region, and that's the forehead. So if we do have oxygenation decreases in other cerebral arteries, these can go undetected. And so the big question becomes, is there a way to monitor oxygenation of all cerebral vascular territories with a high depth sensitivity? And we're proposing the use of this new NIRS device called the kernel flow. So to start off, it addresses our clinical problem because it's capable of full head monitoring. Additionally, it is a time result system, so we can obtain these measurements with a high depth sensitivity. It's a pretty impressive piece of technology, so I'm going to dive for a few minutes into the design of it. It's a high density system with 52 sources and 306 detectors across the head. If we zoom into one of these hexagonal modules you see, in each of them, there's one source in the middle and then 6 detectors surrounding it. So if we emit light at any of these sources, we can detect it at any of these 6 detectors or any of the detectors in surrounding modules as well. So between all of these different sources and detectors, there's over 2,000 possible channels for oxy and deoxyhemoglobin measurements. The optodes in the system are also spring-loaded to make the best possible scalp contact. And it has a very high sampling rate. And finally, they have this automated cloud-based processing pipeline for easier and faster data processing. And so because it's such a new device, there is a need for more research to assess the full capabilities of the system. And so my study objective was to assess the sensitivity of the kernel flow to sudden changes in regional cerebral oxygenation. And so to achieve this objective, we first recruited 10 healthy young volunteers to participate in 2 different paradigms that would robustly induce oxygenation changes. And both of these tasks have been previously demonstrated both in the literature as well as in our own labs. And so the first paradigm was a compression of the carotid artery. And so if you compress one of the common carotid arteries, what we observe is a transient and large blood flow and oxygenation decrease in the entire ipsilateral hemisphere. And so we know the expected change, and so we can look for that. And then the second paradigm, one you're probably all familiar with, is a finger tapping exercise. And the reason we picked this is it would allow us to test for more localized and smaller scale oxygenation changes. And so here is an image of one of our participants, and that's the device on the head. And we also used this neck brace you see here during testing. And that was to both minimize motion artifacts, but also offset the weight of the device to maximize participant comfort. And so once we had collected our data, the kernel pipeline removes low quality channels that are missing expected physiological signals like the heart rate. And then once the data is returned, we're able to calculate oxy and deoxyhemoglobin concentrations in our lab. And so as I mentioned, again, it's a time result system. We obtain this time of flight distribution. And so we can use different characteristics that are more sensitive to this right skew or those late arriving deep brain photons to carry out the calculation of oxy and deoxyhemoglobin. And so one of those characteristics is called the first moment. It's the average time the photons took to return to the detector. It has a higher sensitivity to the brain than continuous wave or traditional NIRS. However, it also has a higher amount of noise. Another characteristic we can use is the second moment. The second moment, in general, the higher the moment, the more sensitive it is to the brain. But the trade-off is that it also has more noise. We've successfully used the second moment in our lab previously, but we were wondering if we're able to use it using this system. And so that was kind of the goal. Moving on to some of my results, we divided the data into the 4 different lobes or brain regions. And we found that only 6 of our 10 participants had at least one good quality channel in every region. And we looked at the distribution of these channels across the different regions. So active channel percentage is just the number of good quality channels divided by the total number of expected channels. And what we observed was that the frontal region had significantly more active channels than any other region. The frontal region with this device is measured through the forehead, and so it has no hair. And then another observation we made was that if you look at the different types of dots on this plot, each different dot is a different participant, and you could see that the circle and rhombus participants consistently had the highest amount of good quality channels across the head, and they coincidentally had the least amount of hair. Now moving on to some of my paradigm results, let's start with the compression results. And so what you see here overlaid on a head model, the dots on the screen are a visualization of the change in oxyhemoglobin between the period of baseline and the period of compression, according to this color bar here. So red is indicative of an oxyhemoglobin increase, whereas blue is indicative of a decrease in oxyhemoglobin during the period of compression. The right side is ipsilateral to the compression, while the left here is contralateral. And you could see here that we do observe the expected decrease in that ipsilateral hemisphere when we look at the first moment. Looking at the second moment for this participant, you can see that it's more noisy. So we're observing some unexpected increases in oxyhemoglobin. And so as a result, we decided to move forwards for our functional data analysis with the first moment. Before I move on, I do want to highlight a second participant here. This was one of our more extreme cases of signal loss as a result of hair. And because we only really had signal in the frontal region for all of our participants, that's where we decided to focus our group analysis for this paradigm. And so here is a time series plot of the group average. With the top here, you see the ipsilateral hemisphere, and the bottom is the contralateral. And this is the first moment. And so you could see that the ipsilateral shows the expected decrease in oxyhemoglobin, as well as an increase in deoxyhemoglobin. And you don't really see any significant change during the period of compression in the contralateral hemisphere. So now on to the activation paradigm, which was the right hand finger tapping task. And so before we analyzed the data, we looked at whether we even had sufficient signal for analysis in our region of interest, which is the somatosensory motor cortex. And we found that only half of our participants had at least one good quality channel in the region for analysis. And what you see here are the data sets from the 5 participants. And so what you're looking at overlaid on a head model is the red lines indicate a significant channel according to our general linear model. And you could see that only 3 of the 5 participants showed activation in the left primary motor cortex. And so kind of to summarize everything I've talked about, in terms of our first paradigm, we observed the expected changes in oxygenation during the carotid compression most clearly in the first moment. In terms of the second paradigm, in only 3 of the 5 participants that had sufficient signal for analysis, we observed the expected activation. In general, we observed some regional changes in terms of the sensitivity of the device that was highly dependent on the presence of hair. And in terms of next steps, we're hoping to recruit more participants that are representative of our target patient population, because those undergoing cardiac surgery are predominantly older with a decreased hair density. So hopefully that would help mitigate that hair concern that we had. And then finally, we're hoping to investigate the advantages of this moment analysis using a prefrontal cortex task to also mitigate those hair concerns. Thank you so much, and I'd be happy to take any questions.
Thank you. Any questions from the audience?
Thank you. I have one question because I see that you are using first movement and second movements to analyze. First movement is an average of time of flight. Second movement is a variance or FWHM, full width half maximum. But all these two values is basically the average of the early photons and late photons. So it's a mean of the weighted superficial information and deep layers information. But in the early part of your slides, you mentioned that what is really interesting to us is that the late photon, which is deeper region. So my question, have you analyzed any, for example, time window? So you can analyze the late photons, which corresponds to the deep layers information.
Yeah, that's a great question. So you're asking why we're not using like gate sort of analysis with our time resolved data. And so the main reason for that is the way that the data is sent back to us. We only have access to the moments. We don't see the raw DTOFs for our analysis. And so that's kind of why we focused our analysis using the different moments and using sensitivity factors to calculate oxy and deoxyhemoglobin.
So you mean the kernel's device, they do not provide you the raw data, but they analyze on the cloud and
yeah, so with the processing pipeline that they go through, we don't see the actual raw data. So that's why we focused on the moment analysis.
Okay, thank you.
Hi, so I'm Catherine, I'm from Kernel. Thank you for taking a look at our system, our prototype. I think everyone in the room knows that hair is an issue for NIRS signals. And since I kind of moved from academia to system building, I learned that there's a lot about heads. It's not just hair, actually. And one of the main things that we found out with this prototype is that it's very shape dependent too. The shape of the head is really more variable than you might expect. So our next gen system has a new better way to kind of adapt to different head sizes. We learned a lot about setting up our source detector pairs and channel coverage, because that's also so important in order to get those good channels that you need. But I just wanted to point out that there are like many things besides just like saying, oh, there's hair, so we can't see it. Like there's a lot that actually happens on a head that is needs to be accounted for in order to get good signals.
Yeah, thank you for bringing that up. That's definitely something to think about for sure. And it's exciting to see that there's a new prototype coming out soon.
Okay, a quick question. So did you happen to measure any of these subjects with some other research near system at the same time to compare results?
That's a really good question. We have not done that yet, but it would be a really interesting thing to directly compare systems.
Okay, one last question.
You wanted to show us a whole head coverage of oxygenation. So in fact, what you look at is not an oxygenation change, but a change in the concentration of oxy and deoxyhemoglobin. So you need a baseline. So without the baseline, you're done with that method, with the changes in moments. My question is, though, when you do a whole head coverage, your results must depend on the thickness of the skull, which changes with across the head. This is in line with what we have heard before. Can you comment on that? What do you expect due to this fact that you have no constant skull thickness?
Yeah, so I think that's a problem with NIRS in general, is the variability of the skull in different locations of the head. But yeah, we used like different source detector distances as well. I didn't really talk about the processing pipeline that we used, but yeah, with hopefully the time resolved and having the short channels in the same places, like it's just a general problem of NIRS. I don't think it's dependent on this system. Thank you.
This concludes our session. It's like the audiovisual team is making sure we don't say anything. So we have a coffee break now, but we're going to make it a little bit shorter. So let's try to come back at 4:25 as much as possible.
So we are starting the session. Please sit down. All right. It's our pleasure to have Mamadou Diop from Western University here for the first invited talk. And welcome, and let's get started.
Thank you, Turgut. So good afternoon, everyone. Turgut, thank you for this introduction. I would like to also thank the organizers for giving me this opportunity to talk to you about the work we have been doing over the last few years on developing technique that will allow us to monitor the brain in neonates. And to be more precise, so we want to develop, we are developing quantitative near-infrared spectroscopy techniques that will help. So we are developing quantitative near-infrared spectroscopy techniques. So I will start with an introduction where I will present the motivation for us to monitor the brain in neonates, because this section is all about neonatal monitoring. So I'm going to be talking about so the motivation. I will provide a rationale for why do we use these techniques. And so the technique we are using is a combination of dynamic contrast enhanced near-infrared spectroscopy and diffuse correlation spectroscopy to develop a technique for continuous monitoring of absolute cerebral blood flow and in the brain. I will also talk about a hybrid optical technique that combines hyperspectral near-infrared spectroscopy and diffuse correlation spectroscopy. This will allow us to monitor changes in cerebral perfusion and metabolism in neonates. As most of you knows here, the neonatal period is a moment of high risk of brain injury. And this is because neonates, particularly preterm neonates, are prone to hemodynamic instability. They often have compromised cerebral autoregulation, and they start with a low basal cerebral blood flow. And because of all these factors, they often have hypoxic and ischemic events. And because the brain, as you have shown in the previous section, has a high metabolic demand and very low substrate. So these prolonged period of hypoxia or ischemia can very rapidly lead to brain injury. Although preterm or neonates of all ages can experience prenatal brain injury, this is more prevalent in neonates with incidence of about 2.6 in preterm infant. Perinatal brain injury is a leading cause of childhood death and disability across the globe. It leads to long-term cognitive and neurologic, sensory, and motor impairments. And therefore, I hope that I have convinced you that it is very important to have a point-of-care technology that will allow us to monitor the brain of neonate in order to detect cerebral ischemia or hypoxia. And eventually, this will eventually be used to alert the clinical team so that they can apply corrective measures. So what is the rationale for us to use optical technique? So this is the work that was done by the group of Keith a few years ago. And in this example, they were doing a continuous measure of, they were doing point measurements of cerebral blood flow and metabolism. And as you can see here, and the metabolism here is represented by cerebral metabolic rate of oxygen, CMR2. When cerebral blood flow decreases, it doesn't immediately cause a drop in metabolism. And it has to reach a certain value. In this case, it was about 35% decrease in cerebral blood flow for cerebral metabolism to start to change linearly with blood flow. So what we can learn from here is that if we are now able to do these measurements concomitantly, this may allow us to gain new insight into perinatal brain injury and ultimately have early biomarkers of brain injury. So now I'm going to go through explaining the different techniques, introducing the different techniques that we're going to use. So the first one is a technique that allows us to quantify cerebral blood flow. And we do this using dynamic contrast enhanced technique. So dynamic contrast enhanced technique is not something that is specific to near-infrared spectroscopy because it's used in MRI using gadolinium DTPA or in CT using iodine contrast agent. But the theory behind is the same. So the example we have here, this is an expression of the Fick principle. And what we're showing here is the relationship between the concentration of the tracer in the tissue is equal to the concentration of the tracer in the arteries. And how do we measure these concentrations? So typically you do a bolus injection of the tracer in the peripheral vein, and the contrast agent is carried through the blood, go to the heart, and is pumped everywhere in the body. And you measure the concentration of the tracer in the tissue, and you can measure the concentration of the tracer in the arteries. Now, how do we measure this? With SfNIRS. So in the case of near-infrared spectroscopy, we use indocyanine green, ICG for short. So this is a concentration of ICG. So we allow a certain period of time to get a baseline before we do the injection of the contrast agent as a bolus injection in one of the artery, in one of the peripheral vein. And we have this characteristic time course of the concentration of the tracer in the tissue. And for the arteries, we use a modified pulse oximeter. It's called dye densitometer, which allows you to measure the concentration of the tracer in the arteries. Now, you have concentration in the tissue, concentration in the artery. What is left is this quantity here, which is the arterial impulse residue function scaled by the blood flow. And in order to evaluate this quantity, where this will allow you to get the blood flow, you have several options. You can use analytical models that allows you to model the impulse residue function R of t, or in our case, you can use model independent approach where you use deconvolution. And in this illustration here, what I'm showing here is this curve is the impulse residue function scaled by the blood flow. And because the maximum of R of t is 1, then the maximum of CBF multiplied by R of t is here, gives you the value of the cerebral blood flow. In this example, the cerebral blood flow was in 50 units of blood flow, which is in millimeter per 100 gram of tissue per minute. So this is how you quantify cerebral blood flow or any blood flow in any tissue bed using near-infrared spectroscopy or NIRS. And it's similar to technique used by MRI, as I said, or CT. And this has been validated previously, and this is work that was done actually at our center. And this is cerebral blood flow measured by NIRS on the x-axis, and on the y-axis you have cerebral blood flow measured by CT. Not only there is a strong correlation between the two quantities, but it's a one-to-one correlation. We very recently also did a validation of this technique. On the y-axis here, we have the dynamic contrast enhanced NIRS using ICG measure with our time resolve technique. And on the x-axis, you have ASL MRI. And this work was done in collaboration with a group of Arjuniot at UPenn. And here again, and this was done in adult, much more difficult to do than in neonates. And here again, we have a one-to-one correlation between the two techniques. Just to say that the technique is quite robust. Now, this is great, but the problem with a dynamic contrast enhanced technique is that anytime you have to measure blood flow, you have to do a bolus injection. And you'll agree with me that that's not very practical. And that brings us to a technique that you guys are quite familiar, which is diffuse correlation spectroscopy. Most of you will don't need an introduction to DCS. It's an optical technique. They use a long coherence light source. And when you send this light, if you look at the surface of the tissue, you will have a speckle pattern. If you have no moving particles, so this speckle is fixed. But if you have moving scatter inside your medium, the speckle will fluctuate. You have different approaches, and the approach we use is that we put here a few mode detector or a single mode detector, and now the size of the detector, which is going to be a single mode fiber or few mode fibers, the core of these fibers is bigger than a single speckle. However, you have now a projection of the speckle into the space of the single mode or few mode. Then you can detect intensity fluctuation. If the light scatters are moving, you can detect a fluctuation in intensity that looks like this. Now, if you do an autocorrelation, if you correlate this signal with itself, you'll get curves that look like what we have in this illustration. So what we have here, the blue curve here, is the autocorrelation we obtain at baseline, which is normocapnia. When we increase cerebral blood flow by inducing moderate hypercapnia, we have a shift of the curve to the left. And we further increase hypercapnia to further increase blood flow by going to higher hypercapnia. We have a further shift of the curve to the left. Now, when we do carotid occlusion, which is a reduction of cerebral blood flow, we have a shift of the curve to the right. And what this is showing is that these autocorrelation curves are sensitive to blood flow. Well, we don't stop there. We didn't stop there. So there are models of light propagation, autocorrelation propagation in tissue, and we take some of this analytical model. You can fit your autocorrelation curve and extract the parameters. And this parameter here, alpha here, represents the fraction of moving light scatters, and dB is a diffusion coefficient. Now, if you look at the changes in the diffusion coefficient, we compute this and we compare it to what we have with the dynamic contrast enhanced technique. What I'm showing here on the x-axis is the changes in the alpha dB, which is the DCS parameter. And on the y-axis, what we have here is the changes in flow measured by the dynamic contrast enhanced technique. And what we see here is we have a one-to-one correlation. It's telling us that the DCS, changes in the DCS parameter, is exactly equal to changes in blood flow. So this was done in a piglet model of neonate, and we have also done this in the clinic. This is a group of 8 neonate that was in the intensive care unit, and we did measurement with the fNIRS, and we on the x-axis, and on the y-axis we have the DCS, and as you can see, still very good, very strong correlation as well. Now, how do we now do a continuous measurement of cerebral blood flow? As most of you know, so with DCS, you can do continuous monitoring. This is a case where this neonate were receiving indomethacin infusion as part of standard of care, and we acquire our measurement. The blue dots here is before indomethacin infusion, and you can see here we have about 75% drop in cerebral blood flow. But what does it mean? And how? So if you do any DCE measurement here, we do your tracer kinetic measurement in any point in here, you can convert this curve into absolute cerebral blood flow. And this is very important because this allows us to see that, well, these values that we had at the baseline correspond to 40 units of cerebral blood flow. And it drops now to 8 units of cerebral blood flow at the end of the process. So this is very important because there are ischemic thresholds. So this will allow you to know if the tissue was ischemic or not. And this highlights really the importance of quantitative NIRS technique. And one thing that was most surprising to us was that when we look at the alpha dB, not the changes, just absolute alpha dB and absolute cerebral blood flow, there was still a very strong correlation. And that was quite surprising to us. And what this is saying is that actually the alpha dB is, in fact, an index of blood flow. And this has been confirmed theoretically by Stefan Karp and David Bose not a long time ago. So the blood flow index is really a measure of flow. It doesn't have units of milliliter per 100 gram per minute, but but there is a caveat here. You need to very accurately measure the concentration, the optical properties of your tissue. The absorption and scattering coefficient has to be measured accurately. In this case, we were using a time-resolved system in combination with the DCS in order to measure the optical properties. I'm going to move now to hyperspectral NIRS. So you guys are familiar with this curve. I'm not going to spend a lot of time. You have water, absorption coefficient of water, oxy and deoxy. If you take the second derivative of this curve with respect to wavelength, this is what you obtain. And why I'm showing this is if you look at this region here between 800 and 820 nanometers, the only chromophore that really significantly contribute to the signal you're going to be getting is water. So we didn't invent this. We adopted from the University College London approach. And if you look at now in this region between 700 and 800, what you have is the main contributor there are water and deoxyhemoglobin. So what we did is, because the UCL group was doing this using this approach with the Beer-Lambert law, but in our case, we fit a theoretical model to the data. And if we do that, in this region, we can get concentration of water. And once we get the concentration of water, we use it as a known parameter in the fitting of this second region here. And if we fit in the second region, we can have a good estimate of the deoxyhemoglobin. And now, once we get these two parameters, we use them. We use them to fit the first derivative of the spectrum. This is the derivative, spectral derivative of the absorption of the attenuation spectrum. And once we get this, we use the first derivative. We can estimate. Can have estimate of oxyhemoglobin. Now, so the concentration of the light absorbers here, water, oxy, and deoxy, can be estimated using this technique. And the beauty of this is that it's very simple. You have a halogen lamp and a spectrometer. Very simple. Very, very simple. And then you just need to do this approach. So it's a very powerful technique in that regard. And we apply this in piglets undergoing hypoxia ischemia. So first of all, we did a carotid occlusion in the first part, and then we reduced the fraction of inspired oxygen. And as you can see, as expected, there is a decrease in flow, decrease in oxygen saturation. And the green curve here is changes in the cytochrome C, which is also a very interesting signal that we can also measure with this technique. So after that, we built a clinical device. We call it NEMO. It was neonatal neuronal monitoring. It was work led by AJ Rajaram, who is now in Boston. You know, the system is soundproof and all those good things that you need to be in a sensitive environment such as neonatal intensive care unit. And we conduct a study where we look at what happened, what is the correlation between cerebral blood flow, oxygen saturation, and metabolism. And metabolism here was cytochrome C. And the take-home message here is that the cytochrome C was extremely stable, and changes in the other parameter did not really affect the cytochrome C. We also apply NEMO for neuromonitoring during ventricular tap. Here you have our colleague, the neurosurgeon, inserting the needle into the left ventricle, and here they are draining the CSF from the head. These are infants that are suffering from hydrocephalus, and we did the monitoring during the whole procedure, starting before 2 minutes. So we're showing here 2 minutes here, but it's a little bit longer than that. Let's just crop the data. And so the area here is before the ventricular tap, and now we're monitoring blood flow index during the ventricular tap and after the ventricular tap. And when we look at at the end, we compare the average parameters before and after the ventricular tap. And what we saw is that although we didn't see any significant changes in both the cerebral blood flow and metabolism, and this was likely due to the small sample size. And when we do our power calculation, it showed that if we had a patient population of 30 patients, we would be able to detect significant change. And this cross here, the red cross here, and these two red cross here, were outliers, and if we excluded these outliers, even with our smaller sample size, the data will be significant. This is showing that this ventricular tap was relieving the pressure from the brain, was improving cerebral blood flow and metabolism. Now, what's next? So use NEMO to conduct now larger clinical study, as I just alluded to in the previous slide. Our sample size for this pilot study was small, so we need to go to larger sample size. We're looking at infant with hydrocephalus, infant undergoing cardiac surgery, and also trying to extend this technology to older infants, because the pediatric population is actually much larger than the pediatric population is much larger than neonates. So this is a simulation that we did a couple of, like last year at OSA Biomed. It was done by one of my undergrads, now a graduate student, Natalie. And this is showing the changes this was simulation that we did. So this is changes in cytochrome C. The black curve here is the expected value when you use CW, continuous wave NIRS, hyperspectral continuous wave. And this is when you use hyperspectral time resolved NIRS. And clearly, when we quantify the difference with hyperspectral time resolved NIRS, the difference between the expected and the recovery value was less than 5. You have less than 5% error, but with the CW, it was around 10%. So for hyperspectral NIRS, I'm not going to spend a lot of time. If you guys have a question about this, just let me know. So we have recently developed a hyperspectral time resolved NIRS based on compressive sensing. And this technique, this hyperspectral, if you now collapse the data on this axis, on the temporal axis, what you have here is something typical to what you will get with CW, hyperspectral CW. And if you now look at a few wavelengths, so you have, in this case, we're showing about 6 wavelengths, so six time resolved curves. And this is across this, our initial system, we go from 700 to 830. Yeah, we have since extended the spectral range. Now we cover 200 nanometers without sacrificing resolution. And here I'm just showing every second wavelength. We conducted some tissue mimicking phantom where we did oxygenation, similar to the work done by Elias group. So this is a homogeneous phantom. The solid curves are time resolved. The dotted curves are CW, hyperspectral CW NIRS. And what we can see is that we recover similar trend, although the CW is now currently overestimated, and we are investigating this. We did also a 2-layer phantom, and this is really, really exciting because it's showing us that, you know, and for the top layer here of the phantom was 10 centimeters, and still we were able to recover the changes. And so these are very, very new results. You guys are the first one seeing this, and really interesting and very exciting for us. So I'll finish by thanking my long-time collaborator. So I will acknowledge here this significant contribution of Keith, Daniela, and AJ, as well as collaborators such as Elias, Gemma, and Vlad. And well, this work was, I didn't do the work in the lab, obviously, so this was done by my incredible graduate student. And it's a picture of us, our Halloween costume of last year, and we actually won our faculty Halloween costume. So if you guys want to join these Incredibles, we are hiring, and thank you very much.
Thank you, speaker. Do we have any questions? We have a few minutes. Please.
Thanks, Mamadou. A very interesting talk and exciting work. For your ventricular tap experiment for the hydrocephalus with the preterm infants, I know you saw like a heterogeneous response, which could be just because like the ICP was very heterogeneous across your population, where some infants had much higher ICPs than others. I'm curious, like, so pre-ventricular tap, if you're able to get a baseline, did you see any difference in absolute measures, like blood flow, like in the preterms? Because I would think that maybe the ones that have low absolute blood flow would have a higher response after the tap.
That's a great question. I don't remember the granularity of that, but one thing that I remember is that the oxygen extraction fraction significantly increased after. So you have a high oxygen extraction fraction before the tap, but it decreased, oxygen extraction fraction decreased after the tap. So I don't remember exactly about the baseline cerebral blood flow index between these subjects, but you may be right, and that could be expected.
It could be just interesting to look at if you do a correlation between baseline absolute values and then the response, the change to the ventricular tap.
I agree. I perfectly agree. That would be actually, that would be interesting to check. You're giving me an idea for a 4th-year undergraduate project to go back and revisit the data.
Please go.
So quite interested in the calibration with ICG actually, and I was looking at the papers and noticed that the diet densitometer is not really something that's available anymore. Do you have any practical advice on how to implement this these days?
Well, that's a great question. And actually, so the group of Arjun, I don't know if he's here. They have developed, I think they have developed a dye densitometer, and I believe it's working very well. So you can approach them, and I think they could be able to help you with that.
Okay, we can take more. Any more questions? I have one. So I'm a DCS guy, and I saw your absolute blood flow. So if I use your system, calibrate my system once, then will my DCS, I see a good correlation between DCS and NIRS that you have. Will my DCS be drifting? Like once it's calibrated, can I use it as a absolute measure? Because it was a very good correlation between the two.
Yeah, so I think if you, for a given population, like a population that is homogeneous, like preterm babies, if you have your blood flow index and you accurately measure your optical properties, I think I strongly believe that you can use the blood flow index itself to compare over days and compare between population, because that's what our results are showing. It's showing that the blood flow index is really like, we've compared the blood flow index and it's not, it doesn't have like a relate. So the relationship is not like 1, but 0.5 or 0. Something, but it looks like the blood flow index is a measure of flow. So I think that.
All right, so time is unfortunately up. So thank you, speaker. Please, applause. Our next speaker, please welcome Sabrina Brigadoy.
Good afternoon, everyone. So during this talk, I would like to walk you through the Baby Glucolite project, which is an ongoing clinical trial which we started in March 2020 in Padova. So optimal glycemic control in preterm babies, those born before 32 weeks of gestation, is still a major challenge. These babies are more likely to suffer from hypo or hyperglycemic changes during the first weeks postpartum compared to late preterm or term babies. And the main reason is that they have an impairment of glucose homeostasis. So they have limited glycogen and lipid stores and also a limited ability to mobilize alternative fuels. And the main reason is that the glucose control system develops during the last trimester of pregnancy. So the consequence is an increased mortality and also pure neurological and neurodevelopmental outcome. And some previous MRI studies show that occipital and parietal areas seems the one more subject to injuries due to hypoglycemia. The symptoms is that most of the time these events are asymptomatic, meaning that the clinical staff doesn't know whether the baby is suffering or not from these hypo and hypoglycemic changes. So the standard therapy consists in a point-of-care blood glucose measurements, usually through heel prick samplings, and these are done usually twice per day. And what happens is that the clinical staff will then decide based on the glucose value that has been measured and what are called operational thresholds, the adjustment in nutrients and glucose intakes for the baby. So these are the operational thresholds. So if the baby is between 144 and 72 milligrams over deciliter, then it's in euglycemia, it's fine. If it's above, this threshold is in a hyperglycemia. If it's below, is in a hypoglycemia. And this could be mild or severe depending on the threshold. So with this standard therapy, many events might go unnoticed because, of course, if we are measuring just twice a day the glucose values, we don't know what's happening in the remaining 23 hours of the day. And this might invalidate longitudinal studies trying to correlate neurodevelopmental outcome later on in life with the glycemic events and variability at birth. So there is a promising therapy which is called continuous glucose monitoring, or CGM, which consists in a small device that is placed in the infant's diet, which can continuously monitor glucose values in the interstitial fluid. And in this way, clinical staff will know in real time the glucose values of the baby, and therefore can adjust nutrients and glucose intakes to try to avoid the baby to go into hypo and hyperglycemia. And some clinical studies in the past year have shown that this has reduced the glucose variability at birth in these babies. So although CGM allows a tighter glycemic control, clinicians are still guided in their choice of nutrients and glucose to give to the baby based on the operational thresholds. And these thresholds do not consider the individual variability and susceptibility to glucose changes, and they do not consider the individual effect of glucose levels on the individual brain hemodynamics. So all of these studies are trying to correlate what is happening in terms of glycemic changes at birth with neurodevelopmental outcome. But the brain at birth is still out of the picture. So they haven't looked at what is actually happening in the brain at birth while these glucose changes are happening. But we know that from previous studies that there is a relationship between the blood glucose level and the cerebral blood flow at birth in preterm infants. And some studies have postulated the presence of what is called a cerebral glucose sensor. So what the Baby Glucolite project seeks to do is to bring back the brain into the picture since birth. And these are the aims of the project. So first, to characterize the individual hemodynamic responses to the hypo and hyperglycemic changes in preterm babies at birth, to evaluate whether a tighter glycemic control can influence brain hemodynamics, to evaluate whether the number, the severity, and the duration of the glycemic changes at birth may have an impact on brain connectivity and the hemodynamic responses at birth to these events, to evaluate whether neurodevelopmental outcome at 1 and 2 years of corrected age is related to the glycemic variability at birth, but above all to the specially specific hemodynamic changes at birth, meaning if how the baby develops is related to how the brain coped to these glycemic changes at birth. And there's a final aim to put forth an initial model whereby both glucose and brain hemodynamics are considered in the clinical decision. So instead of operational threshold, we can get functional threshold, individual functional thresholds. How we do that? By combining continuous glucose monitoring with continuous brain hemodynamics monitoring through DOT, diffuse optical tomography. And we perform that during the first weeks postpartum, which are the more sensitive to these changes. So this is the project outline. We are enrolling preterm babies born in the NICU of the University Hospital of Padova, and the inclusion criteria are they have to be less than 32 weeks gestation or with a birth weight less than 1.5 kilos. We randomly assign the baby to one of two groups. One is the experimental group, one is the control group. So in both groups, we perform DOT acquisition for approximately 1 week postpartum. And in both groups, we perform also CGM acquisition. So we have a continuous monitoring of the glucose levels. So the difference between the two groups is that in the experimental group, the CGM device is kept open. So there are alarms that will warn the clinical staff if the baby is going towards an hypo or hyperglycemic threshold, so that the clinical staff can intervene in real time to try to avoid the baby to go into hypo or hyperglycemia. While in the control group, the device is kept blinded. So it's the alarm is off, meaning that the clinical decision of the clinical staff will be based on just the two measurements that are usually done for the standard therapy in the clinical unit. At the end of the first week, we have the end of treatment, meaning that we remove the DOT and the CGM device, and then we follow up the baby at 12 months of corrected age and at 24 months of corrected age for evaluate the neurodevelopmental outcome. So the current situation of the project is that we have recruited 47 babies, and we aim to reach at least 60, with a median gestational age of 30 weeks and a median weight of a bit less than 1.5 kilos. We have analyzed 26 CGM traces, 1 DOT data, and 20 patients performed the 12 months follow-up, and 9 performed the 24 months follow-up, and it happened just a few months ago, the first 24 months patient. So just very preliminary results on some of this data. So concerning the CGM, this is an example of a glucose trace that we have, for example, for one of the baby for the entire acquisition time. What you can see are the dashed line are the different thresholds for hypo and hyperglycemia, and we can see that in most of the time the baby is in euglycemia. But then it had some events of mild hypoglycemia, and toward the end of the treatment was almost all the time in severe hypoglycemia. What we want to do with these traces is, of course, compare what is happening in the experimental in the control group, and to compare the two groups mainly in terms of number of events, duration of these events, the severity of these events, and also glucose variability and time spent in euglycemia to evaluate whether the sensor is actually having a good impact on glucose variability. For the DOT data, we use an NTS system with 8 sources and 8 detectors for a total of 64 channels that were placed according to the layout you can see on the right with a sensitivity matrix. So we are covering most of the head. This is a simple pre-processing pipeline that we are using with aim to perform image reconstruction with a specific head template. This is an example of DOT data in one of the patients. We can see that there are a lot of motion artifacts because the acquisition is continuous. So also when the parents are cuddling the baby or where the clinical staff is doing any clinical intervention, the acquisition is still going. And if we zoom in a bit, we can see the good quality of the data that we can acquire between the motion artifacts. So what we want to do with the DOT data is to characterize the responses to hypo and hyperglycemia, and also to compare whether these responses are different in the experimental in the control group. And we would like also to evaluate whether there is a difference in the connectivity pattern, comparing the connectivity pattern at the first day of acquisition with the one at the last day of acquisition, and evaluate whether number, duration, and severity of the glycemic events in between had an impact on the change in connectivity. These are some preliminary results for the DOT data. So these are image reconstructed for 4 mild hypoglycemic events at the time of peak of hypoglycemia. So what I'm showing is the change in total hemoglobin concentration. So we can see that there is a heterogeneity in the spatial pattern. And what we can see here in the last 2 columns are actually severe hypoglycemic events. So what we can see is that during the severe event compared to the mild event, we have a higher increase or decrease of total hemoglobin concentration. For this patient, we also perform a correlation analysis between the change in total hemoglobin concentration at the time of peak of hypoglycemia and the peak of hypoglycemia. And we did that for the 6 different regions of interest that you can see in the picture on the left. And what we found is that for the frontal and the more occipital areas bilaterally, we could find a negative correlation, meaning that when the baby is in severe hypoglycemia, there is an increase in total hemoglobin concentration. That might be a compensatory mechanisms of the brain bringing more oxygen and glucose to the neurons that are needing it. While in the parietal area, we can see the opposite correlation, meaning that this might be the area that are more suffering and might be prone to injury in this baby. In terms of neurodevelopmental outcome at 12 months of corrected age, what we do is to administer the Bayley Scales of Infant and Toddler Development, 3rd edition, which is a clinical scale that provides clinical score for the 4 different domains, which are the cognitive domain, the language domain, the motor domain, and the social-emotional domain. And we also perform 2 computerized tasks. One is called simultaneous stream change detection task. It's a task to evaluate visual memory capacity. And another one is called the gap overlap paradigm, which allows us to evaluate the orienting of attention, so the efficiency of the attention system. And what we measure are behavioral measures. So we just look at saccades, meaning how long the baby was looking at the stimuli we're presenting or in which direction it is looking. For the 24 months follow-up, we perform exactly the same follow-up we perform at the 12 months, but we add also at the behavioral measure also a high-density EEG acquisition to evaluate with a better picture how the brain is processing the memory and attention stimuli we are presenting. Very, very quick look at the behavioral results at the 12 months follow-up. These are some promising results. What we saw is that for the experimental group, there seems to be a higher memory capacity. So they are able to memorize 2 items in their memory compared to the control group, which seems to be not be able to memorize 2 items at the 12 months follow-up. So these are promising results. So I want to thank all the people that have participated in this project and also all the funding agency that allow the project to start and to continue. And above all, University of Padova, which is celebrating this year its 800th anniversary from its establishment. Thank you.
Yeah, time for question.
Yes, hi Sabrina, great work collecting all this data from this very sensitive population. Together with your glucose measurements, have you looked at hematocrit? And the second thing I want to ask you as well is that on the premise, we know that they need to do quite a lot of blood pressure control. Have you looked also at the blood pressure control of those, the blood pressure of those babies at all?
Okay, so we haven't looked at them yet. So we have the hematocrit. We haven't looked at it yet. While for the so blood pressure, yeah, it's also a confound. We should keep that into consideration, but unfortunately, we don't have these measurements available. So there was no way.
At least you will know if they use, if they use drugs to control the blood pressure.
Yeah,
you might have
sure.
those logs.
Yeah,
might be helpful to know that.
Yeah, thank you.
Any other questions? I also wanted to ask along the same line as Ilias about the confounders. Any other drugs given to these babies, their birth weight, other things, how do they affect the sensitivity to the glycemic effect?
Yeah, so actually, I agree with all of you. I mean, there are a lot of confounds because these babies are all different in terms of also like possible pathology that I have. Of course, we are excluding from the beginning all those that have some pathology that we know before birth, but we are trying to enroll all the other babies that are born and we don't know yet. So yeah, we are trying to keep everything in consideration. So we have like a really big Excel file with all of the clinical things that happen to the baby. And the final aim is when we will build a statistical model that will take everything into consideration, also to keep all these factors into consideration in the statistical model. So yeah.
Last minute questions. Thank you. Ahmed? Not working. No? We have lost the mouse. Yeah. Okay. Five.
Good afternoon, everyone. It's my pleasure to be talking to you today, and I am one of the few clinicians in the room, and it's really an honor to be listening to all the work that you guys have been doing. So my talk today is about early cerebral oxygen saturation and brain injury in extremely preterm infants. This work has been published really just a couple of months ago, and I acknowledge all my collaborators and co-authors in this work. And I just have a couple of disclosures, which is not relevant to my talk today. So prematurity continues to be a big problem that we face in our world. And prematurity is really the major cause of death in the first 1 month of life. In the US, 1.5% of infants born every year are less than 30 weeks gestation. We know that the preterm brain is very vulnerable to injury. And I want to focus on one area of the brain So although as you see in this graph, by increasing gestation age, there's increase in the size of the brain and increase in the cortical folding, there is a structure called the germinal matrix that continues to grow significantly, usually until 23 weeks. And then by 28 weeks actually start to shrink in size. And this germinal matrix is very important. It is a highly vascularized area in the ventrolateral surface of the lateral ventricles that's responsible for production of neuronal precursors between 10 to 20 weeks, and then also contribute largely to the production of glial elements. And there are, this germinal matrix is at high risk to bleed due to intravascular, vascular, and extravascular factors. We really believe that the intravascular factors contributing to hemorrhage are the most common, and this include really causes that increase cerebral blood flow, decrease cerebral blood flow, and more importantly, fluctuation in cerebral blood flow. So this diagram just shows like the multiple factors that cause decreased cerebral perfusion early on when babies are born prematurely. And usually in the first few days of life, actually there is more like increased cerebral perfusion. And with this decrease in cerebral perfusion and increase, these preterm babies are at higher risk to develop germinal matrix intraventricular hemorrhage. And as we talked today in multiple sessions, one of the reasons why these babies are at higher risk because they usually don't have good autoregulation and they have more like passive circulation. So we typically classify hemorrhage as 4 grades. So the first grade, the one you would see on the left, this is just you have bleeding where the germinal matrix is. When blood goes inside the ventricles, that's what we call grade 2 or intraventricular hemorrhage. And as you see in the graph to the right, when this is a huge amount of blood distending the ventricles, this is what we classify as grade 3. And what we used to call grade 4 is an area of periventricular hemorrhagic infarction. And this happens mainly when you have large amount of blood in the ventricles, causing venous congestion and infarction surrounding it. And really the rate of severe IVH, when they look back for the last 2 decades, has not declined that much in spite of improvement in prenatal and perinatal care. And very recent data tried to look at the relation between the grade of IVH and long-term neurodevelopmental outcome. And as you see, if you don't have a bleed, and in really this extremely preterm infants, your risk of having neurodevelopmental impairments is like 13%. If you have the highest grade of bleed, that can go close to 80%. So it's really correlating to the long-term neurodevelopmental outcome. So how can NIRS help this dilemma? Can NIRS really help us to predict which baby will have a bleed and maybe do something that actually can prevent hemorrhage? We know that there are previous research that looked at the relation of cerebral saturation. Since cerebral saturation is a clinical tool we have, we're very interested to use it. And we think that it is a surrogate for cerebral blood flow, presuming that you have a stable oxygen content and extraction. So some studies did show that there's actually increased cerebral saturation in the first few hours of life to be associated with developing of germinal matrix intraventricular hemorrhage, while other studies have shown decreased cerebral saturation, and this continues even like up to 2 months of age following development of hemorrhage. So the aim of the study is really to look at our clinical data. As a background, we have been using near-infrared spectroscopy in our unit in the neonatal ICU at the Brigham and Women's Hospital NICU since 2018. So all babies less than 28 weeks get NIRS monitoring for the first 72 hours of life. And the aim of this study was just to go back and see, can we have an association between change in cerebral saturation the first few days of life with the presence, severity, and timing of IVH in extremely preterm infants. And we also tried to explore the relation between cerebral saturation and brain injury score on term equivalent brain MRI. As I mentioned, we have a very detailed clinical protocol to use NIRS in our population. So this was a retrospective study. We looked at data between January 2018 to August 2020. Infants were less than 28 weeks, and we used the first 108 hours of NIRS recording. We use Infos device as our source for cerebral saturation. And we did use some analytic work by our collaborators in the at MGH to help us to clean this data that was recorded clinically. And this data cleaning ending by rejecting an average of 5.4% of all data. We also use a clinical cranial ultrasound. We have a more or less detailed clinical ultrasound protocol that we do it day 1, day 3, and day 7 in all these babies. For the purpose of this study, we try to compare the cerebral saturation between babies who have a bleed or no bleed. We also try to look at the different cerebral saturation according to the severity of hemorrhage. So we defined grade 1 and 2 as mild and grade 3 and 4 as severe. We also tried to look at a subgroup where we had 2 ultrasound, one done in the first day and one at day 3. And we looked at those who have a bleed that developed in the first ultrasound, and another group who did not have a bleed and then had a bleed at the follow-up ultrasound. And then we also used our term equivalent MRI, which we use routinely in this population, and we use a Kido-Core scoring, which takes in account white matter, gray matter, as well as cerebellar injury. And we classified babies according to the Kido-Core score into 4 groups. So our we used the descriptive statistics. To the way we dealt with the cerebral saturation, we actually got the median saturation for each hour for each infant. And then we calculated the median saturation in 9 12-hour intervals. And these are the intervals you'll be seeing in the results. So, and we modeled the association using linear mixed regression to take into account repeated measures within subjects nested within intrafamiliar clusters. And we adjusted our model to potential confounders, mainly gestation age at birth, birth weight Z-score, baseline hematocrit, base deficit, and PCO2. And in our trajectories, which I show in the graph, we use the least square means and 95th confidence interval. And we try to create a ROC analysis curve to assess the predictive value. We just look at the demographic and clinical data. We saw some difference between those who have IVH and no IVH, as expected to be lower gestation age, have less hematocrit, more likely to be intubated, have more likely to have sepsis and retinopathy of prematurity. These are very well expected for the sicker babies who develop a bleed. Looking at the cerebral saturation in this graph, and you can see that babies who have no IVH in orange and those who have IVH in blue, and you can see that the cerebral saturation started almost very similar. And at about 36 hours of life, you can see that those who have a bleed have a divergent drop in their cerebral saturation, which was statistically significant. When we adjusted this to all the confounders I just mentioned, we could see the same trend. The divergence happened in about 48 hours, and you can see that it's mostly still statistically significant. And we try to create like a ROC curve to see is there a cutoff at 36 hours that can predict the presence of hemorrhage? So at a cutoff of 73%, we got just a sensitivity of 75%, that specificity of 6%. When we tried to look at different grades of hemorrhage, we didn't really see a difference between those who have just mild IVH. In this case, these are the ones in blue versus those who have severe IVH in red. So both of them differed from those who don't have IVH, but there was no difference between the subgroups. Also, when we tried to look at early and late hemorrhage, we didn't see a big difference except in the first 24 hours, where there is a tendency for those who have early hemorrhage to have a higher cerebral saturation. And when we looked at the term equivalent MRI scores, as you see in this graph, we didn't see a difference for those who have normal, mild, moderate, or severe injury on MRI. And the question is, why do we have low cerebral saturation in those who have IVH? It's not very well defined, but this could be related to low cardiac output early on. It could be related to a hemodynamically significant duct after the first few days of life. Could be related to altered brain metabolism in injured tissue, leading to increased oxygen extraction, or altered region of blood flow in cortical or subcortical regions in presence of hemorrhage. We try to control for associated anemia by adding it as a confounder. We acknowledge there are some limitations. This is a retrospective study. However, we think one of the strengths that we used clinical data that has been collected clinically at the bedside. We don't have very consistent PDA data or patent ductal self-users data. Although we're doing ultrasound, we didn't have like an ultrasound at birth to know which baby had a bleed to start with. And we did not collect simultaneous oxygen saturation to calculate oxygen extraction. And we do not have long-term follow-up. In summary, we have shown that babies who have germinal matrix interventricular hemorrhage exhibit divergent RSO2 trajectories in the first few days of life when compared to those who don't have a hemorrhage. And in those who have hemorrhage, this decline occurred after the second day of life. And we propose that non-invasive monitoring of cerebral saturation using NIRS might provide useful bedside tool to monitor the development of germinal matrix hemorrhage, and possibly in the future can assist us in developing interventions aiming to prevent IVH. Thank you, everyone.
Okay, yeah. I have one question. So impactful results are very clinically useful, but I noticed it's probably one channel NIRS device, and we've seen a lot of multi-channels, laterality stuff. Is it even meaningful in these young babies in terms of looking at different spaces, and is it even possible or practically NICU environment?
Thank you, John. And I would say we have not technically used this. I think what we're trying to use now is the technology with our collaboration with your lab and Mari's lab to use diffusion correlation spectroscopy as a more accurate way of looking at cerebral perfusion in general and cerebral blood flow, which we think is very much contributing to the pathogenesis. My guess, hopefully it will help, I think, to use it in this population more to look at regional changes of injury. I don't know it will be 100% helpful immediately after birth. I think it will be helpful more as follow-up and as we monitor these babies and monitor the effect of injury on their different or differential development and perfusion to different areas of the brain. At least this, I think, would be more likely to be more helpful.
Yeah, thanks for the very interesting talk. I was interested that when you compared like the mild bleed to the severe bleed, you didn't really see much of a difference in the NIRS. And was that expected to you, or do you know why that might be?
My guess is more related to the numbers. The numbers were very small, and it ended by really having just much less number in the mild, like when you divide them into subgroups. That will be my guess. I think there are older data that did show a direct relation between severity of hemorrhage and the reduction in cerebral saturation. So that was my only thought. Maybe if we have a good sample size, we will be able to demonstrate this.
Okay, thanks. Let's thank the speaker again. Thank you. Okay, the next speaker will be Felix Schokman online. So should we, yep, can you switch on the Zoom? Yep. Okay, all right. So please go ahead.
Yes, can you hear me? Yeah, thank you very much for the introduction. See. About 7% of the neonates here in Switzerland are born premature. And this is about 10% of the babies in America. And we at the neonatology department at the University Hospital of Zurich, I have a great experience in treating these neonates and also performing NIRS studies using NIRS oximetry and fNIRS. And it has been already discussed that a huge problem is that the autonomic nervous system and the CNS is not well developed in preterm neonates, making it possible that brain damages can occur. And we at the University Hospital at Zurich, we have one new method to somehow calm the neonates and to prevent an additional stress that the preterms experience in the neonatology. And this method is nothing else than creative music therapy. So a music therapist is humming or singing in front of the neonate, with the neonate, and also with the mother. And this is a procedure that lasts about 15, 20 minutes, in some instances also longer. And according to the observations, it really reduces the stress on the neonate. It calms it down and reduces also problems in breathing, for example. And one question we had was, how is the effect, or what is the effect of this therapy on the cerebrovascular oxygen saturation and the hemodynamics? And of course, NIRS is a good method to investigate this question. So we did do a study with 20 preterm born neonates. They were clinically stable, and we optimized the whole setup so that all these measurement devices and the whole procedure was not causing additional stress to the neonate. It was not so easy to do this optimization because you have to really ensure that all these measurements and the procedure is not disturbing the neonate. But we finally had a good protocol, and we were successfully measuring 20 neonates. We used a specific NIRS oximeter that was developed by our laboratory and is now commercially available by a spin-off company, Oxyprem. The device consists of 2 light detectors and 2 light sources and 4 different wavelengths. It is a self-calibrating multi-distance measurement device that can measure absolute values of StO2 and also relative changes in oxy and deoxyhemoglobin. We measured at the auditory cortex and the prefrontal cortex. And these are the results for one neonate. As you can see, there was a strong increase in the tissue oxygenation during the first block of singing, which was 10 minutes long. And in the second block, this increase was not so markedly visible. We observed nearly similar changes in the auditory cortex and in the prefrontal cortex. There was a drop in the fractional tissue oxygen extraction in the auditory cortex as well as in the prefrontal cortex. There was an increase in the arterial oxygenation, especially in the first part of this music therapy, and also an increase in the pulse rate, but a quite steep one, which declined then after the second phase. So we observed an interesting phenomena. All these changes were quite different between the subjects, between the neonates, and we could classify the different reactivity types into two subgroups. The first group consisted of 10 neonates, and they showed an increase in the tissue oxygenation in the auditory cortex during the first singing interval. The second group showed a decrease in StO2 in the auditory cortex during the first singing interval. And now we investigated if these two groups are somehow different with regard to changes in systemic physiology or other parameters. So the first interesting thing was for me, of course, to look if a change in systemic physiology was different between the two groups. And I looked at the changes of arterial oxygenation measured with pulse oximetry, but the changes were not different. The pulse rate increase or decrease was also not different. But what was different was the hematocrit. The hematocrit as the volume, the percentage volume of red blood cells in the blood was different or was related to the magnitude and the sign of the hemodynamic responses we measured. And surprisingly, also the height of the neonate was correlated with the changes in the cerebrovascular tissue saturation measured at the auditory and prefrontal cortex. This is visualized in the correlation plots here at the bottom. Then I investigated if there are other correlations, and I found that there's also the variable sex that was different. So in subgroup 1, we had more female neonates, and in subgroup 2, with the negative response, we had significantly more male neonates. And this was the only parameter, the demographic parameter, distinguishing these two groups. In the next step, I looked if the time-dependent changes of the NIRS parameters were correlated with changes in systemic physiology. And therefore, I used a generalized additive model, which is basically a generalized form of a GLM with the possibility to fit nonlinear function in an unsupervised way. And this is the result of the fitting. So I could see that in subgroup 1, with the positive response, there was a positive relationship between StO2 and the arterial oxygenation, and a nonlinear relationship between StO2 and the pulse rate. However, in the subgroup 2, there was a positive relationship between StO2 and the pulse rate, and almost no correlation with the arterial oxygenation. So the coupling between the NIRS-related measured parameters and the systemic physiology was also different between the two groups. And this is somehow also related to a study we conducted a few years ago. In this study, we used visual stimulation and also investigated the hemodynamic responses at the prefrontal cortex and the visual cortex. And we observed that there are also different types of hemodynamic responses. And we could classify these hemodynamic responses into 3 groups. We could see that in some of the neonates, there was a clearly positive an increase in oxyhemoglobin at the visual cortex. In some, there was a clear decrease in the oxyhemoglobin. And in another group, there was almost nothing happening during the visual stimulation. And in this study, we could find that the 3 groups were related to the weight of the neonate at the measurement. So this second study supports also the notion that there are some subgroups, and it's maybe not a good idea to average over the whole group, but to look at different patterns that emerge, especially when you do fNIRS with neonates and especially preterm neonates. So our study, it demonstrates the impact of creative music therapy on the neonates' physiology. And it highlights also the need to analyze fNIRS measurements in neonates separately, depending on the response pattern, to avoid erroneous conclusions when, for example, only determining the group average of the signal changes. And with this, I'm at the end, and thanks for the attention.
Thank you. Questions for Felix? Please use the microphones. No, please. All right. You start then, Ilias.
I start. Hello, thanks for the talk. I have just a basic question about what is music therapy?
Well, music therapy in this context is a person that is in front of the neonate, is holding the neonate and, for example, singing or humming, or is using an instrument to produce tones, music during a specific time frame, for example, 20 minutes.
Okay, and another question. Is the singing somewhat systemic? And is this controlled? For example, if the mother sings that specific song, or if this song has been heard by the baby, or I'm wondering if some difference between groups are maybe explained by these more environmental factors.
Sure, yeah, no, we control for this. It's always the same song, basically, or the same melody, and it's quite an easy and abstract melody that is not related to any well-known music. So it's more a humming sound that that is not related to a music that, for example, the neonate was exposed by other sources, like a radio or something like this.
Okay, thank you.
Yes.
Thanks, Felix. An excellent talk. It's good to see you as well online. Two questions. So the first one kind of relates to the previous question. Where is the mom in all this sort of protocol? Is the mom holding the baby, or somebody else holding the baby, or have you control on that? And the second question comes to the oxy and deoxy. Have you looked the oxy and deoxy in the music therapy? In particular, have you looked at the correlation between oxy and deoxy during the music therapy?
Regarding the first question, the position of the mother was always controlled. So the mother was at the same position. The mother was not holding the neonate. The music therapist was touching the neonate during the whole session. And with regard to the second question, we only looked at the StO2 and the total hemoglobin for this analysis. But of course, there are many information in the data, and the coupling between oxy and deoxyhemoglobin is another thing. Also to look at, for example, the coupling between the oxy and deoxyhemoglobin in specific frequency bands would be interesting. So, yeah.
More to do. Thanks, Felix.
Cheers. Fabrice?
Yeah. Bonjour. Do you have any differences between the age? Because I think the age range is 32 to 36 weeks gestational age. And do you have any difference between the youngest one and the oldest one?
Yeah, this was the first one that we investigated, because I was expecting that the age is the main factor, for example, explaining the two groups. But in this case, it was not statistically significant.
Yeah. And did you check also for the sleep stage? I mean, or something like this, or when you did the recording, or?
Good point. We had this as an additional parameter that we tracked, and in most of the cases, the babies were asleep. So in instances where the baby was in an active sleep stage, there were always often there were movement artifacts. So I had to exclude these time periods for the analysis. So at the end, I had data that were mostly covering phases where the neonates were really sleeping in a quiet way.
Okay, merci.
Thank you. If there are no other questions, I would like to invite the next speaker. Julie Uchitel from University of Cambridge.
Thank you. Okay.
Just wait for the slide.
Oh, sure. Yeah. Fine.
Perfect.
Okay. Yeah. Okay. Hi, everyone. My name is Julie Uchitel, and I'm coming from Department of Pediatrics, University of Cambridge, and UCL.Hub. Today, I'd like to talk to you a bit about functional imaging the preterm brain with combined high-density diffuse optical tomography and EEG. So, in the ideal scenario, an infant is born at term, and this contributes to them having healthy sleep development. In this case, they're sleeping at home and able to have protected sleep, which in turn contributes to healthy brain development. At the same time, it's been shown that term birth is independently associated with healthy brain development. And altogether, you can probably understand how these three factors contribute to healthy neurodevelopmental outcomes. In another scenario, though, the preterm infant is born early, where they spend the earliest days of their life in neonatal intensive care units, where changing light and sound levels can interrupt sleep patterns. It has been shown that these interrupted sleep patterns are associated with altered brain development in preterm infants relative to their term-born counterparts. And at the same time, preterm birth is independently associated with altered brain development. And then we can understand how, once again, these three contribute to impaired neurodevelopmental outcomes in these infants. It's also, this is the great question that the previous person asked. Sorry, I'm missing your name, but about sleep states, because it's very relevant to this talk as well, in which we specifically looked at sleep states and differences in functional connectivity across sleep states. So for a bit of background for the audience, sleep states in newborn infants are associated with different patterns of EEG brain activity. And these two sleep states, active sleep and quiet sleep, can be comparable to REM sleep and non-REM sleep in adults. So when I say different EEG patterns on the top, you see here, it's an AEG trace, and it shows you sleep-wake cycling. And on right below that, it'll show you the raw traces of what you'd expect to see during active sleep and quiet sleep. Active sleep is continuous EEG patterns, and quiet sleep is what they call tracé alternant. At the same time, these two states are also associated with different behaviors that change across gestation. So in earlier preterm infants, we tend not to see the expression of behaviors, but in older infants, we tend to see in active sleep, they're moving around a lot, they have rapid eye movements, quiet sleep, they're completely still. So in a previous study, now under review, the LUMO sleep study, we performed cot-side imaging of functional connectivity networks using a wearable HD DOT system known as LUMO. If you popped in over next door, you may have seen the LUMO booth, which is the LUMO system is modular in design, allowing for improved wearability. Each tile comprises 4 detectors and 3 light sources. And for studying newborn infants, we developed a neonatal specific headgear comprised of 12 tiles, providing us with 36 light sources and 48 detectors for about overall 400 viable channels for imaging. So this study was actually conducted during COVID, cot-side, as in very much cot-side in the maternity ward, not in any kind of scanning room or imaging facility for experiments. And a lot of adaptations were also needed to be made for the sake of doing this during COVID. In our analyses of these data, one type of analysis we did was a seed-based correlation, which is you are probably familiar with. You pick a seed, calculate the correlation between that seed region and all the other regions of the brain. Doing that for the motor cortex, we found relatively robust maps of correlation down the motor cortex and on the contralateral hemisphere. And then we also used another technique called connectome-based independent component analysis, which we use to detect statistically significant differences in connections between active sleep and quiet sleep. And for one of the networks that demonstrated intrahemispheric connectivity, we found stronger connectivity, intrahemispheric connectivity during active sleep as compared to quiet sleep. And I should also note here that for scoring active sleep and quiet sleep in these babies, we were able to use just behavior alone because it's, as I mentioned before, very robust in term-born infants. So we then aim to adapt the system to preterm infants to study the next aim, which was how does this process become impaired in these infants? We developed a 9-tile system, same HD-DOT system as before, but instead of 12 tiles, 9 tiles, developed a preterm specific headgear solution for this. And up top here, you can see the headgear on a doll, to the right on an infant. And then on the bottom left, you see the sensitivity profile of this array. In theory, you can add more tiles to the back of the head, but for here, our purposes are to compare the same regions between term and preterm infants. So that's why we put the coverage as that. Okay, so as just initial validation that this 9-tile system was working, because we made it from a previous 12-tile system, just looking at the log drop-off curve, you see that we are able to keep a really good number of channels across different distances, and we're also getting really deep, or rather very long channels. So looking on the right, you see we have a good number of 50, 55, and even some 60 millimeter channels that we're keeping. And then here's an example of raw data, just to give you an idea of it. I think it's really helpful to see. And the spike here, the spikes are motion artifact. Motion artifact when the baby's asleep. Why? Because we have periods of active sleep when they move around, or if there's movement or a sound in the environment, they may startle. So highlighted here in light blue is a period of what we detected as motion artifact free data. If you're using HMR motion artifact in the Homer toolbox, that's what we use to find this here. And it's a typical example of what we would use for our data processing in further steps. Okay, so EEG integration, coming back to why did we bother to put EEG into the system if we were able to use HD-OT in older infants? Again, it comes back to we wanted to look at sleep states specifically. And in preterm infants, you can only detect the differences between the two states using EEG. You can't see it from the behavior, it's just not clear. So we used 5 EEG channels spread across the more frontal regions to be able to have enough that we could actually see the traces we wanted. And then to see what it looks like, combined system. The colored wires that you see here are EEG electrodes, and the HD-DOT cap is on the infant. On the right image, you can see the EEG amplifier in the back with the cot in the infant's cot. Okay, and to give you a more bird's eye view of what we're doing, this was in Cambridge on the neonatal intensive care unit. On the bottom left, you can see the HD-DOT EEG headgear on the preterm infant while they're in their cot. On the trolley, you have the HD-DOT system, a recording laptop, an EEG system, also with a video camera. And so what was really nice about this was setup took approximately 25 minutes, the longer part just being really from EEG. For those of you that experience with EEG, putting on electrodes and putting on pace can take a really long time and getting the impedance to where you want it. So without EEG, bringing Lumo and setting it up on the NICU takes about 5 to 10 minutes. So very convenient for working in these kind of settings where time is of the essence between feeds and medical procedures. Okay, so as a preliminary check of are we actually getting what we want to be getting from EEG and from HD-DOT, we first looked at the EEG data and said, okay, do we see sleep states? The answer is yes. Top image is a screenshot of the EEG trace during active sleep. So the signal there is continuous, which is an indicator of active sleep in the infant. And the infant's eyes were closed and they had fast breathing and rapid eye movements. In the bottom trace, you see, or in the bottom image, you see trace alternate, which was with stillness in the infant, no eye movements, and slow, even breath. Okay, so EEG, check. Moving on to HD-DOT, are we getting what we actually want to get? So we constructed functional connectivity matrices, looking at the correlations between all nodes in the DOT reconstructed data. And we see HBO, and HBR correlations, good to go. Okay, so then we did, like we did for the preterm infants, seed-based correlation maps, looking at specifically this matter motor region, and then also the prefrontal region. These two seeds we used in the term infants. For the motor seeds, they look relatively robust, seeing correlation down the length of the motor cortex, and then on the contralateral motor cortex for HBO and HBR. And for the prefrontal cortex, we see contralateral correlation relatively well for HBO and not as much for HBR, but still correlation restricted to the regions surrounding the seed. Okay, so what are we doing next? So far, the total number of infants we've only studied was 6. I should have mentioned that those seed-based maps were actually only based on data from 3 infants. So getting maps like that from 3 infants is pretty good. And we're also hoping to do repeated measures of our infants. So studying infants at 1 week apart to kind of create a developmental profile of what the resting state functional connectivity networks look like for these infants. Questions we're hoping to answer with this project as it's in its infancy. that was bad joke. Hoping to figure out how to best make comparisons to term infants. Also doing the question of how do we manage these thousands of nodes in the gray matter surface mesh space. In the term infants, we used a cortical parcellation approach. Unsure if we're able to do that here because there are currently no fMRI-based cortical parcellation atlases for preterm infants. Motion artifact is also a big consideration, as for all fNIRS studies. Structural priors in our data here, we used one atlas, one MRI head model atlas for all infants that was based on their average gestational age and average head circumference. But would it be better to use something that's head circumference matched, 10-20 point matched? And then also, how do we think about relating functional connectivity network features to neurodevelopmental outcomes? So, yes, thank you. So none of this would be possible without the amazing team at the NeoLab, Dot Hub, and Cambridge, my PhD supervisors, Rob Cooper and Tuppen Austin. And I'm happy to take any questions.
We can take a few, please.
Thanks for the great talk. I was wondering, like it's been shown in adults that the spectral component of the NIRS signal during sleep is changing a lot between the different sleep stages. I was wondering if you tried that on your 2 stages here. What was the spectral component and how this could impact the connectivity after?
That's a really great point. Thank you for that question. We haven't specifically looked at spectral components, and I can imagine that this would fluctuate between the states. But given that we're dividing our data into active sleep and quiet sleep, and then doing direct statistical comparisons between those data segments, we won't be able to see those kind of more fluctuation trends, but it would be really interesting to look at that. Thank you.
Go, please.
Okay, so nice work. So I was just wondering, since you can actually put your seed in literally hundreds of places, would actually be a preferable network, resting state network that you were looking for? Because I noticed that you actually showed the sensory model network the most. Would that be kind of the one that you actually would aim? Would that, I mean, I guess, is there any preferred network that
Yeah.
we should look at?
Yeah, that's a really great question. From what I would assume from that is that we'd probably want to go for visual and motor networks, just because these are the two earliest to develop in utero. So if we're looking at preterm infants and we want to make comparison to term infants, we probably want to grab networks that are going to be sure are going to be there in the earliest or the youngest infants possible. Frontal was chosen here because our infants were were born at 30 to 35 weeks gestation. So that emerges by then. But if we went lower to like 26 to 30, I would probably say let's go for motor and visual. Thank you.
Okay, let's thank the speaker again. Thank you. Our next speaker and the last for today is Frederick Lange.
Oh, yeah. Okay.
Sorry, there was some technical difficulties. I was allowed to use my computer. I'm cheating, sorry. Great. Thank you very much for the introduction, and thank you everybody to staying for the last session. Today I'm going to talk about the relationship between the cerebral oxygen metabolism and the oxidative state of cytochrome C oxidase in neonatal encephalopathy. So birth asphyxia or hypoxic ischemic encephalopathy is a significant burden as it occurs in 1 to 3 per 1,000 births in developed countries, and it accounts for up to 30% of children developing cerebral palsy. So in order to try to limit the effect of the HE on the brain development, there's a need of a courtside continuous cerebral monitoring as early as possible in order to rapidly adapt the care and preserve the brain tissues. So in order to do that, our group has been specialized in developing broadband NIRS instrument, as Mamadou has presented, sometimes called hyperspectral NIRS. And so as he presented, this type of NIRS allows us to retrieve the traditional hemoglobin changes parameters together with absolute values of brain tissues oxygen saturation via the broadband fitting that he explained, and also having access to the oxidative state of cytochrome C oxidase, which will be an indicator of the mitochondrial function and therefore the cerebral metabolism. So we have shown the clinical relevance of that particular biomarker in the clinic, as the changes in cytochrome are significantly correlated with magnetic resonance spectroscopy, which is the gold standard, and it's also associated with the injury severity of these neonates. If you want also to get some information about the blood flow, you can use the diffuse correlation spectroscopy technique. And it has also been applied in combination with NIRS, so either time domain or frequency domain, in order to get information both about the cerebral oxygenation and the blood flow. So what we propose is to pair the BNIRS and the DCS in order to get a more complete picture of the brain status of these babies by having access to the cerebral blood flow, cerebral oxygenation, and cerebral metabolism. One of the nice possibilities that we have combining this technique is also to be able to calculate the CMRO2 index by combining the data from the BNIRS and the DCS, which is also a marker of the brain metabolism and the oxygen consumption by the brain. And we think that combining this with the cytochrome signal can help us to better understand this oxy-CO contrast, which is quite complicated to explain physiologically. And actually, there's some very recent work by Keith Lawrence lab who have looked at the correlation between the CMRO2 and the oxy-CO event and the oxy-CO signal in preclinical model, looking at various events like ischemia. And so we wanted to investigate the potential of using the coupling between the CMR2 and the cytochrome as a biomarker of brain injury. So to do that, we used our Florence device, so that combines the BNIRS and the DCS. I won't go into much detail, but we have our BNIRS part here, which allows us to acquire spectra. And we have our DCS device with a 785 nanometer laser. You can see the physical dimension of the equipment here with the BNIRS part, the DCS, and the electrical thing. And so for the recording, we have only one channel with that system. We've got one BNIRS source, one DCN source, and we have a detector bundle that combines the BNIRS and the DCS and that redirect the signal to the relevant equipment. And so as the DCS wavelength is right in the middle of our detection bandwidths for the BNIRS, what we do is that we do a sequential measurement. So we turn the BNIRS on first and then the DCS, etc., to avoid any crosstalk between the modalities. Regarding the post-processing, we first perform the broadband fitting that give us the information about the optical properties of the tissue, and so we can extract the STO2 values. Then we calculate the changes in our concentration using the modified BLM below. Then we fit the DCS data using the optical properties that we calculated in the broadband fitting. And we integrate the STO2 and the BFI to calculate the CMRO2 index. And then we time synchronize everything. So this is the setup in the NICU. So we try to monitor the babies for several hours each day for the first days of life. So you can see our optical equipment here. We also get access to the systemic data. We can also eventually have access to the EEG data. And you can see a picture here of our babies with our optical probe on, and you can see the EEG wires here. And on top of that, we will also get the MRI information that will be acquired a few days after that. So now I'm going to show you some recordings that we've done with this system. So this here on the top, you're going to see the oxygenation recording with the STO2 in red and SaO2 in blue. You got the BFI in black and CMR2 in red here, and the cytochrome here. So here I call it a stable recording because we couldn't identify any drastic physiological event during that recording. The SaO2 stayed stable. Despite that, we could see some fluctuation in our parameters in STO2, to some extent in the BFI and CMR2 and to the OxyCO, but there was no drastic event, as you will see later on. So what we tried to do is to look at the correlation between these variables during this stable period. And so what we've seen is that we have a good correlation between the CMRO2 and the BFI and the CMRO2 and the STO2. However, the correlation between the CMRO2 and the OxyCO and the BFI and the OxyCO was not present during that relatively stable period. So you can see the correlation values here for the whole recording. So in stable measurement, we've got coupling between BFI, STO2, and CMRO2, but we don't have any coupling between OxyCO, BFI, and CMRO2. And now I'm going to spend most of the last minutes that I have to guide you through this particular recording, which is quite full of events. So it's a recording of that lasted 3.5 hours. It was on a term baby with HAE that developed a lot of seizures. So what you can see on the top here is the heart rate values. Here you can see the oxygenation values, and here you can see cytochrome, CMRO2, and BFI. These are all relative changes from the baseline, so these are all deltas. I show you here the absolute values for the heart rate and the oxygenation value. So at the start of the recording, we had normal physiological values. And then there's three main period in that recording. The first one is extensive seizures. The second one is hypoxic event, and the third one is again severe seizures. Then we had a recovery to the baseline in the last half an hour or so. So I'm going to guide you through these three particular events. So here is the first seizure periods. So what I have to stress here is that we could identify these seizures live on the EEG. However, because the EEG is part of the clinical data set, it takes longer for us to get hold on the raw data. So I couldn't process that data yet. But we've identified this period. And we also know that during prolonged seizures, there's a significant increase in the heart rate, which is quite obvious here. And so it's going to be a good indication of where the seizures are in that data set. And so if we look at our oxygenation parameters, we see that with the seizures, there's an increase in the STO2. The SCO2 tend to decrease also. And for the BFI, CMRO2, and OxyCO, you see that there's an increase in the baseline value as the seizures keep happening and happening. And you can see some nice correlation between the signals here. So here I'm going to focus on one of them so it's a bit easier to see. Again, the data are changes and normalized to the beginning of that event. And so what we can see here is the clear increase in the STO2, the relative decrease in SCO2. And what we see here is a typical neurovascular coupling behavior. So you see the increase in the CMRO2 at first, that drives the increase in the oxidation of cytochrome, which, so this marks the increase of the demand. And then you've got the increase in the blood flow that overshoots that demand. And so as there's more oxygen than needed, the CMRO2 goes down again. So that's typical neurovascular coupling. That's what we've seen across this first period of seizure. And this is how the values correlate. So then after that period, there's been a hypoxic event, which is likely to have been driven by lumbar puncture. So we know that this lumbar puncture can induce some hypoxic event in these babies because they are manipulated and their lung can be compressed during that procedure. And so that's what we see in that recording. So you get the decrease in tissue in arterial saturation. So yeah, it goes down to about 85%. It's matched with the decrease in the STO2. And for the other parameters, we see the typical response with like the increase in CMRO2, which is not met by the increase in BFI. So the BFI stays stable. Therefore, we have a reduction in our mitochondrial function. So it's a typical behavior of the hypoxic event. And then there's the last period. So again, a very big seizure. You can see the very high increase in the heart rate here. Again, normalized to the beginning of that period. You can see here, interestingly, that the SAO2 goes up, the STO2 goes down. And there's actually mixed response to our other signals. So at first you get your increase in CMRO2, increase in BFI, and increase in oxidation. But then the increase in oxidation stays up, but BFIs go down here. And the CMRO2 keeps climbing, which at the end of this event is not followed by the cytochrome. So there's a reduction here. And here the CMRO2 increases again, but the cytochrome doesn't respond and go back to being reduced. So we think that this clearly marks an increase in demand that is not met by the supply. Yeah. So what we've done here is we've done our correlation between cytochrome and CMRO2 over 10 minutes of data with a sliding window. And here you can see what we see. So during the first seizure period, you see an increase in the correlation between these two variables. And then during the hypoxia, it goes down again. And there's a negative correlation during the period where we think the demand is not met. And then when at the last part of the recording, it goes back to the normal state. So in conclusion, we have developed a BNS DCS instrument suitable for clinical use, able to monitor parameters related to cerebral oxygenation, blood flow, and metabolism. We've been able to use it in the NICU, and being able to have access to a highly multidimensional data allows us to study the effect of pathophysiological events on the brain metabolism more clearly, because we have this combination between CMR2 and cytochrome. And we think that the coupling between CMR2 and OxyCO is a potential biomarker of the brain injury. As you can see here, with an uncoupling when there's no particular change, when we see an increase in the positive correlation, positive coupling, we think that it's an increase in demand that is met, and a negative coupling, an increase in demand that is not met. So we think that is an exciting biomarker. And with that, I'd like to thank the team, particularly the doctors that helped to collect the data, so Kelly, Vinita, and Turgut, and ICFO and Turgut for the DCS help, the funders, and I thank you for your attention.
Questions?
Hello. Yeah, thank you for your talk. I was just wondering, with the different like events that happened, I think you said you took like a baseline like right at the start of the event.
Yeah.
How long was that baseline? And did you like, because it's on the order of like,
Yeah, so basically the changes that I was showing, I've renormalized it just to show it better. So at first I took a 10-second baseline at the beginning, and then just for the sake of showing the different events, I've renormalized it. But I think you shouldn't do that in your actual data set when you want to analyze the data. You should compare it to a like baseline period that doesn't change at all.
Okay, and did you experiment with like increase, like changing the length of the baseline, like given like the length of the data?
For what, for the correlation?
Yeah, just for when you're like showing the changes in the different parameters.
Oh, well, I tend to try to take a baseline that doesn't change. So at the very beginning of the recording, it was stable for minutes, so it was okay. But yeah, again, as I said, it's just for showing the changes but to analyze the data, you shouldn't renormalize to each event.
Okay. And did you have any problems with like motion artifacts over the long?
Yeah, I think on one of the traces that I showed especially during the lumbar puncture, there's a sudden drop, which I think is a motion artifact, as the baby was quite shaken a lot during that procedure. Yeah. We have a relatively good contact, so we don't have that many, but yeah, sometimes it's clinical data. So sometimes you have artifacts, and actually for clinical data, it's quite difficult to correct because it's not like your typical fNIRS where you can check that. We could use accelerometers actually, that could be a potential, but we don't have that much problem with artifact at the moment.
Okay, thank you.
Thank you.
One last quick question and
merci for your presentation. I have a question. In the second session, I mean, you have an increase in the oxygen saturation, peripheral oxygen saturation. And so this suppose that before the seizure, it was the child was in hypoxia, in cerebral hypoxia, or I mean peripheral hypoxia, because the SAT was you cannot increase the SAT more than 100%.
Yeah.
And so, do you think that the state of the oxygenation state before the seizure might interact with the CMRO2 value and with the CCO value, and might be one of the reasons why the change are different from the previous, I mean, the first seizure?
It could be potentially. Plus, during that recording, there's been some drug administered as well. So I'm not quite sure about the effect of that. We need to have some chats with the clinical team. But yeah, the increase here, basically we go to 97 and basically completely saturate for the SaO2. So indeed, that could affect the CMRO2 as you said, we can go higher than 100%. And the way that we calculate SMRO2 here, it's heavily, it's dependent on your two main parameters like saturation, tissue, and arterial. So if there's some artifacts or some weird events in the arterial, then it's going to affect our CMRO2. So that's possible, but I think for this data set, the changes are genuine. But I'm happy to discuss it more.
Yeah,
maybe.
Merci.
Thank you.
Okay, thank you for everyone. This concludes our session and the evening. Thanks for coming. Bye.